Good morning and welcome to day 5 of the AMALathon. Thanks to everyone that's joined me this morning. Let's get started. So what we're going to cover today is topic 3 biology. We're going to finish off topic 3 biology. Then we're going to finish off topic 4 biology in our normal session at 10 a.m. Then at 11 a.m. we're going to continue and finish off topic 4 chemistry, and then hopefully at 12 o'clock in our last session we'll be finishing off topic 4 physics. That means we'll have finished off biology and we'll have finished off physics. So tomorrow's session will just be finishing off whatever needs to be completed. So there's topic 5 chemistry that needs to be finished, and it's possible we might not finish all of the topic 4 biology and physics as well. So tomorrow's session might not be as long, and most likely we'll start at 10 o'clock. Tomorrow's session will just be to finish things off. So let's get started. As per usual, if anyone has a question, just put it straight into the live chat and I will do my best to answer it live. So I hope you're all doing well. You're all rested. You've had a good chance to do some good revision outside of the AMALathon as well. And you're all feeling good and confident to getting amazing GCSE results. So, bacterial disease. Gonorrhea is a sexually transmitted disease. It's an STD caused by the bacteria Neisseria gonorrhea. Now you don't need to know the name of the bacteria. It's fine to hear it. It's okay to try and say it, but you don't need to memorize it. And it affects the reproductive system and can lead to serious complications if untreated. Now, the issue with a lot of bacterial infections is the treatment is antibiotics. So, due to survival of the fittest, which is a topic that we're going to cover soon, the bacteria that survive are the ones that are really strong, or the ones that have some amount of resistance. So over time, as people keep taking antibiotics for illnesses like gonorrhea, for example, a type of gonorrhea or type of bacterial cell can come about that is resistant. So this is one thing that actually happened with gonorrhea, where a natural resistance has occurred. So it's become more and more difficult to treat. So the symptoms of gonorrhea are painful urination and thick yellow or green discharge from the genitals. So it's clearly not an illness that anyone would want to get. How does it spread? Unprotected sexual contact. And it can also spread from mother to baby during childbirth. And how can it be prevented? Regular sexually transmitted disease testing for those who are sexually active, and to avoid multiple sexual partners. So, black spot disease, which is a fungal disease. So black spot is a fungal disease that affects rose plants, causing dark spots on leaves and reducing plant health. So the symptoms, it has black or dark brown spots on the leaves. The leaves start to turn yellow and fall off. There's weakened plant growth and fewer flowers. Now we're going to look at why that actually happens in just a second. But how can we stop the spread? How does it spread? Water splashes from the rain or irrigation. So it means it gets into the water and then it can spread through the water. It's a fungal disease, which means it can release spores. So the wind can take those spores and then it can infect other leaves as well. And warm and humid conditions favour the growth of the actual fungal disease, the actual black spot. So in certain conditions, it grows faster. Now, how to prevent and control it? Remove infected leaves and plant debris. So if there's an infected plant, you need to make sure you clean up around it. The easiest way is just to remove the plant. Avoid overhead watering to prevent moisture build-up. and use fungicides regularly. Now, what we need to understand from this is, why would it stop growing? Because we've got these black or dark brown spots on the leaves. We've got the leaves turning yellow. And if the leaves are turning yellow, that indicates that we've got less chlorophyll inside the leaves. So on our green leaf, it's the green part where photosynthesis occurs. Now, if any of that green part is not available, what does that mean? It means there's less photosynthesis occurring. So these black and dark brown spots means there's no photosynthesis occurring there. These leaves that turn yellow, that means there's a reduction in chlorophyll in those areas. That means there's less photosynthesis happening. If there's less photosynthesis, that means there's less glucose for respiration. There's less glucose to convert into amino acids, which then turn into proteins, and we need that for growth and repair. Hence, we have weakened plant growth and fewer flowers. So, malaria, a serious and sometimes fatal disease caused by Plasmodium parasites, and transmitted through the bite of an infected female Anopheles mosquito. So it does need a vector. So the symptoms of malaria are high fevers and chills, sweating and headaches, muscle pain and fatigues. How does it spread? It spreads through mosquito bites, but only through the ones that have the only ones that have bitten people who are infected. So for example, if you go to certain countries, they'll have mosquitoes there. The mosquitoes won't cause malaria. So if you see a mosquito, you can see mosquitoes in the UK as well. It doesn't mean that they'll pass on malaria. If you go to a country where malaria is common, then there's a bigger chance that the mosquitoes around there may have malaria within them. Now, mosquitoes aren't affected by the malaria itself. What's affected are the ones that they bite and try and take blood from. Now, this is really rare, but it can pass on through blood transfusions, and also it can pass on from the mother to the child. Now, how can we prevent that? We can use mosquito nets and repellents. We can drain stagnant water to reduce mosquito breeding, and we can wear long sleeve clothing in mosquito prone areas. So not wearing t-shirt and shorts, for example. Okay, so our defense system. The human defense system protects the body against pathogens, bacteria, virus, and fungi that cause diseases. It contains two main types. So we've got a non-specific defense system, which is our first line of defense. So this is the body's natural barrier to preventing pathogens from entering. So here's our natural barrier. We've got our skin. Now the skin acts in two different ways. I know some teachers, and some of you in school, the teachers may have described it as like a wall, or if you have a castle, it's the big strong wall that stops people from entering. But also the skin has bacteria on top of it. It has something called microflora, which is a good bacteria. It doesn't cause us any harm. So because our skin has this thin layer of bacteria over the top, essentially there's no room for any other bacteria. So if a bacteria that might cause skin infection or might cause us some sort of infection lands on our skin, there's no room for it to propagate, for it to increase in numbers. Then we have mucus. So mucus is made, one of the places where mucus is made is within our lungs. And in our trachea and within our lungs, we have these tiny little hairs called cilia. And what happens is when we breathe in any germs, so for example, we breathe in, and all the time, whatever air we're breathing in, it is usually full of bacteria, it's full of dust, dirt, viruses, lots of different things. But that's not really a problem. Because what happens is the tiny cilia catch a lot of this dust, a lot of the bacteria, and then when our body flicks up the mucus, all of these things that have been caught on the cilia go into the mucus. Now when it reaches the top of our throat, 99% of the time we actually swallow it. It then goes into our stomach, and our stomach acid then destroys the microorganisms. Then we've got cilia, which I've just explained. Stomach acid, so that means if anything enters our stomach, so for example the mucus, when we drink food and water, if it has any bacteria or anything else inside there, the stomach acid kills them. Because if we have a strong amount, but if you have an acid which is high in pH, it will denature the enzymes. It denatures the proteins. In other words, killing the bacteria, because the bacteria is a protein, or is made up predominantly of proteins. Then tears and saliva, they contain enzymes. So our tears contain enzymes like lysozymes that break down and destroy bacteria. Our saliva also contains enzymes, but our salivary enzymes are more for digestion. But here, whenever our eyes water slightly, what that actually represents what's in that water is that enzyme, lysozyme, which then, as an enzyme does, breaks things down into smaller pieces. So it breaks it down, breaks the bacteria down. Okay, now we've got our specific immune system defense, and this targets pathogens once they've entered. So we've got phagocytes, or which is where phagocytes do phagocytosis, and they are white blood cells that engulf and digest pathogens. So usually what happens is, when a white blood cell, a lymphocyte, so here's a lymphocyte, it releases antibodies. Now when it releases an antibody, that antibody is specific to a certain bacteria or virus. So if we have a bacteria, for example, and it has a bacteria will have antigens attached to it. Now I'm sure we will be going through this in a bit more detail afterwards. It has antigens, which are protein groups specific to each bacteria. Now when the white blood cell releases the antibodies, each white blood cell has antibodies. So these things I'm just drawing now are antibodies, and each white blood cell releases antibodies specific to a certain bacteria's antigen. When they attach like this, not only do they stop the function of that bacterial cell, they also cause the clumping together of these bacterial cells. So now, when our phagocyte comes along, now I know I'm aware I'm drawing this a bit like a Pac-Man, but obviously it doesn't look like a Pac-Man. What it essentially does is it grows around it. So if this is your bacterial cell, phagocytosis means it grows around the bacterial cell and then digests it, breaks it down. Now, memory cells. Once a white blood cell has the correct antibody, or has made the correct antibody to destroy a bacterial cell, then it multiplies rapidly. So it can kill off that bacterial infection or whatever the infection is. Now, after the infection's gone, after that bacteria's been destroyed, then the body doesn't need that many white blood cells anymore. So what it does is it removes the majority of them, but it leaves a few behind. We call those memory cells. That means if that bacterial cell enters the body again, we have a secondary response, which is really fast. It's really fast because we now have memory cells that know how to destroy that particular bacteria. When we encounter a bacteria for the first time, our body doesn't know how to break it down. And that's what I'm explaining here. So where lymphocytes then produce the correct antibody, and that can take a little bit of time. But the second time we get that infection, that this bacterial cell, is the second time we've seen it, third time or fourth time, at that point we have memory cells that already have the correct antibodies. And then antitoxins. So our lymphocytes, our white blood cells, also release antitoxins, which neutralize toxins produced by bacteria. So again, it's something that we've covered: bacteria release toxins. And those toxins attach to our cells and stop them from working properly. So, a few questions. How does the skin help protect the body from infections? So the answer, the skin acts as a physical barrier that prevents pathogens from entering the body. What is the function of mucus in the respiratory system? Answer, the mucus traps microbes. The mucus traps microbes and dust particles in the airways, preventing them from reaching the lungs. Name the enzyme found in tears and saliva that help destroy bacteria. Lysozyme. Explain how stomach acid helps prevent infections. Stomach acid kills bacteria and other pathogens that enter the body through contaminated food or water. Specifically, what do they do? They release, the stomach acid denatures the protein, and bacteria, virus, etc. are proteins. What role do cilia play in the human defense system? So cilia are tiny hair-like structures that move in wave-like motion to push mucus and trapped pathogens out of the lungs, preventing infection. Now, in one of the questions, sebum has been mentioned. Now, sebum is an oily substance that protects the skin. So you are correct. It does form a protective coating on the skin, and it does help protect the skin from harmful pathogens. But it's not something that you necessarily need to know for GCSE. It's not something that's on the spec. Teachers do sometimes mention it as additional information, particularly if you have a teacher that's got a university degree in biology. They're going to have an even better and deeper understanding of all the different defense systems. But from a AQA specification perspective, what we've got on the previous slide is what you need to know. Well, not this slide, sorry. This slide. No, not this slide either. We'll get there eventually. This slide. Okay, so what we've already mentioned here, phagocytosis, that uses the phagocytes, where white blood cells engulf and digest the proteins. Then we've got the production of antibodies. Antibodies stick to antigens and destroy pathogens, so that's been explained as well. And production of antitoxins. So here we've got phagocytosis. As you can see, the phagocyte grows around the pathogen. So now the pathogen is engulfed and it can then be ingested. Here we've got antigens and antigen binding sites. So it's what I was showing before. This is an easier way to show it. Antigens are protein groups, and the antibodies attach to these protein groups and stop the bacteria from functioning. And antitoxins are a protein that attaches to a toxin and then stops it from working. So in a sense, the same way an enzyme has a specific substrate, the antitoxins attach to a certain part of the toxin and that stops its function. So it is all about specificity. And this goes back to protein production. So when proteins are made, amino acids, the order in which the amino acids are, so this is what the order of the amino acids makes a specific protein, and that leads to its specificity. Now, describe how pathogens cause infections, and describe how the immune system defends the body against these pathogens. So this is just some advice on how you're answering questions. So we should be aiming for level 3. So all 6-mark questions, we should be aiming for level 3, which is you provide a good description. So in this case, for this particular question, it would be providing a good description of pathogens. It says describe how pathogens cause infections and how the immune system responds, and not to make too many errors or omissions. So it's where you have a comprehensive answer. So the reason I don't usually go through this when I go through 6 mark questions is because what I'm going through as part of the 6 mark question automatically is a comprehensive answer. So here's the answer. How can pathogens cause the infections? Bacteria and viruses are pathogens. They reproduce rapidly inside the body. Bacteria produce toxins. Forget the word poisons, although that's correct. And viruses live and reproduce inside the cells. So I've shown this before. The viruses use our ribosomes to make themselves. So within our cells, we have ribosomes, and what the virus does is it injects a single strand of DNA, which we call an mRNA, and then as it goes through the ribosome, our ribosome is then able to convert that into a chain of amino acids, and then that folds into a 3-dimensional protein. And in this case, that 3-dimensional protein is the virus. So that means the virus hijacks our machinery, hijacks our cells, our ribosomes, to make lots of them. Eventually, the cell becomes full of viruses and it lyses, it splits apart, and those viruses are then free to infect lots more cells. But how does the immune system defend the body? So what we've just covered, it ingests the pathogens, bacteria or viruses. It destroys pathogens, bacteria or viruses. So here we've got, this first one is phagocytosis. So the phagocytes grow around the pathogen and then ingest it. Then we've got our normal lymphocytes. Which destroy the bacteria or the pathogens. They produce antibodies and they produce antitoxins. Why do they produce antitoxins? To counteract the toxins. And this leads to immunity from the pathogen. So at this point you can mention memory cells as well. Okay, vaccination. So everything we've just covered will help us to understand vaccinations a lot better. So vaccination is a process in which weakened or inactive form of a pathogen is introduced into the body to stimulate the immune system to produce antibodies. This helps protect against future infections by the same pathogen. So what that means is a weakened or inactive form of a pathogen is introduced to the body. So we've got a weakened or a dead version. Dead version of the vaccine that's added into the body. Once it enters the body, initially because it's a brand new bacteria that the body's not seen before, or virus, what will happen is the white blood cells, the lymphocytes won't know how to destroy it. Eventually, a lymphocyte will be made that has the correct antibodies. Remember, even the dead version still has antigens. Now, the antibodies are released, and these dead or weakened versions of a pathogen are destroyed. Now, at that point, because this white blood cell has the correct antibodies, it starts to multiply. The body starts producing, not multiply, the body starts producing lots and lots of them. So remember, these are made in the white, made through adult stem cells in the bone marrow. Now, we've got all of these white blood cells, and now all the bacterial cells have been destroyed, or enough for the person to feel better. Now the body does not need this many white blood cells, so what it actually does is it removes the majority of them, but it leaves a few behind to act as memory cells. So now what we've got, the white blood cells that are left behind are memory cells. So now if the real version comes along, that memory cell multiplies rapidly, releases the correct antibodies, and very quickly destroys the pathogen, so therefore preventing infection. This is why, if you put vaccination to one side, this is why the first time we get ill from something, we get really ill. If it ever comes to us the second time round, we don't usually get ill because our body has a protection against it, or we get slightly ill. A good example is something like chickenpox. You get chickenpox once. But you don't get it a second time round, because by that point our body now has memory cells to protect us against it. So the benefits of vaccination: they protect against serious diseases. So vaccines help the body build immunity, preventing severe illnesses and complications. It prevents the spread of infections, because widespread vaccinations reduce disease transmission, protecting entire communities. That's herd immunity. This is something that's really important and something to keep in mind. If around 80% of a population has been vaccinated, then the other 20% stay protected. And the way to look at that is like this. If you've got, we'll just use 10 people. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. And this person here is not vaccinated, and that person there is not vaccinated. Then the fact that the majority are vaccinated, it just means there's no one to actually, there's very few people that can actually get ill and transfer it to others. So because very few people are in a position to actually get ill and pass on to others, generally speaking, the remaining population stays okay. So this is widespread vaccination reduces disease transmission. So this reduces the disease transmission, and that protects entire communities. That's herd immunity. That's every once in a while we have certain generations that don't vaccinate because of different reasons. Delaying vaccination is a different thing, but not vaccinating at all. So we have had a few years in the past where we haven't reached herd immunity, and so infections increase. So benefits of vaccination: it lowers epidemic and pandemic risks. So vaccine controls outbreaks by reducing infection rates and limiting disease spread. It can eliminate diseases like smallpox. So smallpox was pretty much eradicated because of a vaccine. This was one of the first vaccines, or if it was the first vaccine that was used, where they used cowpox. So cowpox looks very similar to smallpox, but it doesn't cause the same level of illness. So they started to give people the smallpox. They started to give people smallpox in the form of a vaccine. And once the person had smallpox and had a bit of a temperature and they were ill for 1 or 2 weeks, they never got smallpox. Although smallpox at a certain time was killing lots of people. So vaccines train the immune system to recognize and fight harmful pathogens. They prepare the body to respond quickly and effectively if exposed to the real infection in the future. So this is what I've already explained, but we'll just read through it one more time. The more times we recap, the better we're going to memorize it. So exposure to a harmless form of a pathogen. A vaccine contains a dead or a weakened version of the pathogen that cannot cause illness. So that stimulates the immune system. The immune system recognizes the pathogen as foreign and then produces antibodies to fight it. Now, while it's making the correct antibody or making the right white blood cells being made, At that point, the pathogen is multiplying and it's causing us to feel ill, and it's destroying our cells. And that's the first time we encounter the pathogen. But once the pathogen has been destroyed, some white blood cells become memory cells, which remain in the body for years. So, what is a vaccine? A substance with a weakened or inactive pathogen that stimulates antibody production. How do vaccines prevent disease? They lead to the creation or the making of memory cells to help the immune system fight future infections. Why are booster vaccines needed? To maintain immunity as it may weaken over time. Because over time, if those memory cells aren't made use of, the body may remove those memory cells. Which disease was eradicated by vaccination? So that was the smallpox. And what is herd immunity? When most people are vaccinated, reducing disease spread and protecting others. So antibiotics and painkillers. So antibiotics are medicines that kill or inhibit the growth of bacteria without harming body cells. They are effective against bacterial infections but do not work on viruses. So the main thing here, they inhibit the growth of bacteria without harming body cells. They only work on bacteria, not on viruses. So penicillin, amoxicillin, anything that's got icillin at the end is an example of an antibiotic. Painkillers. Painkillers relieve symptoms of illness. They relieve pain and fever, but they do not kill the pathogen. So they're dealing with symptoms, they're not dealing with the cause. So antibiotics deal with the cause, painkillers deal with the symptoms. They help the body feel better while the immune system fights the infection. Things like paracetamol, ibuprofen. Okay, drug discovery. So drug discovery is the process of identifying and developing new medicines to treat diseases. It involves multiple stages, including research, testing, and clinical trials to ensure the drug is safe and effective before public use. So this process, many sections of it you just need to memorize. You need to know them just in case a question comes up. Questions on this usually come up once every couple of years. So these are examples. Now usually with drug discovery, so I should point out my background is actually a pharmacist. I'm a school teacher, I've been a teacher for 15 years, but my degree was in pharmacy. But pharmacy wasn't for me, so I became a school teacher. So I know when you go through this process of drug discovery, a lot of the initial starting point is actually from natural remedies, sometimes from what we would call old wives' tales. So, for example, when we're unwell and someone says, "Oh, add a little bit of this to water, or do this, or do this." So I remember once I had a toothache and someone told me to get some clove and just to put the clove next to that area in my gums, and the pain went away. And what's inside clove is actual pharmacists, the ones who are working in industrial pharmacy, have identified the part within the clove and made medicine from it. So willow bark, people were using willow bark for many years for pain relief and anti-inflammatory. We've now, pharmacists or industrial pharmacists have, so we're talking about the pharmaceutical industry now, they've identified which part of the willow bark causes that pain relief, and then they made aspirin. Foxglove makes a drug called digitalis, which treats heart conditions. It causes it causes the blood vessels to relax. Then you've got poppy plants, that's where morphine comes from. The cinchona bark, which is where quinine comes from, which treats malaria. And Pacific yew, which makes the drug Taxol, which is used in cancer treatment. Okay, I'll come back to monoclonal antibodies because that's a triple award. So the slides have got slightly out of place. I did change this, but it's reverted back to the original. Now, there's different stages. So this is not monoclonal antibodies. So I must have opened up the wrong presentation because I corrected all of this before. There are different stages. There are different stages of developing drugs. There's discovery, development, preclinical, and then there's phase 1, 2, and 3 clinical trials, and then there's a peer review. Peer review where different scientists check each other's work. So the different stages of developing new drugs. First is discovery. The first stage is discovery where scientists identify potential substances that could be used in medicine. So that's usually natural remedies. So for example, penicillin was discovered in mold, while other antibiotics have been developed by studying soil bacteria. So once a promising substance is identified, it moves to the developmental stage. Now, they may modify its chemical structure to improve its action or make it easier to deliver in a form like a tablet, injection, or spray. So an example of this is quinine. Quinine is something that was used to treat malaria. Now, to make it more effective, they can add extra chemical groups to it so that it absorbs better into the blood, or it is able to pass by a cell membrane better. So they modify what they know to work to make it more effective. So preclinical. The next step is preclinical testing, where the potential medicine is tested in the lab before being used in humans. So scientists test the substance on cells. So this is something I did myself when I was studying to be a pharmacist. You can get things like oil and water, or octane and water, because they don't mix. They actually in one respect. So for example, oil and water, just to give you an example, because the oil floats on top of the water, in one respect that mimics, not fully, but in a little bit, it mimics the membrane. So if you put medicine in one part, you can see how easily it moves into the other part. So initially what they'll do is test like that. Then they'll do tests on cells or tissues grown in the lab, and they see how it works and whether it's toxic. So they're trying to understand how the medicine is going to react once it enters the body. Then they'll move on to small animals. The medicine is tested on animals to assess its safety and effectiveness in a whole organism. So now phase 1. In phase 1 clinical trials, they take a small number, so this is only phase 1. They take a small number of healthy individuals, then they give them the medicine that they've been testing on animals and cells, just a small amount to test for toxicity. Test for toxicity, side effects, and how the body processes, how it reacts to the medication. Because when medication goes to our body, it goes to the liver. And what the liver does is it breaks it down, and it can break it down to something that's toxic. It can break it down to something that's beneficial. So there are actually some medications that when we take them, what happens is once we've taken them, they don't actually work till they've gone through the liver, because the liver then breaks it down to something that's quite effective. There are other medications that the liver could break down to something that's poisonous. So that's the issue here. So if it breaks down to something that our liver is going to break down to something that's poisonous, then that's a no-go. And it won't go to the phase 2 or phase 3 of the clinical trials. Now, here, only low doses are tested at first for safety, and the healthy individuals are used as it's too great a risk to use an ill person. And they'll be paid well as well anyway. So, phase 2. Small number of patients. So now we're looking at people who actually are unwell. And this is to find the optimum dose, to see if the drug works and to check for side effects. So the trial is conducted as a double-blind study to avoid bias. Now, double-blind study means the doctor's giving the medication. So let me explain the next bit first. So one group is given a placebo, which does not contain the drug. So usually like a sugar pill. Everything else between the two groups, such as age and other relevant factors, are pretty much kept the same. So they can do a comparative. Now, when we say double-blind study, it means one group is given a placebo, one group is given the actual medicine, but the ones in the two groups of patients, they don't know who's been given the placebo and who's been given the medication. And the doctors giving the placebo and the medicine, they don't know which one they've given either. So that means it avoids bias. No one knows who's been given what. So they can take the data from it, and they can be accurate with that data. Because what can happen is, if a company really wants to, by the way, to make medicine, at this point, already by this point, companies will have spent close to, billions of pounds. And so sometimes you have these big pharmaceutical companies that spend billions of pounds, and then the medication doesn't go to market because of a toxicity issue or something's not quite right with it. So to avoid bias, you have a double-blind study, so no one manipulates the data to make it seem like everything's okay. And finally, phase 3, where a large number of patients are tested. So again, this helps to find the optimal dose. It's still to check if the drug works and to check for side effects. Again, the trial is conducted as a double-blind study. One group's given a placebo, and the majority of things like age, sex, like the health level, all these different things are generally kept the same to the best ability. So there's a comparative. So what are the ethical implications of using placebos? So placebos are inactive substances designed to resemble a real drug, and are used to controls in clinical trials to compare the effectiveness of a drug against no treatment. Now, while they help determine the true impact of the medication, they raise ethical concerns, as many participants may receive a placebo instead of actual treatment, and it could be the actual treatment works. And the reason why sometimes with some of these trials, the reason why some people actually go to these trials is because they have they've got the illness, and it's almost like a last hope. And so if someone if someone's got a stage 4 particular type of cancer, and they've been told there's no treatment, but there is this one clinical trial, it could be a miracle cure, and the person thinks, well, I've got nothing, this is a chance, I'll take it, and they get given the placebo, and all the people that took the real drug got better, and the ones that took the placebo didn't, then it means there's an ethical concern here, and people would obviously see that as unfair. Okay, to summarize, developing new medicines involves discovery, preclinical testing, the clinical trials to ensure safety and effectiveness. The rigorous process protects patients and public health by verifying that drugs are both safe and beneficial before widespread use. Okay, now the remainder of this topic is monoclonal antibodies, then it's the uses of monoclonal antibodies, and all of this section is triple award, is separate science, and then there's detecting plant diseases. So all of this section till the end is now separate science. So I'm going to go through that first before I continue with topic 4 biology. Okay, so what are monoclonal antibodies? They are highly specific proteins designed to target a single antigen, making them incredibly useful in medicine and research. Now, if anyone isn't doing separate science, you can take a 10-minute break and then join us again at 10 o'clock. Bastin, I know that question was asked on Monday, and I'm really sorry because it's not a question that's needed for GCSE specification. I can't give it a priority. As soon as I've got time, I will answer it. It is, and like I said, the problem is the question takes a few minutes to answer, and the aim is to finish the curriculum. So I don't want to move away too much from the curriculum to answer the question. But I think the question is excellent. I do really want to answer it, and I do have it in mind. So I am sorry. Now, monoclonal antibodies are highly specific proteins designed to target a single antigen, making them incredibly useful in medicine and research. So a hybridoma cell is a cell in which a lymphocyte is combined with a particular tumor cell. So we're going to go through this. How are they made? So we take lymphocytes and we create hybridoma cells, and then that produces antibodies as the hybridoma cells divide. So a mouse is injected with the antigen. Now remember, the antigen is the protein part. Of the outer line. Well, it's a protein group. It's a protein that's on the outside of the bacterial cell. Now, the mouse is injected with these antigens, or like a dead version of that cell. Now what that means is the mouse starts to produce antibodies against it. So we look back at this picture here. The mouse has been injected with antigens. The mouse has now started to make antibodies inside. This stimulates the mouse's immune system to produce lymphocytes, white blood cells, that generate the desired antibody. Now, what do we know about white blood cells? How are they made? They're made through stem cells. So they don't reproduce, they don't multiply by themselves. So this is our first step. The mouse has been injected. The mouse is now starting to produce the antibody that we need. Okay, apologies. I'm just going to have to go back and explain it. Right, it's okay, sorry about this. If you just bear with me for 1 second, get to the right page. So what they use instead is a tumor cell. Now, what it is about tumor cells that would be useful in this case is that they rapidly multiply. It's uncontrolled division. So you've got tumor cell which are rapidly dividing, and you've now got the mouse's white blood cells, which can make the antibody. So what we make now is a hybridoma cell. In other words, a hybrid. We combine the two. We take this plasma cell, this white blood cell, and the tumor cell, and we make a hybridoma cell. So now what we've got is a cell that's rapidly dividing, going through mitosis rapidly, but it's also producing the monoclonal antibody, the specific antibody that we were trying to produce. So, explain the role of hybridoma cells. So, hybridoma cells are produced by fusing a lymphocyte with the desired antibody with a tumor cell. They can divide rapidly and produce large quantities of a specific monoclonal antibody. And how a monoclonal antibody is produced? Monoclonal antibodies are produced by fusing a specific lymphocyte with a tumor cell to create hybridoma cells. So it's essentially the same question here. These hybridoma cells divide rapidly and generate large amounts of identical antibodies for medical use. Okay, uses of monoclonal antibodies. So monoclonal antibodies have a wide range of use in medicine, diagnostics and research. So here are some of the key applications. So they can be used in cancer therapy. Some monoclonal antibodies target specific cancer cells. Pregnancy tests. So monoclonal antibodies detect human, HCG in the urine. So what actually happens is the HCG attaches to the monoclonal antibodies, and that's why we see, so in a pregnancy test, it's about to come up, so I'll explain it then. But in the pregnancy test, they are monoclonal antibodies that are attached to a dye. Now, if the HCG attaches to those, they form a line, and then we can see that line within the pregnancy test, indicating a pregnancy. So, monoclonal antibodies in pregnancy tests detect a hormone called HCG, which is only present in pregnant women's urine. So the test strip has special monoclonal antibodies that only attach to the pregnancy hormone. Now, if HCG is there, the antibodies stick to it, forming a pair, a complex, and this helps show a positive result on the test. So you see this second line. The second line is produced when the HCG attaches to the antibodies, the monoclonal antibodies. So the complex then moves along the strip and attaches and reaches a region where there's another set of antibodies is attached, creating a visible line. So this is the part that the pregnant or potentially pregnant lady would urinate on. So they have to urinate. As the urine moves through, it attaches to the HCG within the urine, if they're pregnant that is, attaches to a first set of antibodies. And you know the bit I've just circled here, there's already another set of antibodies there. So when the first set of antibodies comes along with the HCG attached to it, they form a complex. And it's that initial antibody that has a dye attached to it. So then you see that as a straight line, and you see it as a colour. Now, the simple and accurate use of monoclonal antibodies has revolutionized pregnancy testing, making it widely accessible and easy to use. Okay. Okay, we can also use it to find blood clots. So monoclonal antibodies can be designed to attach to specific markers on blood clots. So these antibodies can also be linked to special dyes that glow under UV light. So these antibodies, blood clots give off a certain hormone. The antibodies attach to that hormone, but this particular antibody, now this monoclonal antibody, has a special dye attached to it. And we can see that under UV light. So when that UV light is shone onto a person, so when it's viewed with a special camera, doctors can easily see where the clot is. And that helps locate the clot faster, making it easier and quicker to treat the patient. So that's life-changing, that's life-saving stuff there. So steps to produce PVY monoclonal antibody. So how is the so this is an exam practice question. I mean, it's gone straight to the answer here. But what are the steps to produce a monoclonal antibody? Now, it says here PVY monoclonal antibody. That part there means nothing. The main part of this question, I know it's not written as a question form, is what are the steps to produce monoclonal antibodies? So they can add whatever, an examiner can add whatever word they want, but it's irrelevant. So if you think about it, it has no actual link. It has no link to the question other than to put people off. So what do? We inject the PVY protein. So the PVY just refers to the bacteria. The protein to the mouse, that triggers an immune system. And now the mouse produces the correct white blood cells or lymphocytes and antibodies to destroy that protein. Now we take those lymphocytes from the mouse and we combine it with tumor cells to form a hybridoma cell. We set the hybridoma cell that produces the correct antibody for PYC. We clone the hybridoma so it multiplies. These clones produce large amounts of monoclonal antibody. Then all it has to be done is purified to separate the two. So that's a really good question. This just shows the importance of doing past paper practice. So like I said, hopefully tomorrow will be a shorter session. So if it is a shorter session, you've got that little bit extra. Yeah, this is triple content, Nadia. We're just about to finish this, triple content, and then we're going to move on to topic 4. So uses of monoclonal antibodies. How can monoclonal antibodies be used in diagnostic cancer tests? So to help diagnose cancer, special proteins called monoclonal antibodies are made, and they attach to specific antigens, markers found on the cancer cells. Now, when special markers like glowing so the special markers, so with the monoclonal antibody, they have like a glowing fluorescent or radioactive tag attached to it, and that helps the doctor see where the cancer cells are. So when this monoclonal antibody attaches to the actual antigen, it has this part here which glows, so we know where they are. Okay, so advantages of using monoclonal antibodies. They are highly specific. They only bind to specific target cells, reducing the harm to healthy cells. They're effective treatment because they can be used to treat disease like cancer and autoimmune disorders. They're accurate diagnostics. They help in medical imaging to detect diseases early, and they are used to treat cancer. Now the issue is that they're very expensive. So if someone does go to the hospital, it's very unlikely monoclonal antibody testing kits are going to be used, unless potentially it's a private hospital or someone's very, very rich. There are possible side effects. They can cause allergic reactions, fever, and low blood pressure. They could lead to an immune response. That means the body might recognize them as foreign and reject them, and they're not always effective. Okay, how does a pregnancy test detect pregnancy? So one, the pregnancy test detects the hormone HCG in urine. The HCG binds to monoclonal antibodies on the test strip. The hormone antibody complex moves and reacts with immobilized antibodies, forming a colored line. Okay, so learn these 3 steps. Look at the past paper practice that's available. from all the past papers on this topic and memorize the answer. It's the same answer every single time. This question comes up quite often, or questions linked to monoclonal antibodies, particularly in the separate science paper, it comes up very often. So when you go through a few of those questions, you're going to see it's the same kind of answer over and over again. So what's the role of monoclonal antibodies in pregnancy tests? They bind to HCG. These antibodies are attached to coloured beads or dyes for detection. If HCG is present, the test strip changes colour, confirming pregnancy. Okay, detecting plant disease. So this is just a few more slides and then we're going to move on to topic 4. So how can plant disease be detected? So number one, when we're detecting plant disease, we've got to rule out environmental causes. So the whole point of this topic is how can we actually detect a plant actually has disease. So number one, we've got to look for stunted growth. Look for powdery mildew, black mold, slimy patches. Now a lot of these things affect the whole plant evenly, but they particularly go to the lower leaves first if there's a nutrient issue. Because if there's a nutrient issue, the leaves at the top will still be photosynthesizing because they're getting quite a lot of light. It's usually the ones towards the bottom. You will look for malformed stems or leaves and discoloration. Now, with discoloration, it's what I explained before. When you've got discoloration like this, why is that a problem? Because it means there's a lack of chloroplast or chlorophyll, which means there's less photosynthesis. Photosynthesis makes glucose, which is needed for respiration. It's needed to make starch. It's needed to make proteins, or initially amino acids and then proteins. So it stops the plant from growing as well. It reduces the energy of the plant. And if you think about the phloem has companion cells which are full of mitochondria. And because of the design of the phloem, when you get to a sieve plate and the companion cell, it's through active transport that the actual movement of the water and the sugar and the glucose is manipulated. So the fact that the sugar water in the phloem goes up and down, left, right, goes everywhere, is because of active transport changing the concentrations on either side of the sieve plate. So next thing, analyze the distribution of affected plants in the field. So if the disease is spreading through a field, it often follows a pattern. For example, diseases caused by soil-borne pathogens might appear in patches, while diseases spread by airborne would be different. So observing how the disease, so if it's airborne, it could potentially be in wider, depending on which way the wind's been blowing. So observing how the disease is spreading can provide clues about its cause. The third thing to look at is diagnostic testing. So first thing, culture the pathogen. So that means you take the pathogen and you put it into a petri dish, that has starch agar, the agar has all the nutrients needed for the bacterial growth. The bacteria grows. Then looking at that bacteria under a microscope, you can identify if it's a fungal spore or a bacterial colony. Or you can detect specific pathogens at a genetic level. Now, fungal spores can be cultured on agar plates to observe their growth patterns. And then you can also use testing kits with monoclonal antibodies to identify the pathogen as well. So this is just using diagnostic testing. So these are more questions in the wrong place. How do plants physically defend themselves from infection? The leaf cuticle is a waxy waterproof layer. So that waxy waterproof cuticle, as in that's the top part of the leaf. So you ever touch a leaf, the top part feels a bit like rubber sometimes. That's the waxy cuticle. It's a waterproof layer that covers the surface of the leaves and stems. It acts as a physical barrier and it protects plants from water loss, pathogens and insect damage. And how do plants physically defend themselves from infection? They've got a cellulose cell wall, which is strong, rigid outer layer found in plant cells, made primarily of cellulose. Now, that means the complex carbohydrate is made of glucose molecules. Now, the glucose molecules have a regular arrangement like this. A really solid structure that gives the cellulose a really solid structure, which allows the cell wall to provide support, protection, and structure. So let's have a look at this question. What are the physical barriers that plants use to protect themselves from pathogens? Answer: plants have got cell walls, they've got a waxy cuticle, trees have got bark, and that prevents pathogen entry. How do plants use chemical defenses against pests and pathogens? So plants also produce antibacterial chemicals and toxins. So there are plants that specifically release antibacterial chemicals and toxins that kill pathogens, but it also deters herbivores from eating them as well. It won't kill the herbivore, but it will at least put them off. And give 2 examples of mechanical defenses. So mechanical means, for example, plants that have thorns, like rose bushes have thorns to prevent animals from eating the plant. Mimicry helps plants avoid predators as well. So there are some plants that have a predatory look to them, and that helps them from being eaten. And how do some plants deter herbivores using toxins? Some plants produce poisons like nicotine in tobacco or cyanide, making them unappealing to eat. So, not nicotine in tobacco or cyanide, but some plants produce poisons similar to nicotine, similar to tobacco. Well, that's right, nicotine in tobacco. So it produces, I didn't read that carefully enough. It produces poisons like nicotine in tobacco or cyanide, making them unappealing to eat. Now, this again, I apologize, these slides have gone completely out of sync. I did correct them 2 days ago, but it's possible I've opened an older version. So this is what we've kind of covered in the questions and answers. So some plants have got thorns and hairs that stops animals from eating the plant. Some of them, the leaves curl to droop when they're touched. That startles animals. So you do have sensitive plants like that. And then mimicry. Some plants look like a dead plant by drooping its leaves to avoid being eaten. There are some plants that look a little bit like a predator as well, as I mentioned. Okay, thank you everyone. I know that session was 1 hour, 10 minutes to get through all of topic 3. We're going to take a short break and we're going to restart at 10:20. So I'll see you all at 10:20. Okay, thank you everyone. Okay, welcome back everyone. And for those that have just joined us, welcome to day 5 of the AQA AMALathon. So let's get started. We are now on, so we have finished cell biology, we finished organization, we just finished infection and response, and we're about to start bioenergetics. So hopefully we'll be able to finish that today. If not, we'll start at 10 o'clock tomorrow and finish it then. We have finished atomic structure, periodic table, bonding, quantitative chemistry, and today we're going to be working on, we're going to be finishing off chemical changes. And same with physics, we finished everything, just got atomic structure to finish. And we've got C5 to finish. So hopefully we'll get that finished by tomorrow. We are on schedule, even after a slightly delayed start on the first day. So thank you everyone that's been coming on live every day. Thank you for those that are watching afterwards. We really appreciate how much support we've had and all the positive feedback that we've had. But we're happy either way. If anyone wants to give negative feedback as well, or give constructive feedback would be the best, then we're happy to take it on board. So we're looking at photosynthesis. Now photosynthesis is the reaction. Carbon dioxide plus water becomes glucose and oxygen. Now there's something missing here, and that is light. And where does this process occur? Within the chlorophyll, inside the chloroplasts. So you need to know this formula. You also need to know that it's 6 carbon dioxides. So for foundation students, you need to know the word equation, and the actual formula equation needs to be known by triple and higher. So we've got 6 carbon dioxides, 6 H2O. It used the energy from light. So which way is the light energy going? It's going inwards. So what do we call, if you think, well, we've not covered this in chemistry, it comes in topic 5, but when the energy goes inwards, we call that endothermic. So photosynthesis is an endothermic reaction. So C6 H12O6. And how many oxygens do we end up with? 6. So important that you memorize that. So what happens during photosynthesis? Photosynthesis is an endothermic reaction. This is quite a well understood topic, and students do really well in this. It's understanding the difference between describe and explain. Once you've understood that, which I think is something that we did cover when we covered enzymes. If you can understand it in this topic as well, it really opens up the number of marks you get. So we know it's an endothermic reaction. We know it's endo because the energy enters. In other words, it's absorbing the light's energy. The energy is transferred to the chloroplast, so the chloroplast absorb that energy, and it specifics the chlorophyll, the green pigment inside the plants that absorb that energy. Now the chloroplasts, they're the cell structures where photosynthesis takes place, and the chlorophyll is the green pigment essential for capturing light energy to drive photosynthesis. So the main thing we need to take from this page is it's an endothermic reaction. Photosynthesis occurs inside the chloroplast, specifically in the green chlorophyll, the green substance, green pigment known as chlorophyll, which actually absorbs the light energy. And how is the plant adapted for photosynthesis? So you look at this image, we've got quite a few different things here. We've got the stomata, and around it we have the guard cells. And that's where the carbon dioxide enters through. So that's really important. Then we've got the spongy mesophyll layer. That provides, that's a space for the air, the carbon dioxide to actually diffuse into. That helps provide a concentration gradient. So because we've got this empty space here, we end up with more carbon dioxide on the outside and less on the inside. Then we've got the palisade layer. This is the layer that has lots and lots of chloroplasts. So if you look at these cells, you'll notice compared to the cells below, they have a lot more chloroplasts. That's because they're on the surface of the leaf. That's where the majority of the photosynthesis is going to occur. And then we've got the upper epidermis and cuticle. Now cuticle we've already mentioned. Why is the cuticle important? It's a waterproof layer. So that means it stops water from evaporating out. Not only does it stop water evaporating out, it stops any microorganisms entering. So we're just going to quickly recap the same words. Palisade cells, these are packed closely together and contain many chloroplasts to absorb a maximum amount of light for photosynthesis. Sorry, my handwriting is not very nice, but that says synthesis. The xylem and phloem we'll come back to in just a second. Let's look at stomata. So stoma is one, stomata plural. These are pores that allow the exchange of gases. So this is where the carbon dioxide enters and the oxygen leaves through. Then we've got the guard cells. So we've got our stoma, which is the hole, and we've got the guard cells around it. These cells control the opening and closing of the stomata. So they regulate gas exchange and water loss. So on days when it's really sunny, the guard cells will fill with water, and when they fill with water, the stomata will appear. So the stomata can be seen at that point. So that when the guard cells contract, when they relax, they close the stomata, the gap. When they contract, they become like a banana type shape and they open. So when it's dull, when there's not much, when it's not very sunny, they close. When it's quiet, when it's sunny, when it's bright, they open. Because they'll be doing more photosynthesis. Now xylem and phloem we have covered already, but we'll go through it. The more we go through, the better. So the tissue transports water from the roots to the leaves, which is essential for photosynthesis. And what's the process? Transpiration. And the phloem, it transports sugars made during photosynthesis to different parts of the plant for energy and storage. And that's called translocation. Okay, now this is the part where some students do really, really well and some miss out on marks. Now what you can see here is the temperature is increasing on the x-axis, and on the y-axis we have the rate of photosynthesis. Now if this question said describe how temperature affects the rate of photosynthesis, and you will give this graph, what would you say? As the temperature increases, the rate of photosynthesis increases, and it peaks at 35 to 40 degrees. So the word increases is missing here. Now we'll come to the next part in a second. Photosynthesis rate decreases beyond the optimum temperature. So now, as the temperature continues to increase, the rate of photosynthesis decreases. So that's our description. Now, overall, this answer has been written as an explanation. But what I want you to see is if this comes up as a question in an exam, it might say describe. So if it says describe, we just say, as the temperature increases, the rate of photosynthesis increases until it reaches an optimum. So it's almost the same kind of answer as when we did enzymes. As the temperature continues to increase, the rate of photosynthesis decreases. So that's our description done. Now if we're explaining, we have to describe and explain. So here, as the temperature increases, the rate of photosynthesis increases. it peaks at 35 to 40 degrees. But why? So now we're saying, because we have an explanation, we need to know why. Because as you increase the temperature, the reactants particles have more energy, so they move more rapidly. Now, for those of you that remember their topic 6, they remember the paper 2 chemistry, what two things do we need for a reaction to occur? Number 1, a collision, and number 2, it has to have sufficient energy. So when you increase the temperature, you're giving the different particles involved in photosynthesis more energy, namely carbon dioxide and water. You're giving them more energy, and also because they move around faster, they're more likely to collide, which means you have a faster rate of reaction. And then we've got another description. When the temperature continues increasing, the rate of photosynthesis decreases. So the photosynthesis rate decreases beyond the optimum temperature because the enzyme has denatured. Now, if the enzymes that help photosynthesis occur have denatured, that means, and think back to enzymes, and we have to mention this every time enzymes comes up. If the enzyme is denatured, it means the active site has changed shape. If the active site has changed shape, the substrates won't fit into the active site. If they don't fit in the active site, then the rate of reaction, the rate of photosynthesis in this case, decreases. Now at approximately 45 degrees, the photosynthesis has ceased entirely. Why? Because all of the enzymes have become denatured. So when we hit a 0 at the end there, why do we hit a 0 at the end? Because photosynthesis has completely stopped. Okay, so now we're looking at how light intensity has an impact on the rate of photosynthesis. So this is a little bit different now. So as the light level is raised, the rate of photosynthesis increases, also up to a point. So as the light increases, so if we're describing this, we're just saying as the light increases, the rate of photosynthesis increases up to a certain point. After that point, it plateaus, it stays the same. Now, if we're saying why that occurs, as the light intensity increases, there's now more energy for the reaction to occur, because photosynthesis does not occur without light. But why has it plateaued? So when we're saying why has it plateaued, it's because we have a limiting factor. It means light intensity, we have initially, light intensity is a limiting factor. Let me just rub this out. So up until this point around there, just before it plateaus, the limiting factor is light. It means there wasn't enough light for the reaction to occur. But then we get to a point where we reach the maximum in our rate of photosynthesis. At that point, even if the light intensity increases, we can't increase the rate of photosynthesis because something else is limiting. So in this case, the limiting factor could be temperature or carbon dioxide levels. And so now to increase the rate of photosynthesis, we have to either increase the amount of carbon dioxide or we've got to increase the temperature. So this one is slightly different to the previous one. Light intensity is initially the limiting factor until a certain point. When we reach that certain point, it stops being a limiting factor. We know that because the line plateaus. We've got plenty of light. Now, light intensity is still increasing, but it's not having an impact on rate of photosynthesis. That means something else is now the limiting factor. And in this case, it is the temperature or carbon dioxide levels. So if we increase those, then what we'll see, and this is another question, the question might say, how will the graph look different if the temperature is increased? How will this graph look different if the carbon dioxide levels are increased? And to get the mark, all you've got to do is draw the line. And all you're doing is drawing the line at a higher point, like that. Okay, it's exactly the same answer. So light intensity and carbon dioxide, the answer is exactly the same, except we're using the word carbon dioxide instead of light. So as the levels of carbon dioxide increase, the rate of photosynthesis increases also up to a point, because the reaction can't happen without carbon dioxide. So if we don't have much carbon dioxide, like from here to here, then there's not enough carbon dioxide, so there's not enough reactants for the reaction to occur with. But when we get to this point here, we now have enough of the carbon dioxide reactant. We have enough of the carbon dioxide, so now we're limited in a different way. There's a different limiting factor. And in this case, the limiting factor is temperature or light intensity. So if we want to increase our rate of photosynthesis, we've got to either increase our light intensity or the temperature. So let's have a look at this question. First thing is complete the equation for photosynthesis. So carbon dioxide plus water makes glucose plus oxygen. And we would put the word light energy here. What type of energy is used in photosynthesis? Light energy. Which part of the plant cell structure absorbs light energy? The chloroplast. Okay, so we've already looked at this kind of question, so we can ignore that. How is energy for the photosynthesis reaction gained by plants? So the light energy is absorbed by chloroplasts. So again, same questions as previous. Okay, now, although at 25 degrees, the rate of photosynthesis in a plant depends on several factors in the environment. These factors include light intensity and the availability of water. Describe and explain the effects of two other factors that affect the rate of photosynthesis. So let's break down this question a little bit more. It says the rate of photosynthesis in a plant depends on several factors. Now, what are those several factors? We know that it's light intensity, it's carbon dioxide levels, and we know it's temperature. Now, these factors include light intensity and the availability of water. Describe and explain the effects of two other factors. So we know we don't need to worry about light intensity, but we're describing and we're explaining the effects of two other factors that affect the rate of photosynthesis. So what we've just gone through will get you full marks for a question like this. And this could be most likely is a 4-mark question, or it could be a 5 or 6 mark question as well. More likely a 4-mark or a 6-mark. So the first thing is carbon dioxide. As carbon dioxide concentration increases, the rate of photosynthesis increases because we're increasing the reactants. However, the rate levels off when another factor like light or temperature become limiting. Done. Effects of temperature. As temperature increases, the rate of photosynthesis also increases because more collisions between enzymes and substrates, and it speeds up the reaction. Now, we could talk about if you continue to increase the temperature, then the enzymes eventually denature, and that reduces the overall rate of photosynthesis, but that's sufficient. So most likely that was a 4-mark question. Okay, so how can you use equipment to investigate the effect of light on the rate of photosynthesis? So I'm sure everyone's done this required practical before, and everyone's familiar with it, but we'll quickly go through it. Now, like I've said before, whenever you have a required practical, my advice is to watch the YouTube clip. So I'll go through it briefly, but it's the YouTube clip that will really, really help. So how can you use equipment to investigate the effect of light on the rate of photosynthesis? So you set up the apparatus, place the pondweed in a beaker filled with water. Then you measure the rate of photosynthesis, turn on the lamp and start the stopwatch. Count the number of oxygen bubbles released per minute. So the first thing you'll be doing here is we set up the apparatus. We add our pondweed. It's important to keep the pondweed the same species. It's important that the starting point of the water has the same temperature, and you put the same amount of water in there, have the same source of water. Now we're going to measure the rate of photosynthesis. So we set the apparatus, now it's time to measure. So you add your ruler and your light bulb, and you have a certain distance. So for example, that could be 50 centimeters. Now you time it for, here it says per minute, but you can time it, for example, for 5 minutes, and over 5 minutes you count how many bubbles are given off. Because what's going to happen is the pondweed's going to absorb the light energy, and that's going to increase the rate of photosynthesis. So it's going to start producing more oxygen, and those oxygen bubbles will be released. So your job, what you'll be doing there is counting the oxygen bubbles. Then you repeat the experiment at different distances. So you do it at 40 centimeters, 30 centimeters, 20 centimeters, 10 centimeters. But whenever you're doing this experiment, you will need to change the water, because of the temperature of the water needs to stay consistent and constant. Now, when the bulb's really, really close to the water starts to heat up, to absorb some of the heat energy. Now, the light bulbs we use now aren't the old-fashioned ones. So the old-fashioned bulbs can cause burns and they get really hot because they're only 10% efficient. But if you think about the new LED ones, they're about 90-95% efficient, or even better in some cases. They don't cause much heating, but still, even a 1 degree difference can cause a difference in your results. So, control variables. How can we control the temperature? You could use a water bath if needed and keep it at 20 degrees so the water stays at one temperature. With carbon dioxide concentration, you can add sodium hydrogen carbonate to the water, and that releases carbon dioxide. Then you've got light intensity. Use a lamp at fixed distances from the plant. So we can control, well, light intensity here is not a control variable. That's our independent variable. So that should not be in this list. Type of pondweed, you need to make sure you use the same species. Time for measurement, just use a stopwatch. And volume of water, use the same amount in each test. Okay, so we've got 3 different types of pondweeds here. Alodia, which is probably the most likely the one that you used in school. Kabomba and Ageria. Now, in my school, we generally use Kabomba because it just provides better results. Now, give 3 variables the student should have controlled to make sure his conclusion was valid. So we don't need to look at his conclusion, we're just saying what can you have controlled. So you could have controlled the mass and length of the pondweed, the volume of water, the light intensity, that would be independent variable, the color of the light, the carbon dioxide concentration, the pH of the water, and the temperature. So all these things would ensure that his results are accurate, so his conclusion is therefore accurate as well. So the thermometer was placed in a glass beaker. Why was it important to use a thermometer in this investigation? Now this is an actual past paper practice question, and these are the kind of questions that really stump students sometimes, because when you see the answer, you're going to see it sounds really obvious. And what students like yourselves who are aiming for grade 9s end up doing is overthinking it. So it says the thermometer was placed in a glass beaker. Why was it important to use a thermometer? So first things first, what do we use a thermometer to do? To measure temperature. Why do we need to check the temperature? So remember, when we are answering why questions, explain questions, first it's because it's talking about a thermometer, you're describing what it does. It measures the temperature. Why? To check the temperature. Why is that important? To check the temperature isn't changing, because temperature is a control variable. And again, this is why past paper practice is really important. Now that we've activated, or we're close to finishing the activation phase, it's just lots and lots of past paper practice. And like I said to you guys before, when with any of our posts, if you reply to them, and in fact I'll make a post next week at some point that just say, do you have any questions from the AMALathon? And then you can, anytime you have a question, you can just put it straight there. And what I'll do and what our team will do is whenever we can, when we're able to answer the question, we'll answer it on as a text answer. Or what we've started now is we've started to make shorts. So if it means we make a quick short to answer it, we can do that too. We will hopefully be on TikTok and Instagram in a few weeks as well. These videos, they'll be about, they're going to be compiled, so all the biology comes together, all the chemistry comes together, all the physics is brought together. So you'll have 6-hour videos that cover everything. So there'll be a lot of shorts made from these videos as well. Okay, so what do we actually use glucose for? So one is respiration. So photosynthesis, the main thing that we need to get from photosynthesis is glucose, and for us, oxygen. So we need glucose for respiration. We need it so it can be stored as starch. Now, glucose is soluble. For example, when you make a cup of tea and you add sugar to it, which is similar to glucose, because both glucose and what we put in our teas are both. What we put in our tea is most likely sucrose, which is made up of glucose molecules, but it's still a small molecule. Now, what happens as soon as you put it in the tea? It dissolves. So the issue with glucose, you can't store it. It'll just dissolve into any liquid and move around. So when it's converted to starch, starch is insoluble. For example, potatoes. A potato is starch. So that's how plants, that particular plant, potatoes and root plants, store their starch. Others store their glucose in fruits and in other ways. But when it's again stored in fruits, because of the outer lining, it helps retain that glucose within that one space. But if it wants to be properly stored, it's converted to starch because it's insoluble. It's converted to cellulose, which is needed for cell walls. It's used to make amino acids. So if you take a glucose molecule, see these hexagons at the bottom? If you add a nitrogen to it, it has now become an amino acid. So it combines with nitrates to form proteins for growth. And it can also be stored as oils and fats. That's for energy storage in seeds. So it is a really important, it is a very, very important molecule. So now we've done photosynthesis, we're moving on to respiration. So respiration, now photosynthesis is an endothermic reaction because it takes in energy. But respiration is when glucose reacts with oxygen. Now, if I got a piece of paper and I reacted with oxygen, what would it produce? We actually call that combustion. So it would produce a fire. Now, respiration doesn't produce a fire inside our body, but it does release energy, just like a fire releases energy. So when you react something with oxygen, we call it combustion. It's an exothermic reaction. So respiration is exothermic, except we don't have little fires all over our body. What we have is the release of energy. So there's not these little fires across our bodies. We instead release energy. And we use this energy for our bodily needs. So we use it for movement, growth, and staying warm. And there's two types of respiration. Aerobic, which uses oxygen and happens in the mitochondria. That provides lots of energy. And we've got anaerobic respiration, which happens in the cytoplasm. And that's anaerobic. An means without. Aerobic refers to the air. So with the air, this happens. So without the air, so without aerobic, without the air respiration. So it happens without oxygen, but it still releases energy, but a lot less energy. I think it's around 1/6 of the amount of energy aerobic respiration releases. But at certain points, it is needed. The anaerobic respiration is required, particularly if we're running, until our fat resources start breaking down energy. And our fat then provides energy twice as much as our carbohydrates do. So what do we need energy for? Like we just said, we need it for movement, keeping warm, and growth and repair. Because cells need energy to grow, divide, and fix damaged tissues. Now, this is the formula for aerobic respiration. It's glucose, which is C6H12O6. Oxygen becomes carbon dioxide and water. Now, if you look at this really carefully, C6H12O6 plus 6 oxygens becomes 6 carbon dioxide and 6 H2O. Now, where have we seen this formula before? We've seen it in photosynthesis, but it's the other way around. So photosynthesis and aerobic respiration are reverse processes. Now, in photosynthesis, light energy is taken in, and during aerobic respiration, that energy is released, but not in the form of light. It's released in a form of energy that our body can then use for movement to keep ourselves warm, for growth and repair. Now, anaerobic respiration occurs in muscles. When we exercise intensely, our muscles need more energy than the oxygen than aerobic respiration can supply. Sometimes our oxygen levels decrease as well, because if you think about all the cells in our body needing oxygen, particularly the muscles that are contracting, they need lots and lots of oxygen at that point. And we can get to a point where the supply of oxygen decreases. So we end up with an oxygen deficit, an oxygen debt. So that's where anaerobic respiration has to kick in. So at that point, our body switches to anaerobic. Now the thing with anaerobic is glucose converts into lactic acid. And now when you're writing formulas, you don't have to write down energy. The reason being, it's not a product. It's what's released, energy is released, but it's not a physical product. It's not made of particles. So anything that's not made of particles, we don't write down as part of an equation. So you don't have to write and some energy. Now, lactic acid is an issue because what lactic acid does, it's an acid. So it causes denaturing of proteins. And where is aerobic respiration? Where is most of the respiration occurring? In our muscles. So when anaerobic respiration starts to happen there, what do we think the acid's going to do to the muscles? It's going to cause some of them to denature. Now we're only talking on a cellular level, so it's not going to destroy the whole organ, but the longer we anaerobically respire for, the more lactic acid is released and the more denaturing occurs. And that's why some people, when they're doing lots and lots of exercise, start to get leg cramps. That's because the lactic acid having denatured some of the proteins. So when yeast and plant cells respire without oxygen, so we have yeast is a living thing, it's a living organism. It respires. Now when it anaerobically respires, because it aerobically respires just like we do, it releases carbon dioxide and water. But when it anaerobically respires, it produces ethanol, so alcohol, and carbon dioxide. And we take advantage of that to make. So if you look at it, it has glucose, and that makes ethanol and carbon dioxide and releases energy. We call that process fermentation, and we use it for bread making and alcohol production. Now, when you have your dough of bread and the yeast is working within there, what happens is the yeast releases carbon dioxide, and that pushes, or in a sense it increases the pressure on the inside, and it pushes at the bread, and hence the dough rises and becomes bigger. The reason it looks bigger is because it's full of air pockets, carbon dioxide pockets inside. Once the bread bakes, the alcohol burns off because it dissipates, it diffuses away because it becomes a gas. It's a gas at 66 degrees. So when you put it into the oven, it will just dissipate. And also, when you cut your bread, the carbon dioxide gas that's trapped in the because when you cut a loaf of bread, you have all these tiny gaps. Those pockets of gaps would have been full of carbon dioxide. That's what's been pushing the bread upwards. So let's do a comparison between aerobic and anaerobic respiration, because this is something that commonly comes up in exam questions. So the type of respiration, aerobic needs oxygen, but anaerobic does not. Products, aerobic makes carbon dioxide and water, but anaerobic, now we're talking specifically in yeast here, yeast cells. So in animals we make lactic acid. In yeast and plants, anaerobic respiration leads to ethanol and carbon dioxide. Now we don't worry about plants, we need to know about yeast though. And relative amount of energy produced. So aerobic produces more energy because the glucose is fully broken down. Glucose is made up of 6 carbons. So all 6 of those bonds are broken, whereas in anaerobic, it's only the glucose is only partially broken down. So that means less energy is released. Now, when we do topic 5, we'll do something called we'll be looking at something called a phrase known as bendomex. Now, I'm not sure if anyone's ever heard the phrase bendomex. I'm using the wrong pen here. Bendomex means when you break bonds, it's endothermic. When you make bonds, it's exothermic. Now, depending on which one occurs more, as in, let's say, with every single reaction, so let's do something really simple. Let's not do something simple. Let's do one that we actually have in front of us here. In C6H12O6 and 6O2, all the bonds have to break on this side. So the bonds are breaking. So on this side, it's an endothermic reaction. On the product side, the bonds have to reform as carbon dioxide and H2O. So that is, the bonds are being made. So that is exothermic. Now overall, both things happen, but more energy is being released when compared to the amount of energy that is being taken in. So overall, this reaction is exothermic. Whereas photosynthesis is the other way around. More energy is being taken in to break the bonds than is being released when the bonds are made. Okay, now you don't need to bring bendomex into any respiration or photosynthesis questions, but I just wanted you guys, consider you're a group of students that are aiming for high grades. It's important for you to be able to see the link between topics. It'll help a lot more when you get to A level, but still it gives you a bit of a deeper understanding, which is good. So what happens to the body during exercise? We have an increased heart rate, so the heart beats faster to pump more oxygenated blood to the muscles, so that makes sense. If we're exercising, we need more oxygen, we need more glucose. So if the heart rate is faster, it's going to pump more blood around the body, and we're going to get more oxygen through the blood, and we're going to get more glucose through the plasma. We have a faster breathing rate. Again, why do we need a faster breathing rate? Because we need more oxygen to enter our red blood cells, and we also, because we're doing more respiration when we're running, when we're doing exercise, we need to remove the carbon dioxide. Because carbon dioxide, after a certain level, is toxic for us. And more oxygen to the muscles. So if we increase the heart rate, we have a faster breathing rate, it ensures that we get more oxygen to the muscles, which means more aerobic respiration occurs. And our body doesn't need to go to the anaerobic process. So the fitter we are, the longer we can last. The longer it takes for the anaerobic respiration to kick in. So what happens if there's insufficient supply of oxygen during exercise? So if there's not enough oxygen, that's when anaerobic respiration starts. So the body breaks down glucose without oxygen. It produces lactic acid instead of carbon dioxide and water. But the issue with that is we have a build-up of lactic acid. and that causes muscle fatigue. So like I said to you guys, the muscles start to cramp up, they start denaturing, and that makes movement much harder. And that means we have an oxygen debt. So after exercise, the body needs extra oxygen. So if anyone's ever done exercise, then afterwards they've taken deep breaths, and they're struggling, they're taking lots and lots of deep breaths. Why are they taking those deep breaths? To catch up on the oxygen that they missed out on. And what that oxygen does is it breaks down the lactic acid into carbon dioxide and water. And then that also helps restore normal oxygen levels in the blood. Right. What I'm going to do with biology is I'm going to continue till 11:05, and then we'll have a 5-minute break. I'm hoping we can finish that in the next 5 minutes. If we can't, it's not a problem. We'll finish it off tomorrow. So yeah, we'll carry on till 5 past, and we'll take a break till 10 past at that point. So why do we have oxygen debt? Because it breaks down lactic acid into carbon dioxide and water. So we still release the same amount of carbon dioxide in the end. It restores oxygen levels, and it helps muscles recover after anaerobic respiration, because that lactic acid will just continue to cause denaturing. The blood flows through the muscles, and it transports the lactic acid to the liver. So in the liver, the lactic acid will be broken down. Okay, metabolism. Actually, we will finish in the next few minutes. Metabolism is all the chemical reactions that happen in a cell or the body. So, for example, when our body turns amino acids to proteins, that's metabolism. That's a chemical reaction. When our body makes lipids, that's metabolism. When our body makes carbohydrates, that's metabolism, because it's a chemical reaction. When our body does photosynthesis, when a plant does photosynthesis, or when a living organism does respiration, that is metabolism, because it's a chemical reaction. So when we use glucose to make amino acids, when plants use glucose to make amino acids, to make starch, to make glycogen, when we turn glucose to glycogen in our liver and our muscles, when it's used to make cellulose, when amino acids make proteins, all of this is metabolism. So metabolism, like I've said, and this is why I'm saying this is not going to take too long, because these are just examples of metabolism. So sugars are the main source of energy for cells. The body stores this in the muscles and the liver cells as a source of energy. So glucose gets converted to glycogen, and glycogen is stored until we need the extra sugar, extra glucose. Amino acids are used to make proteins. So these proteins can be used for growth and repair. So I know this is just repeating over and over again, but what is metabolism? It is the chemical reactions that occur in our body. So any chemical reaction is metabolism. Now, what factors can affect metabolic rate? If you just look at this information here, what's happening as the age increases? The metabolic rate is decreasing in males, and it's decreasing in females as well. So what factors actually affect metabolic rate? Genetics. So how we break down different types of foods. If we break it down fast or slow, that's genetics. Some people have fast metabolisms because they release more of certain hormones. Our body mass index, so higher BMI can lead to a slower metabolic rate. So rates of respiration and things like that decrease. Exercise. So when a person has more muscle, that means there's more respiration happening inside those muscles, so that increases metabolic rate. Okay, excellent. So that is the end of our AMALathon biology. So well done, everyone. Thank you. That's the end of the first section of the AMALathon. So tomorrow there won't be any biology. We'll start at 10 o'clock and we'll move straight on to any unfinished chemistry, which will be topic 5 chemistry. Okay, so we're going to take a break, and we'll be back in 5 minutes. So we'll restart at 10 minutes past 11. Thank you, everyone. Okay, welcome back everyone. Let's finish off chemistry topic 4. Okay, before we start, there's just a question here that says, just asking about paper 2 content. So paper 2 content we are going to finish. We're going to complete in May half-term holiday. So when we go to breaks, if you look at when it says register for AMALathon 2, in AMALathon 2 we're going to have 2 sections to it. Before every single exam, we'll have a 3-hour session where we'll be doing lots of past paper practice linked to topics that our analysis has shown to be the most likely topics that will come in the exam. Now in the past, I've always been around 80%, minimum 70% accurate. So we'll be going through that in 3 hours on the weekend before each exam. So we'll be covering foundation, higher and triple award questions. And then that means we'll have a session on the weekend before the biology paper 1, on the weekend before the chemistry and physics paper 1. So that'll be 3-hour sessions. Then during the half-term holidays in May, I'll be doing the same kind of AMALathon that we just done now for paper 1, but we'll be doing it for paper 2. And after the May holidays, you'll have your paper 2 exams. So the weekend before each one of those paper 2 exams, and one of those weekends we'll have to do 2 sessions on a Saturday and a Sunday because you've got both exams in that week. Then we'll have the pre-exam workshops where we'll be going through lots of past papers. So I hope that answers the question, and it'd be nice to see you all at those sessions too. So if you want to join us, you need to register below. There'll be a charge for that one. It's not expensive. I think you'll have to check with the admin team. I think it's £108 for the 36 hours because we'll be doing 18 hours because it's 6 exams, and we'll be doing a 3-hour session before each exam. So that's 18 hours of pre-exam workshop, and we'll be doing another 18-hour AMALathon. And as you know from this one, it hasn't been 18 hours. We've done quite a bit of extra time as well. So we did a 1-hour extra session yesterday and today. So it works out to be 21 hours. But it'll only be 18 hours that we're charging for. So I think it works out to be, so it's 36 hours and we're charging £3 an hour. So if you can make it, brilliant. We'd love to see you all there. And please let others know if you've enjoyed it. When we finish, please give us a positive review, or please give us some constructive criticism so we can improve even further. It is AMALearning, like I've said to you guys before, the aim of AMALearning initially, our long-term goal, or one of our big aims, is to make education affordable. So we're currently in the process of making an app which hopefully will be released next year. The whole YouTube side just was a bit of a side effect. It wasn't something we were aiming for. But the videos that we started, the animations that we started to make for the app, we just thought it's probably good if we put it on YouTube so other people can take benefit until the app's made. And then we had an idea about doing the AMALathon, where one, it would help us fulfill one of the goals where we're making education affordable, or in this case free, because we do have a lot of subscribers that come from Pakistan, India, China, some parts of the Middle East, Indonesia, Malaysia, Philippines, and some of the kids in some of those countries. I'm not including the Middle East in this, especially I suppose Pakistan or India. You do have students who are logging in, but they wouldn't necessarily be able to afford the paid sessions. This is the first time we're doing this, so any constructive feedback at the end will be really appreciated. Otherwise, I do sessions like this all the time on Zoom for my students inside school and outside of school. So what is oxidation and reduction in terms of metal, when a metal is oxidized? And as you can see, this is part of the reason why we were going to give revision notes for, we were going to provide the notes for the session, as in every single PowerPoint. They're not PowerPoint, sorry, they're Canva presentations. But for whatever reason, when things have gone across and when some amendments have been made, there's things out of place, out of sync. Some sentences have gone out of sync. So for that reason, we've not shared them yet. There's a lot of amendments that need to be made. I've been doing them live and I've been trying to sort them out prior to sessions, but you've probably come across a few of those amendments. Okay, so what is oxidation and reduction in terms of oxygen? So when a metal reacts with oxygen, it becomes a metal oxide. When a copper reacts with oxygen, it becomes a copper oxide. When copper and oxygen, we went through this yesterday. We are on slide 12. When we do share them, then it'll be through our WhatsApp channel. So as soon as they're ready to share, if you go to our WhatsApp channel, but as long as you've joined our WhatsApp channel, then all the information will be there. So how does reactivity affect how we extract metals? So if it's more reactive than carbon, then we do electrolysis. If it's less reactive than carbon, so all up to copper, then we use, I should have done it on this side, then we do reduction with carbon. And what that means is the carbon takes the oxygen, it takes all the other bits away, leaving a pure metal behind. And then you've got things like silver and gold, which are just found like that. They're found pure. So when you go into a mountain and start digging for silver and gold, or gold mining, you find pure pieces of silver and pure pieces of gold. So gold and things like unreactive metals like gold are found pure as pure metals in the Earth's crust. But most metals are found as compounds. So then we have to extract them. Now, if the metal is less reactive than carbon, then we can heat with carbon and that removes all the impurities, all the extra bits, the oxygen, etc. And that's called reduction using carbon. So essentially what I've just explained. Now, carbon is more reactive than some metals like zinc or iron. So the carbon displaces the zinc or it displaces the iron and takes the oxygen away from them. And that is a redox reaction, because in this case, the carbon is taking the oxygen, it gains an oxygen, so it's been oxidized, and the metal has lost oxygen, so it's been reduced. Now, I did go through this yesterday, but it is important that I go through it with you now. So I'll go through it with you again. Now, you need to be able to do oxidation and reduction from a half equation perspective. So every equation that we look at now, we'll start looking at from a half equation perspective. We did it yesterday, but we're just going to keep doing it because it's something that needs to be embedded. Now, if those that are on the chat, if you can guys, if you're happy to give me an indication of how well or how confident you feel with half equations, I'd appreciate it. You don't have to write anything. My assumption with all these will always be that the more we do, the better. So if you agree with that, you don't have to give any additional comment. So now silver is Ag+, nitrate is NO3-, and we add to that zinc. Now zinc has no charge because it is an element, so the electrons and the protons are balanced. Then we've got zinc 2+ again, sorry, not again, but just zinc 2+, nitrate NO3-, and silver. Now this is an element, so it has no, okay, thank you Nadia, and it has no charge. Why does silver and zinc not have a charge, because they are elements. Anything that's an element, anything that's on the periodic table, has the same number of protons and the same number of neutrons. Sorry, same number of protons, same number of electrons. That means they have a charge of 0. Now, once we've broken everything up into ions, the first thing we do is we delete anything that looks the same on both sides. So what looks the same on both sides? NO3 minus. So we can delete those. Those are spectator ions. Now what we're going to do is, we're going to now look at each individual element. So let's start with silver. Now on our left hand side, our reactant side, we've got Ag plus. Let's put the arrow in the middle. On the right hand side, we've got Ag, silver. And so that's, we've looked at this and we've looked at this. Now let's do another half equation. Let's look at zinc and Zn2 plus. So we've got Zn on this side, and the other side we've got Zn2 plus. Now what we've got to say to ourselves is, if this is Ag plus here, what has to happen to Ag plus for it to become just Ag? We've got to get rid of that positive. How do we get rid of a positive? So if you've got +1, how do we make it 0? We do -1. And in science, in chemistry, how do you -1? You add an electron. So now that plus and the minus cancel out, and we've got Ag, silver. Now that silver, that Ag plus, has gained an electron. So remember, oil, oxidation is loss of electrons, oil, and reduction is gain of electrons, in gaining electrons. So here, that has been reduced. Now let's look at zinc. What does zinc have to do to become zinc 2 plus? Think of everything in group 1 or group 2. Why do they become +1 or +2? What do they lose? If we want to become positive, what do we have to lose? We have to lose our negativity. So that means this zinc here has to lose electrons. Now, to become 2 plus, specifically, how many electrons does it have to lose? It has to lose 2 electrons. So we always put the lost electrons on the product side, because it's a product, it's something that's being lost. Now, if something loses electrons, then we're on oil. Oxidation is lost. So that is oxidation. And this is why it's called redox, because look, redox. One thing's been reduced and one thing has been oxidized. Now I'm going to erase everything I've just shown so you can see the presentation and how the presentation shows it, because I know the way I'm writing things sometimes can be a little bit rough. So here we go. The silver, so look, the first thing they've done is they've removed nitrate ions, because it's on both sides. So they've rewritten it without, I just crossed them out. That's the easiest way for me. And again, half equation here, Ag plus electrons, so that means that's been reduced. Zinc becomes zinc 2 plus 2 electrons, so that's been oxidized. So now, whenever an acid reacts with a metal, in actual fact, it is a redox reaction. So they could ask you questions linked to redox, particularly if you're doing higher. Not particularly 100, but it only occurs in higher. It's not something that will come in foundation. So redox reactions, you need to have that understanding of half equations for, because they could potentially take an acid reaction with a metal and ask you a redox question. But when an acid does react with a metal, it makes a salt and it makes hydrogen, which is what we're looking at now. So zinc and hydrochloric acid make zinc chloride and hydrogen. And what they're saying here is the hydrogen gains electrons, so it's been reduced. The zinc atoms have become ions, so it's gone from zinc to zinc 2 plus. That means it's lost electrons, so zinc has been oxidized. So these kind of sentences do help, Because if you've gone from an element on this side to part of a ionic compound on the product side, then automatically you are this. As in, it's lost electrons and it's become oxidized. If you've gone from this side and it's become an element in the product side, then that means electrons have been gained. They've been reduced. So if you're struggling with half equations, at least understand it from that perspective. If it goes from element to ionic compound, it has been oxidized, it's lost electrons. If it's gone from a compound to an element, then it has gained electrons and been reduced. So a neutralization reaction happens when an acid reacts with a base to form a salt and water. So for example, here we have hydrochloric acid, which is HCl, sodium hydroxide, which is NaOH, becomes sodium chloride plus water, H2O. What determines the name of the salt? So if something reacts with hydrochloric acid, so if a metal reacts with hydrochloric acid, it becomes a chloride. If something reacts with sulfuric acid, it becomes a sulfate. If something reacts with nitric acid, it becomes a nitrate. If it reacts with ethanoic acid, it becomes a ethanoate. Now, this is not very common. I've never seen a question linked to where you've had to write ethanoate. But the other three, very common. So, let's predict products in the following reactions. So we've got magnesium oxide and hydrochloric acid. So what we're going to make here is a salt plus water. Whenever an oxide reacts with an acid, it makes salt plus water. But what's the salt? It's magnesium chloride plus H2O. Now if you've got a carbonate, when a carbonate reacts with an acid, it makes salt, water and carbon dioxide. So if it's an oxide, what do oxides make? Salt plus water. What do carbonates give us? Salt, water and carbon dioxide. And what do we name the salt? Magnesium nitrate. So whenever it's nitric acid, it's a nitrate. Here we've got sodium hydroxide. So whenever it's magnesium, when it's an oxide or a hydroxide, it always makes water and salt. Now we've got sodium and sulfuric acid, so that's going to be sodium sulfate, name the salt. Here we've got copper oxide and hydrochloric acid, so it's going to be copper chloride. And because it's an oxide, we automatically get water. Lithium carbonate. So the carbonates always make three things: salt, water and carbon dioxide. We've got lithium and chloride, the chloric part, so that's lithium chloride. Sodium hydroxide, so that's the same as this one here. It's going to make salt and water, so we've got sodium nitrate. Okay, so it's important that, and it's important because these are common questions. So it's important that you can write down this as a word equation. That if it says you've got magnesium, you've got an oxide and hydrochloric acid, you know that it makes magnesium chloride plus water. If you're given lithium carbonate, for example, you look at that and say, oh, it's a carbonate and an acid, so automatically we're going to get a salt, water and carbon dioxide. Now water, carbon dioxide we know, but what's the salt? And you know it's hydrochloric acid, so you know automatically it's a chloride that's reacting. So we've got acid, a substance that releases hydrogen H plus ions when dissolved in water. So, and we know something is an acid when it releases H plus. So the acid part of any solution, like hydrochloric acid, in fact, let's quickly look at this. Let's look at some examples of acids. We've got HCl, H2, so HCl is hydrochloric acid, H2SO4 is sulfuric acid. HNO3, nitric acid. What's common in all three? H+. In alkali, it's a type of base. So if a base dissolves in water, then we call it hydroxide. It's the OH minus part that makes it a base. So for example, NaOH, LiOH. Now, a base is a substance that reacts with an acid to neutralize it. The difference between a base and alkali is the base is a solid. We only call it so it's a solid. We only call it an alkali when it dissolves. So not every base dissolves and becomes an alkali. Neutralization is when an acid and a base react to form a salt and water. Now, if you think about it, what represents an acid? What part of the acid is an acid? It's H+. And what part of an alkali is alkali? OH minus. Now, if you bring those together, what does that make? We've got 2 hydrogens and we've got 1 oxygen, so H2O. The plus and the minus cancel each other out. And pH scale is a number scale from 0 to 14 used to show how acidic or alkaline a solution actually is. So this is a really good question. It says the apparatus shown in the diagram was used to investigate the rate of reaction of excess marble chips. Now marble is calcium carbonate, CaCO3. And it's excess of marble chips reacted with dilute hydrochloric acid. So that means we've got CaCO3 reacting with HCl. So let's do it here. Notice they've given us all the information. CaCO3 plus HCl, and the salt formed is CaCl2. That doesn't seem like it's complete, so we're definitely missing something. When a carbonate reacts with an acid, it makes 3 things. It makes a salt, so we've got calcium chloride, the salt, but it also makes H2O, and it also makes carbon dioxide. Now the only thing left to do is balance it, and this is actually not too difficult a balancing, because if you see here, how many chlorines do we have on the left hand side? Over here we've only got 1 chlorine, but over here we've got 2 chlorines, so that means we need to double up. So we put a 2 there. And now if you were to count all the calciums, all the carbons, all the oxygens, all the hydrogens, all the chlorines on the left and the right hand side, it's now balanced. So always start with the imbalanced, the one that isn't balanced, and then work your way from there. So here we had 1 chlorine, here we've got 2 chlorines. So we dealt with that imbalance first, and just by dealing with that 1 imbalance, because also we've got 1 hydrogen here and 2 hydrogens here, we dealt with both those imbalances in one go by adding a 2 before the HCl, and the whole thing's become balanced now. I'm just going to erase this and show you the actual answer. Oh, the answer's gone. Okay. So as I was explaining to you with some of the issues, I amended this just last night, and all the amendments have gone. So we do need to look into why this is occurring, because we've transferred all our PowerPoints to this format, so it had our branding on. But we have understood now. The technical team has understood anyway how to sort the problem. So by the time we get to AMALathon 2, we won't have any of these issues. You will get the notes at the beginning, and you won't have this kind of problem. Like I said, this is our first go, so we knew that we'd make some errors and some mistakes, but we'll correct all those errors and mistakes by the time we get to AMALathon 2. But as long as you guys are understanding what I'm saying and you're taking benefit, then we're at least heading in the right direction. So how do we use this equipment to make pure dry salt. So I'm going to explain it using this, and we'll go through the theory in a second. This beaker has got acid inside it. Now what we want to do is we want to react out all the acid. So we get spatulas full of metal oxide, for example, a base. So something like copper oxide is a base. This could be H2SO4. And we're adding to it copper oxide. Now what that's going to produce is copper sulfate plus H2O. But we want to completely react out all of the acid. So you put in one spatula of copper oxide and then you mix. That ensures that the reaction occurs. So we stir it. Then we add a second one, spatula, a third one, a fourth one, a fifth one, sixth one, until we start to see an excess of copper oxide at the bottom. Copper oxide is a black powder. So we start to see the excess at the bottom. Now, that excess at the bottom we don't want, but what we now know, this liquid is copper sulfate and water. So now the next thing we do is we take this whole solution and we put it through the filter. And what's left in behind is the copper oxide, the excess copper oxide. And in here, we have just copper sulfate and water. Now to make pure dry crystals, we've now got to remove the water. And how do we do that? We do crystallization. So we put our liquid into evaporating dish and we start evaporating off the water. The best way to do this is in a water bath because it maintains the temperature. The best way to do this is at a temperature below boiling point. So at this point, what you now do, when you get to the point where a lot of the water has evaporated, and you have a saturated solution, meaning with the small amount of water that's left, there's no more space for the salt. So you're starting to see the whiteness or the color of the salt. The blue, in this case, copper sulfate is like a bluish color. So this is the slight bluishness in the liquid that's left. So what you can do is you can take a small splint and dip it in, and if you start to see a little bit of the blue color on the splint, then you know it's time to put it on a windowsill, or you can put it in a water bath and let it now naturally just evaporate off. And the longer it takes to evaporate, the bigger the copper sulfate crystals you get at the end. So that's exactly what this whole process is saying. So I've still shown it there if anyone wants to read through in their own time. But what I've said was sufficient. Now this is how you'd answer this 6-mark question. So describe a method to make pure dry crystals of magnesium sulfate from a metal oxide and a dilute acid. Now, if we say metal oxide and we're making magnesium sulfate, we have to therefore use magnesium oxide. What's the only place we're going to get a sulfate from? Which acid gives us a sulfate? Sulfuric acid. So this question, the first marks are for identifying what we need to add. Beyond this is that we're going through the same process I've just gone through. So now we're adding magnesium oxide in excess to the warmed sulfuric acid. You don't have to write warmed, but sulfuric acid, until all the acid has been reacted out and we've got excess of magnesium oxide at the bottom. And obviously every time we add a spatula in, we stir it, and that helps the reaction happen better. And then we want to remove this extra magnesium oxide, so we use the filtration process. In the filtration process, all the excess magnesium oxide is left behind, and we end up with a filtrate. We end up with a filtrate at the bottom there that has magnesium sulfate inside it and water. Now we need to remove the water, so we do crystallization. So we heat the solution in the evaporating basin until crystals start to form. The best thing is to use a water bath, so it's at one temperature. Leave it to cool so the crystals grow, and once you've done that, you just pat dry the crystals and it's done. That's your 6-mark answer. Now most people will have understood the process, but the second they see magnesium sulfate, that you've got to make magnesium sulfate, they go they go blank. And that's why understanding is really important here. How do we make magnesium sulfate? We can react. So if you had magnesium carbonate and sulfuric acid, you would make magnesium sulfate. If you reacted magnesium oxide with sulfuric acid, you would also make magnesium sulfate. If you reacted magnesium hydroxide with sulfuric acid, you also make magnesium sulfate. So, but in this question, it's specifically telling you it needs to be a metal oxide. So therefore, magnesium oxide. Now in another question, another 6-mark question, particularly in a higher or a triple award, they might just say, how would you make magnesium sulfate? You have to decide then if you want to use magnesium carbonate, magnesium hydroxide, magnesium oxide. But the outcome's the same, so it doesn't make a difference which one you use. So how does this method ensure a pure salt is made? We're using an excess of the base, and that means all the acid has completely reacted. We're filtering the solution to remove any excess unreacted base. Then we're heating the filter solution gently to remove the water. Then we're leaving it to cool slowly to again remove all the water. So the only thing we're left with at the end is crystals, salt, the pure salt crystals. Even then we pat them dry just to make sure there's no remaining water. So how could the student know when all the hydrochloric acid has reacted? How could the student know when all the hydrochloric acid has reacted? They would know because some of the solid still remains. How could the student... So the whole point here is that why is this solid left behind? Because now there's nothing left to react with that solid. So if there's nothing left to react with the solid, that means the reaction is complete. How could the student obtain the zinc chloride solution? As in, how can we separate now? So how could the student obtain zinc chloride solution from the reaction mixture when all the hydrochloric acid has reacted? So how are we removing the excess solid? We filter it. And now we've got the crystals. Now that we've got a solution, a zinc chloride solution, how do we end up with the crystals? We just heat it and then we leave it to crystallize. So the whole crystallization process. So how do we know the reaction is finished? We can see that there's some solid left behind. That means all the acid has reacted. How do we now separate the excess from our solution, our zinc chloride solution? In this case, it's zinc chloride. We filter it. That takes all the solid out. And then what do we do next? We heat it, or we just go through the crystallization process, evaporate all the water off slowly, so we're left with crystals. Okay, pH scale and neutralization. So acids are, like we said before, they release H plus ions. So when you put an acid, like for example, you put HCl into this solution, into water for example, it dissociates. Dissociate means it breaks apart into its ions. So it becomes H plus and Cl minus. So that means it releases the H plus ions. Now with a alkali, for example, let's say you've got NaOH, sodium hydroxide, the same thing happens when you dissolve it into water. It breaks it apart into the Na plus and OH minus, and it's the OH minus that causes the alkalinity. Now, how can the pH be determined? This is something that I'm sure everyone's familiar with, but if it's between 0 and 6, it's acidic. If it's 7, it's neutral. 8 to 14 is alkaline. So I'm not going to waste your time with that. So how can the pH of a solution be determined? How can it be determined? You can use a pH probe. Now you can use lots of different, like you can use universal indicator, but that's not as accurate as a pH probe because that gives you an exact pH number. So it might be, for example, 3.3, 4.2, whereas universal indicator is based on your judgment. So you might say, oh, it's 3, it's 4, there's no in between. Explain what the color that universal indicator turns to tells us about the substance. So if it turns red, orange, or yellow, or a lightish kind of green, it indicates it's an acidic solution. If it's green, that's neutral, and if it's blue, purple, anything in between, it's alkaline. Okay, so we've said acids release H plus ions, alkali releases OH minus ions, and when we mix the two things together, we get neutralization because the H plus reacts with the OH minus to give us H2O, gives us pure water. That's why water is classed as neutral. So write the simplest ionic equation which represents the neutralization reaction, and that's H plus OH minus becomes H2O. Write an ionic equation to show how water is formed in this reaction and state the sources of the ions. So it says here, write an ionic equation to show how water is formed in this reaction and state sources of the ions. So we're talking about a neutralization reaction here. So H plus comes from the acid, the OH minus comes from the alkali, and when they react together it makes H2O, water. Okay, now titration is specifically triple award. So I'm going to go through titration. I know I said I wasn't going to go through anything in the morning, but I will go through titration in the morning. Just in case we've got pupils here right now that have not are not doing triple award. So titration, we need to go through the process, what titration is used for, and then we've got some really good. This one is an amazing question, a really good past paper practice question. This is that I definitely want to take time to go through with you. So tomorrow's session, we're going to start at 9:30, and we specifically just to go through. Actually, no, sorry, I'm saying 9:30. We don't need to do 9:30. Sorry, 9:45. So I'll be using unless questions and answers come up. I do mean questions and answers linked to the spec. So at 9:45, I'm going to go through 4.2.5 chemistry, which is titration. So a strong acid is one that completely dissociates or breaks apart, ionizes in aqueous solution. So what we mean by that is HCl, HNO3, H2O, so forth. So we again look at HCl. When it enters, so I'll actually do it here. HCl, when it enters water, it becomes H plus and plus Cl minus. Now notice this reaction is going in one direction, meaning all of that HCl dissociates and breaks apart to make H plus and Cl minus, meaning all of the H plus is available to act as an acid. Now when we say a weak acid only partially breaks apart or dissociates in aqueous solution. So if you look at ethanoic acid, it becomes H plus and CH3COO minus, but here we have a reversible reaction. Now if it's a reversible reaction, that means H plus and CH3COO, they are reacting to make more ammonia, to make more ethanoic acid. At the same time, the ethanoic acid is breaking apart to make more of the H plus and CH3COO minus, meaning at no point is every H plus available to add to the acidity. So with a strong acid, it completely breaks apart. So all the H plus is available. But in a weak acid, a certain percentage of the H plus is available because it's a reversible reaction. So that's why we say it only partially breaks apart, partially ionizes. So examples are ethanoic acid, which is vinegar, citric acid, which is found in lemons, carbonic acid, which is found in fizzy drinks. So strong acids is an acid that fully ionizes in aqueous solution, so it releases all the H plus. A weak acid is an acid that only partially ionizes, so it only releases some of its H plus ions. Now, some people assume dilute means weak, but they are two different things, and you've got to understand the difference. When we say strong and weak acid, we're talking about how much H plus is being released. When we say dilute, we're talking about how much water has been added to it. So you can get a strong acid, you can add loads of water to it, and it has now become dilute. So dilute means an acid that contains a low concentration of acid particles in a large amount of water. So if I've got a strong acid, and for example, I put a small amount there, now I add lots and lots of water to this. What have I done to my acid particles? I've lowered the concentration of the acid because I've made it spread out through all the water. So within a specific space, there's now less H plus ions. I've spread them out, but it's still a strong acid because it's fully dissociated. Now, if you look at concentrated acid, it's an acid that contains a high concentration of acid particles with very little water. So you can have a weak acid that you don't add much water to. So for example, vinegar is predominantly acid, ethanoic acid. So that's concentrated acid, but it's a weak acid. Whereas if I add water to that vinegar, then it becomes dilute. So I hope that makes sense. It's important to be able to differentiate between the two. So pH tells us how many H plus ions are in the solution. So if we've got more H plus, then we have a lower pH because it's more acidic. If we have less H plus, then it has a higher pH, like vinegar and citric acid, etc. And then that has a higher pH. Now that doesn't mean it's more alkaline. It just means it's closer to where the alkaline bit starts. For alkaline to start, we need more OH minus. So what would be correct here is that it's less acidic. So stronger acids have more H plus ions and a lower pH. Now if two acids have the same concentration, the stronger acid will have the lower pH. Now each pH unit change equals 10 times difference in the H plus ions. Now I'm going to explain what I mean by this. And what I mean by this is as follows. So let's say I said at pH 5 we have a concentration of 0. Actually, I'll do it here. So we've got pH 5, pH 4, pH 3, pH 2. Now the concentration of the acid at pH 5 is 0.005 grams per decimeter cubed. For it to become pH 4, and we're talking about grams per decimeter cubed of H plus. So it's got this much H plus inside it. For this to become pH 4, I've just got to multiply that number by 10. So pH 4 has 10 times more H plus. So that's going to be 0.05. pH 3 is has 10 times more H plus than pH 4. So pH 3 is going to be 0.5. pH 2 has 10 times more H plus than pH 3. So that's going to be, again, 10 times. So questions like this do come up. So it is important to understand that the difference between one pH unit and the next one. So when it becomes a stronger acid, what we're doing each time, we're multiplying by 10. So to go from pH 4 to pH 3, we times by 10. pH 3 to pH 2, we times the concentration by 10. pH 2 to pH 1, we multiply the concentration by 10. pH 1 to pH 0, we multiply concentration by 10. So let me repeat that. If we want to go from a pH 6 to a pH 5, what do? To the concentration, we multiply it by 10. If we want to go from pH 5 to pH 4, what do? We multiply by 10. If we want to go from pH 4 to pH 3, we multiply by 10. I know some of you probably get sick of me saying this, but I want to really drill this in to everyone's minds so it's properly understood. How do we go from pH 3 to pH 2? We multiply by 10. How do we go from pH 2 to pH 1? We multiply by 10. How do you go from pH 1 to pH 0? We multiply by 10. I hope that makes sense. Hope everyone's understood that. So well done, everyone. Now, electrolysis, we are going to complete tomorrow. So that's going to be at 10 a.m. So at 10 a.m, we're going to start from 4.3.1, electrolysis. Okay, so we're going to take around a 5. Also, let me just answer this question really quickly. What we're multiplying by 10 is the concentration, not the pH, the concentration. So concentration of H plus. Thank you, Nadia, that's a really good question. So for example, if it's pH 5 and the concentration of H plus is 0.005 grams per decimeter cubed, if I want pH 4, what do I do to this number? I times it by 10. So now I've got 0.05 grams per decimeter cubed. Now if I want this to become, I want the solution to become a pH 3, I need to have 10 times more H plus. So we've got times 10 of this concentration. So it would be 0.5 grams per decimeter cubed of H plus. So each pH unit, as you get more and more acidic, is 10 times difference in H plus to the concentration of H plus. Okay, thank you everyone. I hope that makes sense now, Nadia. Otherwise, I'm happy to go through it again. Okay, now we're going to restart and we're doing our topic for physics. And we're going to move on to topic for physics. We're going to move on to topic for physics at 12:05. So we're back in 5 minutes. Thank you everyone. See you in 5 minutes. Okay, welcome back everyone. So we are on our final topic for physics, which is atomic structure. Now the first part of atomic structure you're all going to be really familiar with. So we're going to brush through it, and if anyone has any questions because they possibly didn't understand it when we covered it in chemistry, because it's the topics we're about to cover right now are the kind of topics that we've already covered in chemistry. So if anyone comes across something that they want me to explain again, just say and I'll happily explain it again. So what's the structure of an atom? So we know from Rutherford's experiments that they worked out that the atom, the nucleus is about 1/10,000th of the size of the atom. Just get the pen. It's 1/10,000th of the size of the atom. So that's something we've already gone through. And like I said, please, you are more than welcome to ask as many questions as you need. So if there's anything, just keep asking. And we know that the majority of the atom's mass is found in the nucleus. We know that electrons are positioned in energy levels at various distances from the nucleus. We know, now this is something I'm going to just quickly emphasize because this is really important. When an electron gains energy, so if it absorbs, for example, radiation, then the electron becomes excited and it goes to a higher energy level. Now eventually, that electron, the higher energy level, will lose that energy, and when it loses that energy, it emits electromagnetic radiation, for example, gamma rays. And then the electron, because it's emitted that, it's lost the energy, it drops back down to its original place. So let me just repeat that. When the electron absorbs energy, it moves further away from the nucleus. Now, when the electron releases the energy as EM radiation, the electron falls back to its original place. So when the electron moves closer to the nucleus, the energy is released. And usually it's released in the form of gamma rays, X-rays, ultraviolet rays. So why are atoms neutral? So again, we've covered this before. They're neutral because it has the same number of protons. An atom has the atoms of an element have the same number of protons and the same number of neutrons. We know the top number is mass number, the bottom number is the number of protons. And how do we work that out? Protons is the bottom number, so that bottom number, that's 11. Electrons is always the same as the number of protons in an atom. And neutrons is top number take away bottom number, mass number take away atomic number. So 23 take away 11 is 12. So that's something we've covered in a lot of detail in chemistry. This is topic 4 in physics, atomic structure. So this is the topic that moves on to radiation. So isotope is when, for example, carbon-12 and carbon-14, they've got the same number of protons, but they've got a different number of neutrons. Now it's important to understand what isotopes and ions are, which is why this topic starts this way. So now if an atom loses an outer electron, it becomes a positive ion. And if an atom gains an electron, it becomes a negative ion. Now the developing the model of the atom. How has the atomic model changed over time? So initially Dalton thought it was a tiny sphere. J.J. Thomson discovered the electron, so he thought it was these negative. If we look at this instead of plum pudding model, we look at it as a cookie, then the chocolate chips in the cookie are negative and the rest of the cookie is positive. But they called it a plum pudding model because that was the chocolate chip cookie of their time. Then we had the alpha particle scattering experiment where they had a thin piece of gold atoms, for example, a gold leaf. So they had a thin layer of gold atoms. They fired alpha particles at it, and alpha particle is the nucleus of a helium atom. So it means it's 2 protons and 2 neutrons. And they fired that. Some bounced straight back, which showed that there must be a tiny heavy center. Some were deflected, and because this helium has a 2+ charge, it means the nucleus must have a positive charge because it deflected it. It didn't attract it, and most of them went straight through, and that indicated that most of the atom must be empty space. So scientists started to think the atom looked more like this. So scientists thought it looked more like this. Now, then Niels Bohr, he suggested that electrons orbited around the electrons moved around the nucleus in fixed orbits. So he's the one who came up with the understanding of electron shells, Niels Bohr. And then finally James Chadwick discovered neutrons, and he discovered that the neutrons were inside the nucleus. Okay, radioactive decay. Okay, so radioactive decay. So by the way, we are from I think it's 4.3, it becomes triple award. But I'll point out when it becomes triple award, at that point, those that are doing combined can log off. So radioactive decay is the process by which unstable atomic nuclei lose energy by emitting radiation. So what it means is the inside of the atom, the nucleus of the atom, is unstable. So it releases something. So it can either release an alpha particle, and an alpha particle is the helium nucleus. It can release a beta particle or gamma radiation. So a beta particle is just a fast moving electron, and gamma radiation is just a transverse wave. It can also release a neutron, which is what we have in the nucleus. And when it releases it, it just calms. The atom becomes more stable. So it has to release release something to stabilize. So the example I can give you is this. Let's say we've got a table, and that table fits 4 people. So you're at school, and in the science lab, you might have tables that fit 6 people, 5 people, or 4 people. So let's just use a simple one. There's room there for 4 chairs. Now, a 5th person squeezes in, and a 6th person squeezes in over here. And that's fine. That's not too much of a problem. But a 7th person, and there's a wall here, a 7th person now tries to squeeze in here. Now, the second that 7th person squeezes in, how are we going to feel when we've got our books out and we're all trying to work? When the 7th person comes in, it's going to feel really cramped. Everyone's going to be moving back and forth. It's going to feel uncomfortable. What's the only way to make things comfortable? It is eject one person. Now, the second we eject, for example, this person now gets ejected, now there's enough space again and things calm again. And that's what's happening in the nucleus of something that's radioactive. It's unstable because it's got too many neutrons inside it. It's got too much energy inside it. So everything's moving around, it's unstable. Then all of a sudden, and this is a complete random process, it emits some of that radiation. And when it emits that radiation, everything calms. So I hope that makes sense. I think that for myself, it's just a nice, easy way to understand it. So if you look here, you've got uranium, which has an atomic number of 92. So every other subatomic particle inside the nucleus is a neutron. So it has a lot of neutrons. Now, for it to become stable, it releases an alpha particle, and because it's now released 2 protons, it's become a different element, but that's more stable. Now, what's activity and count rate? Activity is the rate at which a source of unstable nuclei decays, and it's measured in becquerels. By the way, with this topic, because it is a topic that's not, I think if it's understood, if it's taught properly, it's really well understood. So I'm hoping, but I want to take my time with it and make sure everyone understands it properly. So please ask lots of questions if it doesn't make sense. Now, tomorrow, like I said, at 9:45, we'll be covering titration. At 10 a.m, we're going to finish off electrolysis, so we'll finish off topic 4. Then we're going to want to move on to topic 5, chemistry. And when we finish topic 5, chemistry, that will be the end of our session. Unless we don't finish this physics now. So if we don't finish this physics, whatever we don't finish from this physics topic now, will be done after topic 5, chemistry tomorrow. And then that will be the end of the AMALathon. But like I said, at the end of the AMALathon, there will be a form that we're just making where you can give us some feedback. And like I said, I will in about a week's time do a post which will invite you guys to ask questions if you need. So if you ever come across a question that doesn't make sense, for example, you're doing the 2020 paper and question 6 doesn't make sense and you just want it to be explained to you, I'll just make a quick short. Either I'll type it in, but for myself, I think it's easier if I just make a quick 30-second, 1 minute, 2 minute video, and we can put that onto YouTube for you and you can watch it. So we'll keep responding to your questions. Now that you're an AMALearning student, you're our student till you finish your GCSEs. So count rate. So activity is the rate at which an unstable nuclei decays. So it's how much radiation is coming out of it per second, and that's measured in becquerels. Count rate, on the other hand, is the number of decays recorded each second by a detector. So here's the different types of radiation, and let's highlight, there's a summary page as well, so we'll highlight this again. But alpha particles only travel 3 to 5 centimeters in the air, and the reason is it's 2 positive protons and 2 neutrons. So the second that collides with the air particle, it knocks out one of its electrons from whatever it's collided into. So it ionizes it. And also when it's collided with something, then it's lost its momentum. It stops moving beyond that point. So it's the most damaging because it ionizes everything around it. It collides with everything. It has a positive charge as well. So that part has an impact on the ionizing effect as well. Now, it is the most ionizing form of radiation. So if it went inside our body, it would cause massive amounts of harm. But the reality is, if we went close to alpha particles, it cannot penetrate our skin because it can be blocked by skin or a sheet of paper. That's how ionizing it is. It starts reacting with the first layer of paper, of the atoms within the paper. It's represented by this symbol, alpha. Oh, I forgot which pen I was using then. By the alpha symbol, which is A in Greek. And it's made up of the helium nucleus, so it's 2 protons and 2 neutrons. So you need to. Yeah, we're going to go through the dangers, but I'll explain it right now as well. So if for example we've got a carbon bond, if an alpha particle collides with that bond, it can cause that covalent bond to be broken because it ionizes it. So essentially it can cause the breakages of bonds. Also, if you had, for example, a molecule and it's got weak intermolecular bonds between the molecules, between two molecules, the ionization effect of the alpha particle can break down that weak intermolecular bond. But in terms of your exam, you don't need to know it in that level of detail. What you need to know is how radiation is harmful, how it can be beneficial for us, and how it can be harmful for us. And that's going to come in a few more slides. Otherwise, a question that, because the reason I just said that is because the question was, could you explain how ionizing is harmful? So I've explained what I've explained, but if I go any more detail, it's not something that's going to come in your exam. So beta particles have a range of, it says 15 centimeters, but it can travel up to a meter. It's less ionizing than alpha particles. Now, remember what I said to you guys before? It is an electron. So it's a fast moving electron. That means it's less likely that it's going to collide with air particles. But eventually it will collide with something. That's why it can go further than alpha particles. So it's less ionizing. But now we would say it's more penetrating because it can go further. It can be stopped by a sheet of aluminium. And its symbol is beta, which is a Greek letter for B. And it is a high-speed electron emitted when, now it says proton, in the nucleus changes to neutron. It's not. It's actually a neutron splits into a proton and an electron. The electron then is emitted. Now if we look at this diagram really carefully, what we've got here is you've got carbon 14, so we've got 8 neutrons and we've got 6 carbons, so it's a mass of 14. Now what happens is one of those neutrons, so one of these neutrons turns into a proton. So if we had 8 neutrons before and one of those neutrons are turned to a proton, how many neutrons do we have now? We've got 7 neutrons. Now one of those neutrons turned into a proton, so how many protons do we have now? 7 protons. So still we have a mass number of 14, but now we have a proton number of 7. And if you think back, if something has a change in the number of protons, what's changed? It's become a new element. So carbon becomes nitrogen. Why does it become nitrogen? Because we've now got an extra proton. So let me explain that one more time so it's crystal clear. When a beta particle is produced, a neutron breaks apart into one proton and it breaks apart into an electron. The electron is the high speed electron that's emitted, and it's not a proton, it's the neutron. And it changes into a proton. So then this electron is emitted, but that proton remains inside the nucleus. So if you look at carbon 14, it has got 8 neutrons and it's got 6 protons. What happens to one of the neutrons? It turns into a proton. So now we had 8 neutrons, but one's turned into a proton, so now we've got 7 neutrons. We've got 1 extra proton, so we have 7 protons. So our mass number is 7 plus 7, which is 14. But now our proton atomic number is 7. When your number of protons changes, so does the element. So now this is nitrogen. So our carbon has turned into nitrogen and it's released the electron. But now carbon 14 was an isotope. Now it's just carbon. Now it's just now normal nitrogen, which is stable. So carbon 14 was unstable and nitrogen is stable. Now we move on to gamma ray. Now gamma radiation is a type of electromagnetic radiation. It isn't very ionizing. Now when it does lead to cancer, so when it by not being very ionizing means it can go through our skin and then it can be absorbed by our internal organs. When our internal organs absorb the energy, it can cause the weak hydrogen bonds between the bases in our DNA to break apart. Now if our DNA breaks apart, when our body replaces it, potentially it can replace it with a mutation. So the more times our cells have to replace, the greater the chance of a mutation. Now, it penetrates, so it's the most penetrating, long distances through air. It can be blocked by thick lead or concrete. Even thick lead and concrete, there's still a small amount of radiation that goes through, but it does prevent a lot of it. Now that's the symbol there for gamma. It's a type of electromagnetic radiation emitted from the nucleus. So things like X-rays, gamma rays, ultraviolet, visible light, all these are electromagnetic radiation, radio waves, infrared, microwaves. This is all types of electromagnetic radiation. It has no charge and it's not made up of particles, it's the movement of energy. That means it's the least ionizing. It can't knock out something's electron. However, it's still ionizing in the sense that if you have an atom, if an electron absorbs some of that energy, it will go to a higher energy level. So here's a nice summary table with the descriptions. So helium, so alpha particle is 2 protons and 2 neutrons. It travels 3 to 5 centimeters in the air. It can be stopped by paper or skin, and it's the most ionizing. It's the least penetrating, but the most ionizing. Now, beta particles are high-speed electrons ejected from the nucleus, and we've already explained it's because a neutron breaks apart into a proton and an electron. And it can go up to 15 centimeters, but it can go up to, scientists say 1 meter and above in the air, but eventually then it gets stopped by the air. And it's stopped by a thin sheet of aluminium because there's so many atoms there. And it's less ionizing than alpha particles. Then we've got gamma, which is electromagnetic radiation, so it's just the movement of energy. Electromagnetic radiation is, for example, the light that comes from the sun. It travels large distances, but it can be stopped by thick lead or concrete, and it is the least ionizing. So quick exam style question. Explain how the properties of alpha, beta and gamma radiation affect the level of the hazard at different distances. So all this question actually asking us to do is talk about how penetrating they are and how ionizing they are. So alpha is the least able to penetrate something at quite a far distance because it can only travel a short distance. So it can't penetrate very far. Why? Remember, this is explained. So this column here is why. Because it travels a short distance. It's the most ionizing, which means, and why is that significant? Because then it causes is most hazardous at close range. Now, beta radiation is second most penetrating, which means it can travel the second furthest. It's the second most ionizing, so it's hazardous at medium range. And then gamma radiation is most capable of penetrating, so it travels the furthest distance. It's the least ionizing, which means it's the most hazardous at long range. So it can travel quite far and still cause harm. So we've got our 2 isotopes of carbon. All this is now saying is describe 2 similarities and 1 difference in the atomic structure of each particle. So we've got carbon 12, carbon 14. So this is something that we covered in chemistry. They're isotopes, so they have the same number of protons. So in both of them, we've got 6 protons. They have a different number of neutrons. So we've got 6 in carbon 12, but we've got 8 in carbon 14. But it says 2 similarities. So the second similarity that we have is electrons. They have the same number of electrons. because ultimately they're still atoms. I've just read electron twice for some reason. They are still atoms. Carbon 12, carbon 14 are both atoms, which means they're going to have the same number of protons and electrons. So they both have the same number of protons and electrons, but different number of neutrons. Okay, nuclear equations. Alpha particle looks like this. It has a mass number of 4 because it's got 2 protons and it's got 2 neutrons. And there's a 2 at the bottom because it's got 2 protons. So when something emits an alpha particle, the top number changes by 4 and the bottom number changes by 2. So let me give you an example. This is a completely incorrect example. This would never happen in oxygen, for example, but I just want you to see a really simple numbers first. So if we had oxygen, if that released an alpha particle, it would create a new element. So take away 4 from the top number, which is 12, and take away 2 from the bottom number, which is 6. Now if you look at your periodic table, what has a mass number of 12 and has a proton number of 6? It's carbon, because it released an alpha particle. Now if we look at an electron being released, a beta particle, look at the impact it has. It has no impact on the top number, but it takes away 1 from the bottom number. So let's look at carbon 14 for this as an example. So let's say we've got carbon 14. Let me just change back to a pen. Yes, Lily, this is chapter 4 and is linked to radioactivity. The name of the chapter isn't radioactivity, it's atomic. It's completely slipped my mind. But it is radioactivity, yes. So if let's say you've got carbon 14. And we know it has just emitted an electron. That has no impact on the mass number, but -1 impact on the proton number. So it means 0 plus something equals 14, so that's 14. And something take away 1 equals 6, so that's going to be 7. And that is nitrogen. Because look, 7 take away 1 equals 6. So we have to balance it out. So that's how the nuclear equations work. Now what we're going to do is we're going to go through some examples. I'll go through a few. There's lots there, so you can always go back to them and try them afterwards. So the release of various types of nuclear radiation can result in alterations to the mass and or charge of the nucleus. So what happens during alpha decay? So this is the example they're giving here. We've got uranium 235. So let's draw that, write that out. So we've got uranium U235. Now, in your exam, you might get a question that says, this is uranium 235 and it's got a mass number of 92. Write down a nuclear equation that shows what happens to it when it decays an alpha particle. So you would already have this information. Now all you do is put an arrow there, put your helium nucleus here because that's an alpha particle, and then just do the maths. So what's 235 take away 4? It's 231. What's 92 take away 2? It's 90. Now you go to your periodic table and find the atom, the element that has an atomic number of 90, which is thorium. Now when you talk about atomic number, when you talk about the element's name, the top number doesn't matter, the mass number doesn't matter. What stays the same is the number of neutrons. So you're looking at the bottom, what stays the same is the number of protons. So what stays the same is the number of protons. So you're looking at the bottom number. So we know it's got 90 protons, so it's thorium. So what happens during beta decay? So this is what I've already shown you. We've got carbon 14. Now the question might say this carbon 14, it's decayed, it's released some beta decay. So write down a nuclear equation for it. So you write down E as 0 for mass number, minus 1 for atomic number, and then you work it out. So we've got 14, take away 0 is 14, 6 minus 1 is 7, so nitrogen. And that makes sense now. 7 take away 1 equals 6, 14 plus 0 equals 14. So here's some examples. Unfortunately, everything's already been filled in. So when you're working through this in your own time, I suppose you, with a piece of paper, you can cover this part up, or I'll cover it up for you. So when you're going through it afterwards, oh, actually, I'm covering up the wrong thing. So what I'll do for you is I'll cover the answers. So you can pause it at this point in the video and practice. And the next slide has the answers on there. Now to make it a little bit harder for higher ability, I'll delete the helium as well. So you have to remember what particle is being released. So you can have a go with these questions afterwards and see that, just to make sure that you can do it. And here's the correct answers. Okay, so same thing here. These are examples of beta particle discharge. So what would happen to each one? When it discharges an electron, it has no effect on the mass number, but you take away 1 from the bottom number. So let's just do one for practice. Let's try this one. Let's do rubidium. So we've got Rb, which is rubidium. And it's going to become strontium plus a beta particle, which is a 0 at the top, minus 1 at the bottom. We don't need to put the negative there, but it doesn't make a difference if you do. Rubidium is 137 at the top, 55 proton number or atomic number, so it has no impact to the mass number. So strontium, this new strontium will still be 137, but it takes away 1 from the bottom number, so that will be 54. So how does alpha and beta decay affect the nucleus? So this is something we've already covered, but we'll just quickly recap it again. Because the more we recap, the better more likely we are to memorize it. So the alpha particle, the mass number changes by 4, and the nucleus, as in the charge of the nucleus, so the atomic number, the proton number changes by so the particle emitted from the nucleus is helium nucleus. The mass of the nucleus loses 4 units, and the charge of the nucleus has lost 2 positives. So if it's lost 2 positives, the nucleus that's left behind would become -2. With the beta particle, it loses an electron negative, so the nucleus that's been left behind would become positive. but both lead to a formation of a new element. So explain why gamma emission does not change the atomic number of an element. So for this, we've got to look back to what is gamma radiation? What is gamma emission? What's it giving off? And what is it giving off? It's just a movement of energy. So there's no particles involved. So gamma emission does not affect the atomic number because it only involves the release of energy, not the change in protons and neutrons. Beta decay does not cause the mass number of an atom to change. So explain why. Beta decay does not change the mass number because a neutron is converted into a proton. So an electron that's released so that means there's no significant change in mass, because when the neutron turns into a proton, you don't actually have a change in mass number, because neutron has a relative atomic mass number of 1, proton has a relative mass number of 1. So you haven't changed the mass. The electron that's released has an insignificant mass. It's 1/1836. So a proton is 1,836 times heavier than an electron. Now, transmutation is the name given to a process where one element changes into another. Now, this kind of question, because the word transmutation is a new word, so people get put off. But again, don't get put off by new words. Look at the context. It's just saying where one element becomes another element. And when does one element change to another element? When does one element change into another element? It changes into another element when it releases alpha particles or beta particles. So explain and compare how two different types of radioactive decay can cause transmutation. So the question is, all the questions asking you to actually do here is compare an alpha particle to a beta particle. And again, I'm just going to reiterate this point because I know I've already reiterated it many times. It's important to do lots of past paper practice. So now, as you're finishing the activate phase, and we're going to finish tomorrow, it's important to dive straight into lots and lots of past papers. So we already know alpha particles, the number of protons decreases by 2. And both processes result. Now that's different. So alpha particles decrease the protons by 2, but beta radiation increases the number of protons by 1. Both of them result in the change in protons, so that's the same. In alpha radiation, the atomic number is reduced. In beta radiation, the atomic number is increased. In alpha radiation, the nucleus emits 2 protons and 2 neutrons, but in beta radiation, a neutron in the nucleus transforms into a proton. Okay. So this question part can be ignored, but we can fill in the gaps. So we've got bismuth. It releases an alpha particle. So we're taking off 4 from the top number. We could do the second question too, actually. There's no need for me to erase that, because it is a separate question. There's no need for me to do that. So bismuth loses helium, a helium nucleus, alpha particle. So that means this new element will be 208, because you're taking 4 from the top number, and it will have 81 protons, an atomic number of 81, because we're taking 2 from the bottom number. Now, lead is Pb. It has a mass number of 210. Mercury is, I think it's Hg, and we're releasing a helium nucleus because we're releasing an alpha particle. So that means automatically our 210 is going to become 206. Now, off the top of my head, I can't remember. Oh, sorry, this question is... I should always click next. So, it's given us half the equation here this time. So, we know helium is 4 and 2. That's an alpha particle. So 210 take away 4 equals 206. 80 plus 2 equals 82. So that means lead has an atomic number of, proton number of 82. So these are the kind of questions you're going to get. Sometimes you will only get that part there and you just have to finish off the rest of the equation. Okay, half-lives. So half-life is the time taken, so the duration for the time taken for the quantity of nuclei of an isotope in a sample to decrease by half. So it's the time taken for the nuclei of an isotope in a sample to decrease by half. Or it's the time taken for the count rate of a sample containing the isotope to drop to 50%. In order for the count rate to half. So we look here, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Let's just assume there's 16 there. The time taken for that to half and become 8. And then the time taken for it to go from 8 nuclei to 4. The time taken to go from 4 nuclei to 2 and then to 1. Now, with half-life, with radiation, generally speaking, the time taken to half is always the same. So this substance has a half-life of 1 day. So let's look at it here. It has an activity of 100. So how long does it take to go from 100 to half of that, which is 50? It takes from here to here. That's 1 day. Now, how long does it take to go from 50, which is there, to 25, which is there? It also takes 1 day. How long does it take for it to go from 12 to 25 to 12 and a half? So that's there to there. That's also 1 day. So that's half-life. This substance has a half-life of 1 day. How can we determine the half-lives of these substances? So that's 600. I'm going to draw it, then we're going to zoom in. Half of 600 is 300. So let's go across. And then move down. And then we'll zoom in a second. Whatever that number of days is, that's the half-life. So I'd say that's about 2.7, 2.8. Now here, we had a count rate of 1,000. How long does it take to get to a count rate of 500? So let's go across here. Go down. So I'd say that's around 4,200 million years. 4.2. No, 4,200 million years, yeah. Right, let me just zoom in so we can see this clearer. Yes, so let me zoom in a bit more. That's too much. There we go. Right, so that, yeah, I would say that's about 2.7, 2.8. And yeah, that's about 4,200. So let's check the answer now. So yeah, 2.7 days, and well, that's put it as 4,500. I would put it as 4,200 or 300, but it's fine. You understand the point, I hope. So let's try an exam style question. Now I am going to give you just a little bit of time to have a go at answering this question, because it's a nice question. If it just shows if you understand it or not. So tritium is radioactive. After 36 years, only 10 grams of tritium remains from the original sample of 80 grams. Calculate the half-life of tritium. So if you can work it out and give me the answer, I'm only going to wait about 30 seconds before I start going through it. And I think there's about 10-second delay between what I say and what goes on to YouTube. So about 20 seconds now, I'm going to start going through it. So if anyone manages to get it in those 20 seconds, that's excellent. If you don't, it's not a problem. Okay, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, so let's look at it like this. We've got 80 grams. And then that halves to 40 grams. And then it halves again to 20 grams. And then it halves again to 10 grams. So how many half-lives have we got? 1, 2, and 3. So then those three half-lives took 36 years. So we're just doing 36 divided by 36 divided by 3. So half-life is 12 years. So the number of half-lives is 3, because it goes from 80 to 40. That's the first half-life. The second half-life takes it to 20, and the third half-life took it to 10 grams. So now we're just doing the full time it took, 36 years, divide that by the 3 half-lives, and it took 12 years. Use the graph to find the half-life of plutonium. So let's zoom into this. So originally we've got 1,000, let's go at 500, move across. Now obviously I should be doing this with a ruler, but I can't do it here that well. So it's going up in 5s, so that's 55, 60, 65, 70, 75, 80. So I'd say that answer is going to be around 80 years. So let's check it. Oh, 85. Yeah, 80, 85 years. I think that looks more like... Actually, no. The line that's been drawn really thin there, that does go to 85 years. But my line was quite thick. But that's fine. As long as we understand what we mean by what I'm showing. Okay, radioactive contamination. After this, I think this is the last part of combined physics. So that means the AMALathon for combined physics will have finished as well. So what's the difference between contamination and irradiation? So contamination is the unwanted presence of radioactive material on a surface, on object, or on a person. Now, irradiation, however, so it's the unwanted presence of radioactive material on a surface or an object or on a person. So that means if something is contaminated, for example, if I've got contaminated pencil, within the pencil, it means there are some radioactive nuclei. Now, if we say irradiation, that's the process of exposing an object or substance to radiation without direct contact with the radioactive source. So that means, for example, a doctor, before they do an operation, what they would do is they take the scalpel and everything they need and they put it in a see-through clear bag. So what I'm trying to draw here is the scalpel. I know it doesn't look like that, but just assume that's a scalpel, not a cricket bat. Now what they do is they blast out with some gamma radiation, but it's short. It has a short half-life, gamma radiation. So because it has a short half-life, the gamma radiation hits the surface. Let me show you in a different color. It hits the surface, and then after that, because after hitting the surface, it kills all the bacteria and the germs, including all the germs within that bag. And then, because it has a short half-life, it dissipates. So all the radiation is gone. It just irradiated it. Now, just before the operation, what the doctor will do is he'll cut this open, the bag open, take out the knife, and they can use that knife, and it's clean. And that way, nothing gets passed on to the individual that's been operated on. Now, whereas with contamination, that means it's unwanted presence of radioactive material, and it can be within the material. So what are the differences? When does it occur? Contamination occurs when radioactive material is physically transferred to an object or a person. But irradiation is when an object or substance is exposed to the radiation. And usually because it has a short half-life, it's not too much of an issue. Does the object become radioactive? Yes. The object becomes radioactive. Like I said, irradiation is on the surface. So if you just hit the surface of it, that's irradiation. If something is contaminated, it's on the inside, so the radiation is coming out of it. So irradiation, no, the object does not become radioactive because it's only on the surface. And once that short half-life radiation dissipates and has gone, it's gone. This, however, is on the inside. So until it doesn't, all the nuclei don't break down, it's going to remain radioactive and stopping the radiation. So stopping radiation requires removing the radioactive material from within that material, not just from the surface, but within it. But in irradiation, the radiation stops once the exposure ends, but the object remains unaffected. So the radiation stops once the exposure ends. So it'll have that exposure of radiation. Once it ends, as soon as, because it's got a short half-life, once it dissipates and the energy goes and the nuclei break down, it's gone. It doesn't cause any issues after that. Okay, 4.3.1 is background radiation. This point onwards is all a separate science. So tomorrow, like I said, our timetable for tomorrow, because it's our last day, at 9:45 a.m. we are going to cover in fact, we're going to use our question answer time to finish off the titration. So let's have a quick look at this. So at 9:45 we're going to do chemistry, 4.2.5 chemistry titration. 15 minutes should be enough for that. Then at 10 a.m. we're going to do electrolysis. So that's where our combined students need to be. After that, around 11 a.m, possibly before 11 a.m, we're going to finish off topic 5 chemistry, which is combined and has combined elements, and it has some triple elements too. And then as soon as that finishes, we're going to move on to P4, atomic structure, and we're going to finish off the separate science. So we're going to continue from 4.3.1, and thank you for your question, Akshat. The drug discovery is part of combined science, but the part that came towards the end that was linked to plants, how plants stop themselves from being eaten, etc. That everything from sorry, my monoclonal antibodies onwards was triple award. Okay, thank you very much, everyone. I hope you have a wonderful afternoon, wonderful evening, and I'll see you all tomorrow morning. Thanks for joining us. Take care. Akshat, if you're still there, that's, yeah, having a basic understanding of the fact that medicine comes from different natural remedies is part of your combined curriculum. So having an understanding of a few different examples is needed. There are, if you when you start looking through past paper questions, you'll see actual questions where it'll say which plant does this come from, or it'll give you an example of a plant and say what medicine came from that plant. It's only usually 1 or 2 marks, but it's still worth having. When you start going through the past paper stuff, you'll see. Right, thank you very much. Akshat, are you going to be joining us for the Edexcel section as well? In fact, Akshat, are you doing combined or sorry, are you doing AQA or Edexcel? I mean, you are welcome to both sessions, but I was just asking out of curiosity. Welcome back everyone to day 5 of the Edexcel AMALathon. So today what we're hoping to cover let's just get this presentation up and then I'll quickly go through today's plan. So from a biology perspective, we have finished key concepts in biology. We finished cells and control. We've finished genetics. Today we are going to finish off B4 natural selection and genetic modification. Tomorrow, the main aim is to finish off health, disease, and the development of medicine. So we are a day behind with this. In chemistry, we finished the key concepts. We have finished states of matter, so we're on schedule. We're going to finish this off, topic 3 off today, hopefully, and then do extracting metal and equilibria. And for physics, we finished off P2, P3, P4. Today we're going to finish off P5, and tomorrow we're going to finish off P6. And we've completed this, and today we're going to finish off astronomy. And tomorrow, the priority is going to be to complete B5, B4 and B6. So if the time runs into the triple award time, then it means I won't get a chance to complete C5. So what I will try to do across the next week is sit down at some point and record that separately. But if I've only got combined students and no one requests it, then it's not a problem. Okay, so today we will be completing P7 astronomy as soon as we finish off the other topics. So this we completed yesterday. 4.1b is triple award. So this has already been completed. So we want to move on to 4.2. Now 4.2 and the few points after that are all linked to natural selection. So explain Charles Darwin's theory of evolution by natural selection. Now with natural selection, if you understand the steps involved, it makes it so much easier. So in theory or in hypothesis, evolution is a gradual change in the characteristics of a species over time. It happens through natural selection, where individuals with helpful traits are more likely to survive, reproduce, and pass those genes or traits on to the next generation. So natural selection is a process where individuals with traits that help them to survive in their environment are more likely to live longer, reproduce, and pass on the beneficial traits to their offspring. So here are the key stages. The reason I've rushed through that part is because here's the key stages. Number 1, there's a change in the environment. So every time you get an evolution-based question or natural selection-based question, you go straight to these points. And if you can add these points in, you're going to get the marks. So number 1, there's a change in the environment. So something in the environment changes, like there's a new predator, or climate change, or food. There's a shortage of some kind. So here's a really good example. A few hundred years ago, during the Industrial Revolution, because we were burning so much coal in the UK, it released a lot of soot, and a lot of the trees, the bark turned into a darker colour, as you can see in this picture. Now, if you look closely enough, the black moth was camouflaging, and the white moth wasn't able to camouflage. So, actually, let me start again. Prior to the Industrial Revolution, the most common moth was this one, because it could camouflage on silver birch trees. Then during the Industrial Revolution, because so much carbon was burnt, the soot that was released darkened the trees. So then the black moth had a better camouflage. So this is a change in the environment. The change in the environment here is the fact that the Industrial Revolution started and the trees darkened. Now, we have variations. Variations within the species, it means individuals have differences caused by genetic mutations. So this is something we covered yesterday. So we've got two different alleles here. The allele for black colour and the allele for white or silver colour. Now there's competition. Both of them are competing for food, for mates, etc., for limited resources. But out of the two, which one's going to survive? So the one that would have survived was the black moth. So the individual with the best adaptations for that particular environment are the ones most likely to survive and therefore reproduce. And if they reproduce, they pass on their genes to the next generation. So these survivors pass the beneficial genes to their offspring. Now, if you can keep those 5 stages in mind, it's the same stages that come up over and over again. So here's a question where you're going to see those stages get added in. So explain how natural selection led to the evolution of the peppered moth during the Industrial Revolution. So it's a 4-mark question, and the answer. Number 1. So it's what we've just gone through, exactly the same. So number 1, the tree darkened due to soot during the Industrial Revolution. Number 2, moths showed variation. Some were dark, some were light. The dark moths camouflaged better, while the light moths were eaten. The dark moths survived, reproduced and passed on their colour, so more dark moths evolved, were born due to reproduction. So if we look at this question here, we've got a change in the environment. We've got our variations. Now, we have competition there. We don't have to necessarily mention the competition, but the dark moths were better camouflaged, so less likely to be eaten. That means they survived, they reproduced, and they passed on. Now, it says here colour. It would have been better to have written genes for colour. Are passed on, so the dark moths not evolved, but the number of dark moths increased over time. And there's the mark scheme answer. This is a slightly different question now. Explain how natural selection could have caused an increase in beak length because of the use of bird feeders. So in this question, it's talking about a bird called the great tits, and I think, So you have these kind of feeders that are in gardens, and the food is inside. And they're saying, once more and more people have introduced these bird feeders into the garden, why has it led to an increase in beak length? So what we're saying is, the population of great tits, which is a type of bird, shows variation. So you've got some that have got short beaks, some that have got long beaks, etc. Now what the bird feeder does is it provides a selection pressure, meaning it provides an environment, a scenario that can make one more successful and one less successful. So in this case, which one's going to be the most successful? The birds with the long beaks, because they're more likely to get to the food. That means they're more likely to survive, reproduce, so that's survival of the fittest. They're more likely to pass on their alleles or their genes for long beak to the next generation, to offspring. So over many generations, you end up with more birds with a longer beak length. So you have an increase of those birds in the population. Okay, 4.3. Explain how the emergence of resistant organisms supports Charles Darwin's theory of evolution, including antibiotic resistance in bacteria. So when we're talking about resistance in bacteria, it's the same process, except bacteria reproduce quicker. They are splitting every 20 minutes. So they go through multiple generations every day, whereas for us to go through 2 or 3 generations, it takes 60, 70 years. So why do bacteria make these adaptations so quickly? It's because bacteria make these changes rapidly because they reproduce very quickly and can exchange genetic material. So there's bacteria, it's split, and now it has two identical bacteria. Now if a mutation occurs, that means though from that particular bacteria, you've now got two new strains. So bacteria reproduce at a fast rate. So what is bacterial resistance? So bacterial resistance is when bacteria develop the ability to survive antibiotics that would normally kill them. So antibiotics, as we'll cover in topic 5 in more detail, kill bacteria. And when they were discovered, it was seen as a miracle drug. And what doctors started to do was oversubscribe it. Someone came in with a cold, a cough, anything, even if it wasn't bacterial, there was no checkup to see if it was a bacterial infection or not. They were just given antibiotics. But what that's led to is resistant bacteria. But why is it they become resistant? And the reason they become resistant is because people have started to eat the antibiotics because they've been told to take the antibiotics, so they've taken it. Now, day one, in their body there's lots and lots of bacteria. Lots and lots and lots of them. And it's past the threshold. So if, let's say, them two there, we're okay. But with all these extra bacteria, the person is feeling unwell. So they take an antibiotic. And we've got a few different ones. So when the person takes antibiotic on day one, all the weaker ones get killed off. They die because of the antibiotic. However, then they take it on day two. And on day two, the ones that are a little bit stronger than the previous one, they get killed off. By day three, they take the antibiotic again and a few more get killed off, but they now feel a lot better. The issue now is they've killed off all the weaker ones, they've left the strong ones. So now which one's going to multiply? It's going to be these slightly stronger ones. They're the ones that get passed on. Had they finished the antibiotic course, they would have killed them all off, and then there was nothing left to pass on. So it's over time, the resistance increases and increases. Now, which ones are going to reproduce and pass on their genes? It's going to be the strong one. And eventually we get to a point where we get a bacteria that is resistant. In other words, the antibiotic no longer works. So how can we reduce the rate of development of antibiotic resistant strains? Well, doctors no longer oversubscribe the antibiotics, and even using types of antimicrobials on parasites and bacterial infections for plants, that's been reduced as well. And when a person is given antibiotics, it's important that they finish the course. So if they're giving it for 7 days, it's important they finish the 7 days worth of antibiotics, not stop taking it after 3 days. So why is the development of antibiotic resistant drugs a concern? Well, it's a concern because we've now reached a point where bacteria, there are versions of bacteria that are resistant to the strongest antibiotics. So it costs a lot of money to go through the process of making new medicine. So bacterial resistance is strong evidence of natural selection and evolution. So it is evidence. When antibiotics are used, only resistant bacteria survive and pass on their genes. Understanding this helps scientists improve antibiotic use and develop new treatments and fight resistant infections. Okay, So this is a question. Explain how bacteria have developed resistance to antibiotics. So now, this is going through our 5 stages that we mentioned. Let's see the mark scheme answer. So, by natural selection. So, like I say to anyone aiming for grade 9, always point out the obvious. We have variation in the population. That's caused due to a mutation, but you don't need to mention both. Now, when the antibiotic is taken, only the resistant bacteria survive the treatment. The resistant bacteria survive when people don't finish the course. And the resistant bacteria are the ones that reproduce. That means their offspring inherit, so they pass on their genes to the next generation. So it goes back to what we did in 4.2, the same points. You've got to keep mentioning those points. That's how you get the marks. Okay, so explain how the fossil record can be used to support the theory of evolution. What are fossils? Fossils are preserved remains or traces of ancient organisms such as bones, shells, footprints, or even imprints of leaves. They provide evidence of life from millions of years ago and help scientists study evolution. So there is an example that potentially T-Rex was a chicken. So here's Ardy and Lucy. They were found 4.4 million years ago. And you can see that they've got certain features which are close to human-like and other features which are not close to human-like. Lucy was found 3.2 million years ago in Ethiopia, and they believe it's about 3.2 million years ago. Lucy's foot bones indicate her feet resemble human feet, but she had curved toes. Bones showed she could walk upright like modern humans. Now, Mary and Louis Leakey, they discovered these fossils that are from 1.6 million years ago. Now, what these fossils, evolutionists try to show using these fossils is how they've changed over time. And one of the main things they try to show with human beings, not all of these are Homo sapiens. What they are other types of organisms that evolutionists would say have eventually evolved to become Homo sapiens or humans. And what they show is that the brain gets bigger and bigger, the skull gets bigger and bigger. So the main evolution in human beings is the fact that the brain has grown. So other type of evidence is stone tools also can be used to evidence evolution. Now obviously the wooden part of a stone tool would have decayed, so it's only the stone bit that's left. Now other fossils, so like tools, also provide potential evidence for evolution, as you can see. The age of these is about 10,000 to 2.5 million years ago, and these are all 6,000 years and less. Now, evidence for evolution, we've got fossils, an artifact that we can do an age assessment based on. There's excavation and evidence that's been collected. There's carbon dating. So if they've found any organic material, or they found carbon-14, anything with carbon-14 in the same area as a fossil, they can carbon date the carbon-14, find out how old it is, and then you can make that estimate that if they're in the same place, they must have a similar age. So if you look at this now, there's a few different ways of using this for evidence. So one is, this is a question, this kind of brings everything together. So this question brings everything together for 4.5, and it's a really good question to learn. So if we look at this question in detail, it's essentially saying, explain how the information in the diagram provides evidence for human evolution. So the lower down you go in the earth, the older something is. Meaning C is the oldest, A is the newest. Now C is less sophisticated and A is the most sophisticated. What does that indicate? That the one that made A must have been more intelligent. And what did we learn in 4.4? That the skull has increased in size, as has the brain. So that evidences that it provides potential evidence for that type of evolution. So tools found deeper underground are usually older than tools found close to the surface. Now in this question it says explain how the information in the diagram provides evidence. So because it's a 6-mark question, you need to compare tool A, B and C as part of your explanation. So tool C is deepest in the earth, so that means it's the oldest. Tool B is somewhere in the middle, so it has a medium age, and tool A is the shallowest, so that means it has the lowest age. It's the newest thing there. Radiocarbon dating can be used to estimate the age of organic material near the tools, like wood and bone. Let's look at the mark scheme. So this is more answers, sorry. So older tools are simple and less refined, so that's less refined. The newer tools are better, more refined, they're more sharp, they're more advanced. And that shows human intelligence has evolved over time. So here's the mark scheme. Area 1 is on the age of the tools. So if you think of everything we've just gone through, that's enough to have got 6 marks. Because area 1 shows the age of the tools. Younger rock layers towards the top, older rock layers towards lower down. C is older than B, which is older than A. Tools can be compared with other fossils from the known time period. Rocks can be dated, as in using. Now we said carbon dating, but the correct word is radiometric dating. So don't say carbon dating, use the word radiometric dating. So area 2, quality of tools. A is the most sophisticated. B shows some evidence of being worked on, and C is basic, simple. And what area 3, what does that show us? It shows that tools give us evidence of greater human manipulation, or it shows greater skill when you go from C to B to A. And it also evidences higher intelligence within humans. Okay, the pentadactyl limb is triple only. So we don't need to cover this in combined. This was covered yesterday with the triple students. So thank you to those that stayed on and went through this with me. So let's move on to topic. Let's move on to specification point 4.7. So 4.7 talks about why we've switched from the Linnaeus model to the 3-domain model. This is from a Linnaeus model perspective that usually comes in foundation, but the 3-domain model is something that has become more and more common over the last 7-8 years in the combined paper and the higher paper and the triple paper. So classification is the grouping of organisms based on their characteristics and the evolutionary relationships. So it's an attempt to show a connection between different animals. So classification sorts animals in organisms into groups based on similarities and differences. So for example, mammals feed their live young milk. Reptiles are so human beings have human beings have hair, fur, things like this. Birds lay eggs. Human beings, mammals give birth to live young. Reptiles give birth via eggs as well. That kind of thing. So these are all It was an attempt to classify animals, diversity of life into different categories such as kingdoms, phyla, class, order, families, genera, and species. So this was developed by Carl Linnaeus. So the thought process, everything comes from kingdom Animalia, then it splits into chordata, the phylum, then into class, order, family, genus, and species. So this word here, genera, it should have been written as genus. Genera is not incorrect, but the better word is, at the moment, based on GCSE level, it would be genus. So what are the 5 different kingdoms? Animals, plants, fungi, prokaryotes, and protists. When we name an animal, we put the genus name with a capital letter first, followed by the species name. So we're saying Homo sapiens. Homo is the genus with a capital letter, and sapiens is the species name. So for example, cat, Felis is the genus it comes from. Domesticus is its species. A dog, Canis is the genus it comes from. Familiaris is its species. So why did Linnaeus' model change? It's because of electron microscopes. So with electron microscopes, we were able to look at everything with a lot more detail, and also genetic research so we were able to look at genes in a lot more detail, and that led to the 3-domain system. So 3 domains are bacteria, archaea, and eukarya. So eukaryotes or eukarya are organisms that have a nucleus within their cells, like animals, plants, fungi, and protists. Prokaryotes are simple single-celled organisms with no nucleus, like bacteria. And archaea are single-celled organisms that also lack a nucleus, but they're genetically different from bacteria and often live in extreme environments. So Carl Woese found that everything came to these 3 categories. So you need to know these 3 categories, and like I've mentioned before, this is the initial phase where we're memorizing, where we're understanding everything, where we're activating our brains. So this is the activate phase. Once this finishes, my advice is to start working on your memorization and your past paper practice. And your past paper practice is the part that will help you understand what needs to be memorized. So evolutionary trees are diagrams that show how different species are related and how they've evolved from a common ancestor. So we can look at living organisms and look at extinct organisms and start to make comparisons and start to look at how they may have been connected. So this is the question. Explain the differences between the Linnaean system and the 3-domain system of classification. This is a really good question to memorize the answer to. So the Linnaean system breaks things into kingdom, phylum, class, order, family, genus, and species, whereas the 3-domain system classifies everything as bacteria, archaea, and eukarya. And when answering this question, you would go into detail as to what bacteria are, so what eukarya are, what archaea are, and what prokaryotes are, prokarya are. So why was the 3-domain classification system actually proposed? Plants are eukarya because they have nucleus within their cells. Plants are not archaea as they're not primitive bacteria that live in extreme conditions. plants. So this is a multiple choice question. So as a multiple choice question, the answers were either eukarya, prokaryotic, a Monera, which is a kingdom, and also Protista. So plants are only eukarya. Okay, with anything that we're going through at the moment, up to 4.11, so from 1.1 to 4.11, we've got a separate video that's linked to Edexcel for every single specification point. So there's around 60 videos, each lasting between 2 to 5 minutes, some a little bit longer, some a bit shorter, that go through one specification point at a time. So if I go cover anything and you feel like there's not enough detail, all you've got to do is watch one of our AMAL videos. So selective breeding, again, it's a very common question, and if you know the steps and the stages, it's a nice straightforward topic. So selective breeding is a process where humans choose specific plants or animals with desirable traits and breed them together to produce offspring that also show those traits. So these are the steps to remember. Number 1, choose the parents with the desired traits. And what do we do with the parents with the desired traits? We breed them. Breed those selected individuals. Okay, now the step 3 has been missed from this. Step 2, sorry. So choose the parents with the desired traits and breed them. From their offspring, find the ones with the desired traits and breed them. So look at the answer. Number 1, So we've got number 1, choose the parent with the desired traits and then breed them. Step 2, from their offspring, pick the 2 with the desired traits or the best characteristics and breed them. And then step 3, repeat over several generations until all offspring have the desired trait. If you mention those 3 things in a selective breeding question, you've got 3 out of 3 marks. It's always the same answer. You just have to adapt it for the question. So what are the positive impacts of selective breeding on plants? So it's improved food security. So farmers can breed plants that produce more food per plant. It's improved quality. Which means it leads to better taste, size, durability that benefits consumers. It's also led to disease resistance. So some plants have been bred selectively. So if they found a plant that has, for example, drought resistance, they've selectively bred until all the plants growing have all got drought resistance. Now also you can combine traits as well. So for example, this wheat might have had a positive trait on this rice, and when they've been crossbred, they've made a new plant that has a higher yield and disease resistance. Farmers can breed plants that produce more food per plant, helping to feed larger populations. But there are issues, and there's some negatives here. So the positives were on the previous slide. The negatives now, it leads to a reduced genetic diversity. So a narrow gene pool increases disease and environmental risks. There's ethical concerns because selective breeding can lead to health issues in animals. For example, this dog which is called a pug. And people usually breed these animals for competitions. But they've crossbreed them so much that these dogs have got issues with the muscles in the legs. They've got issues with their breathing as well. That's because they've reduced the genetic diversity, the gene pool so much that now when they're crossbreeding them, they're more likely to get genetic disorders and unintended consequences. It can lead to an overemphasis on one phenotype, and that might neglect other important traits. Now, some traits might not be important straight away. They could be important when a certain viral infection or bacterial infection pops up. So that's what we've just covered, the negative impacts. I'll just leave the summary there so it can be read if you're rewatching the video. So here's a really good question that brings everything together. Describe how race dogs alive today have been selectively bred to run faster. So remember the 3 steps we mentioned before. Step 1, identify the individuals with the desirable trait and breed them. Now in this case, it's finding the male and female dog that can run really fast. And then and breeding them. Then from the offspring, finding the male and female dog that are the fastest and breeding them. And then repeating that step over multiple generations until the majority of the offspring or all of the offspring are now super fast. So let's look at the mark scheme and how what we just did compares. So there we go. Breed animals that can run fast. From the offspring, selection of offspring that can also run fast, and repeat the process over many generations. That's the answer over and over again. Alongside that, you need to memorize the positive impact of it and the negative impact of it, because you can get questions linked to that too. So let's move on to not 4.9, because 4.9 is triple award only. So we're going to skip past this and move straight on to genetic engineering. And before we go through the process, we're going to go through how genetic engineering benefits us. So it can benefit us in medicine because we can genetically engineer, for example, bacteria to make insulin. You can genetically engineer a plant so it's cold resistant or resistant to a particular type of insect. And then it also helps with biotechnology. But technically they're both examples of biotechnology. So what is genetic engineering? Genetic engineering is the process of changing an organism's DNA by adding or removing or altering genes. Now the one we're going to be looking at is the adding of genes. So for example, if we add the part of our genes that make insulin and we add it to a bacterial plasmid. So plasmid is part of the DNA of a bacterial cell. Now, what would the bacterial cell do? Because we've added our insulin gene, when the bacterial cell will now make insulin. So we add, remove, alter genes to give it new characteristics. Scientists take a useful gene from one organism and they insert it into the DNA of another organism. So we're looking more at the inserting of a gene. So why is it beneficial? Because we can enhance crop yields. So for example, if there's a plant that always dies because it's too cold, or it's constantly getting a certain viral infection or bacterial infection, or is eaten by a certain insect, then you can genetically engineer, you can add genes that help counteract those issues. Now if a farmer has counteract those issues, they're going to get more of that particular crop. So that means increasing crop yield. You can develop disease-resistant crops, which I've already mentioned. That means you also increase the crop yields. You can improve industrial processes, create new medical treatments, Okay, 4.11. Now 4.11, these four key words are very, very important. Restriction enzyme, that's the enzyme that cuts the DNA. Ligase, that's the enzyme that joins two different parts of DNA together. Sticky ends, when you cut the DNA, you leave a small section so when you cut the human DNA or wherever the desired gene is, and when you cut the bacterial DNA, sticky ends are the parts that are joined together using the ligase. And a vector is, in this case, for example, bacteria. Something that then the new gene is put inside. So what we want to do here is describe the stages of genetic engineering in detail. So first thing, purpose. What's the purpose of restriction enzyme, ligase, sticky ends, and vectors? We've already mentioned this. The restriction enzyme and ligase, they're like molecular scissors. They cut the DNA at specific sequences, creating small fragments, leaving sticky ends. Ligase is not like molecular scissors, sorry. It's an enzyme, but don't forget, enzymes can either break things apart or they can attach things. So ligase is an enzyme that attaches things. So it joins the cut DNA fragments together, like that. Ooh, seems to have missed the gap there. So it should have stopped there. And the ligase would help join that together. Now, vectors are carriers that deliver the desired gene into the target organism. And sticky ends, when restriction enzymes cut the DNA, they leave exposed bases at the ends. Now the exposed bases this is from the bacteria, and the exposed bases from the gene that's come from, for example, a human being or from an animal. They have exposed bases that then join together, and they join together using ligase. So what actually happens? Let's go through it one step at a time. Number one, the desired gene is identified. Number two, it's cut using the restriction enzyme. Number three, it's inserting the genes inserted into a vector. Oops, sorry. So what this means is we've got the DNA, and we locate the desired gene. Okay, so it could be here, and we cut it. Now when we cut it, now this is DNA, so it would have bases in between. I'm just going to draw the bases as straight lines rather than showing the two bases that actually join to each other. Now once it's been cut, so we've got our main part, so that could be, for example, for insulin, and then we've got these extended parts, which are the sticky ends. Now when we go to the bacterial cell, a plasmid is a loop of DNA. And what we do is, using the restriction enzyme, a small section is cut out. So for example, this section is cut out, but when it's cut out, sticky ends are left. Then this gene is put into that gap there and sealed together using ligase. And then using a vector, this is put into a bacterial cell. And now this bacterial cell will start to make insulin, which can then just be purified from the bacteria. So in summary, genetic engineering uses restriction enzymes, ligase, sticky ends, and vectors to modify DNA. And it has new traits with important uses in agriculture, medicine, and industry. So these 4 keywords need to be memorized and remembered, and you've got to understand how to use them in as part of genetic engineering. Like I said, we have just put the genetic engineering video up, 4.11, so you can watch this. The only benefit of watching the animation is that you can see all the parts moving around and how they all combine together. So it works really well alongside my explanation. So explain how bacteria can be genetically engineered to produce human insulin. So this is a question. So in stage 1, identify the desired gene. Scientists identify the human gene responsible for making insulin. The gene is cut out of the human DNA using a restriction enzyme. So this is the answer that I would memorize. I've explained it, but this is the memorization page. Stage 2, cut the DNA with a restriction enzyme. So the restriction enzyme cuts the DNA at specific sites, creating sticky ends that can bind to complementary sequences, allowing the insulin gene to be inserted into another DNA. So how do we do that? We insert the gene into a vector. The vector, in this case, is a plasmid, a circular loop of DNA. It carries the insulin gene into the bacterial cell. That plasmid is cut with the same restriction enzyme, also creating sticky ends, and they match the insulin gene sticky ends, and that allows the genes to be inserted. And then we join the 2 bits of DNA. So we've now got the DNA from the human being to make insulin. We've got the gene from the bacteria to make that will act as a vector. We've cut a section out of the bacteria, just big enough for the DNA from the human being to be added in. And then we join the DNAs using ligase. DNA ligase joins the sticky ends of the insulin gene and plasmid, creating a stable recombinant DNA. And that recombinant DNA, this step ensures the insulin gene is stably incorporated into the plasmid. Now, the recombinant plasmid is placed into a bacterial cell, which takes up the new DNA. In bacteria, this allows them to produce the human insulin, which can then be collected and used to treat diabetes. Now, this was really important because prior to this, we were treating diabetes using insulin from cows and pigs. So once the animals were slaughtered, their pancreas was removed, and from the pancreas they would take out insulin. So here's the mark scheme, and this is a 6-mark scheme that summarizes all of 4.11. It's a fantastic question to memorize and learn. So the gene that codes for human insulin is identified in the human DNA. So lots of obvious things that we wouldn't normally write are part of this mark scheme. This is removed using a restriction enzyme. The plasmid of the bacterial cell is also removed using a lysozyme. Don't worry about that part. The plasmid is cut open using the same restriction enzyme, leaving complementary sticky ends. And the human gene is inserted into the bacterial plasmid. The human gene is inserted into the bacterial plasmid using the enzyme ligase. Then after that, the plasmid is returned to the bacterial cell, and the bacterial cell multiplies, making lots and lots of insulin. Okay, now 4.12 and 4.13 are triple only, so we don't need to know about these. And 4.12 and 13 will be on AMALearning soon as well, for those that want to make use of it. Okay, final topic of topic 4 biology. Evaluate the benefits and risks of genetic engineering, selective breeding in modern agriculture and medicine, including practical and ethical implications. So benefits. increased crop yield, reduced pesticide use, it's cost effective for farmers. So what we mean by this is, if you genetically engineer something so that it doesn't have, so it has, for example, it has a resistance to cold, you're going to get more of a crop yield. If you look at it from a selective breeding perspective, you can selectively breed a crop that provides more food, so that increases the crop yield. Having said that, this is specific genetic engineering, but some of them can be done for both. You can genetically engineer something so it doesn't require pesticide, it produces its own toxins. That means it's more cost effective for farmers. So if you have a plant that's genetically modified, that has reduced, that doesn't need as much pesticide, that's a saving for the farmer. And if it makes it makes more of the crop grows bigger and you have a bigger yield that comes from it, that means the farmer makes more money. Then you've got examples of improved food quality, for example, golden rice. So golden rice has been genetically modified. I think it's vitamin C. So it's got more vitamin C than your average rice. And also we can use genetic engineering to make insulin for diabetes. But the risks, if we genetically engineer, if a plant is genetically engineered and is genetically engineered so that it releases a pest or a toxin that kills off a certain insect, it could also end up possibly harming another species. The genes could mix with a wild plant, and then give it a resistance, an additional resistance, meaning the weeds would become super weeds and spread. We don't actually know the long-term effects, so it could impact ecosystems. Now, there are ethical concerns. Some people object to genetically modified foods. That's because we're mixing. So, for example, there was a fish. So, fish can generally survive in cold climates. So, they took the gene from a fish that helped it survive in cold climates and they put it into a potato. So, that potato will grow through winter. So, that's the kind of thing people would have an ethical issue with. And then genetically modified crops could reduce biodiversity. Again, because certain insects that feed off those crops would die out, and that could then have a knock-on effect on other insects. Also, it could kill non-target species. So what are the benefits and risks from selective breeding? So benefits, we get higher yields and better quality. Animals can be bred to be safer, so you could end up with a hornless cattle, or if you had a wild animal, you could keep finding from the offspring, you could keep looking for the softest one or the calmest one, keep breeding them until you can have them as pets, or that they're safer. Essentially, that's what happened with dogs. Farmers can choose plants and animals that can live well in tough places. For example, they could find a plant that doesn't need much water. I think the goats that you have in hotter countries can survive the drought, whereas the ones that you have, goats that have in colder countries, produce fattier meat. So they've crossbred those and then selectively bred those so that you get a high amount of meat and they're able to survive in harsher climates as well. Now, the risks, it reduces genetic variation because you're reducing the gene pool every single time, because you are reproducing offspring that are genetically very, very similar. And breeding for one trait can harm other traits. So it could have an impact on health, fertility, which is the case with some animals like the pug. It can cause inherited problems. For example, hip dysplasia in dogs. So that's one of the problems that occurs in pugs now. And traditional breeds and crop varieties might be lost in the pursuit of getting this perfect animal or perfect crop. So here's a past paper question, and so what we're going to finish on now, and then take a 5-minute break. Describe one benefit and one risk of selectively breeding chickens. So the benefit is that it produces more food. There's fewer chickens needed for the same amount of meat. But the risk is there'll be less variation. There will be less variation. You could lose useful genes, lose traits which may be useful in the future. It could lead to health issues related to larger bodies. So this actual question, when it was first came out in an exam paper, it showed like a smaller chicken. I should never even bother trying to draw a chicken. Then it showed I'll just draw a circle. Then it showed a bigger chicken and an even bigger chicken. The biggest chicken was monstrous. It looked like a monstrosity. It was massive, big legs, a lot of meat. You can see those kind of chickens. Their mass would cause them issues in the legs. In fact, that does actually happen now. Okay, thank you everyone. We're going to take a 5-minute break, and we'll restart at 2:32. So thank you everyone. See you guys in 5 minutes. Welcome back everyone. Let's complete topic 3, chemistry. Okay, sorry, excuse me for just 1 second. Just need to check one thing. Okay, apologies, let's make a start. So, what are acids and alkalis? So an acid is something that releases H plus ions. Whereas an alkali is something that releases OH minus ions. So if we look through, let's look at some examples of an acid. HCl, H2SO4, HNO3. So we've got hydrochloric acid, sulfuric acid, nitric acid. They all contain H. So when they dissociate and break apart, so when you add HCl to water, it breaks apart into H plus and Cl minus. H2SO4 will break apart into H plus, 2 H pluses and SO4, 2 minus. HNO3 will become H plus and NO3 minus. Is the H plus, that's the acid part. Now with alkali, for example, sodium hydroxide, when that breaks apart, you end up with Na plus and OH minus. I'm going to show you something that's going to come up multiple times. But now if we react an acid with an alkali, what happens? The H plus from the acid reacts with the OH minus, so the hydrogen ion, positive hydrogen ion, reacts with the hydroxide ion to make, we've got 2 hydrogens there, 1 oxygen, H2O, makes water. So that's why we call it neutralization when you add an acid to an alkali, because it makes something neutral like water, and water is neutral. So how can we tell the pH? When we look at a scale like this, we know if it's between 0, 1, 2, 3, 4, 5, 6, it's an acid, and 7 is neutral, and 8, 9, 10, 11, 12, 13, 14 are alkali. And there's a few different types of indicators that help us understand that. So if you use universal indicator, if it's red, that indicates 0. So red, orange, yellow indicate acidic, neutral is green, and alkaline are usually blue to purple. But in Edexcel, you need to know a few different indicators, not just universal indicator. So this is litmus. If you've got red litmus, if you add it to, if red litmus turns blue, then it means it's an alkali. If blue litmus turns red, it means it's an acid. If nothing happens, so you put blue litmus in and you put red litmus in and there's no color change, that means it's neutral. Now here we've got an example of methyl orange, which initially in an acid is like an orangey red color. Now in a neutral or alkaline solution, it stays yellow. Now you do need to know the color changes. It does come up as a 1 or 2 mark question, so it's not necessarily a priority thing to memorize, but you should know these color changes. Phenolphthalein Now the colors here are completely wrong, so let me just adjust them slightly. So when it's in acid, it stays colorless, but in alkali it turns a pinkish color. And if you ever do put phenolphthalein, if you've ever done the experiment at school, you'll notice it goes from colorless to pink all of a sudden at the point where it goes from acid to alkali. And when it is colorless initially, I'm not sure if that shows colorless or not, but yeah, that's what happens. So phenolphthalein goes from colorless when it's an acid or neutral, and as soon as it becomes an alkali, it turns pink. So this is something that for those who are doing triple award, you need to understand when you're looking at titrations. Now, how can the pH of a solution be determined? Use a pH probe, and that gives more precise measurements because it displays the exact pH number on the screen. So compared to what we've just shown, what we've just gone through, it's much more accurate. Okay, hydrogen ions. Now, the more hydrogen ions something has, change my color. So the more hydrogen ions something has, the stronger it is as an acid. The more hydrogen, so the more H plus ions, the lower the pH, the more acidic it is. The fewer the H plus ions, the less acidic it is. So it's closer to 6 or 7. It becomes more alkaline as it starts increasing in the amount of OH minus. Now, if we're at pH 6, whatever the concentration of pH 6 is, pH 5 has 10 times more concentration, 10 times more H plus ions. Whatever your concentration of H plus for pH 5 is, pH 4 has times 10 times more pH. Yes, this is combined and it's triple. All of topic 3 is combined. So when you go from pH 5 to pH 4, again, we multiply by 10. So the number of H plus ions increased by 10. So it says here, each pH unit change equals 10 times difference in H plus ions. So what that means is, if we've got pH 5 and we want it to become pH 4, if in pH 5 we have a concentration of 0.05 grams per decimeter cubed, that's our concentration of H plus ions. If I want to change it to pH 4, I've got to make sure I've got 10 times more H plus. So if you times that 0.05 by 10, and we get 0.5 grams per decimeter cubed. Now, if I want it to become pH 3, what do I do to the concentration of H plus ions? I multiply by 10. If I want it to become pH 2, what do I do to the concentration of H plus ions? I multiply by 10. I multiply by 10. So you can see here from pH 5 to pH 4 to pH 3 to pH 2, what have I done to the concentration of H plus? I've multiplied it by 10. Okay, core practical. What happens to pH when an acid and alkali are added together? So then we've got a dilute hydrochloric acid. Now what this actually means is, we add dilute hydrochloric acid to a beaker, we use a clean glass rod to take a drop of the solution, and we place the drop of universal indicator. We place the drop on the universal indicator paper on a white tile. Wait 30 seconds and compare the color to a pH chart and record the pH. Then you stir and you would repeat. Now what we're going to do is we take an alkali. In this case, we're taking a base. A base is the solid version. When it dissolves into a liquid, then we call it an alkali. So you add that in. Same process, you stir, take one drop, put it on universal indicator paper, compare to the pH chart. And you keep doing this till we get to the point. We get to a pH of 7, and then eventually we get to a pH of it that it becomes an alkali. And what this is actually showing is this. Initially, what's this full of? It's full of H+. Now I'm going to show the H+ as red dots. So let's erase what I've already put there and let's use red dots instead. So now I'm filling it with red dots. And what do my red dots represent? H+. So the red dots represent H+, lots and lots of H+. Now as soon as I add the alkali, what does the alkali do? H+ plus OH minus becomes H2O. It just converts it to water. So I put my first spatula of alkali, and it starts reacting with the H+ to make water. And what's happening to my pH as I'm doing this is becoming bigger and bigger. So it's going from 1, pH 2, pH 3, pH 4, pH 5, pH 6, until we get to pH 7. Now if we add alkali into it, now what's happening is we're adding OH minus to it. So every time we add OH minus, what happens? Now that it becomes more and more alkaline. So dilute and concentrated. So when we say dilute, we mean dilute solution is where you have a large amount of solute dissolved in a large amount of solvent. So it just means you've added a lot of water to it. So when you dilute an acid, a dilute acid is where you've got less acid, more water. And when we're talking about concentrated, then what we mean by that is where you've got more of the water. No, sorry, let me start again because I've said one thing wrong now. So when we say dilute, we mean more water, less acid or alkali. When we say concentrated, we mean more of the acid or alkali, less water, which is different to strong and weak. When we say strong, a strong acid is one that completely dissociates, completely breaks apart, ionizes in water. So let's have a look at this. If you've got HCl, when you put it in water or into a solvent, it will break apart into H+. Now that is a one-way reaction. Once that H+ has broken into H+, that HCl has become H+ and Cl-, H+ and Cl- aren't going to start reacting with each other to make more HCl. It's not a reversible reaction, meaning whatever HCl is inside here, as soon as it gets added, it breaks apart into H+ and Cl-. All the H+ is available to react. Now when we're talking about a weak acid only partially breaks apart. And that's because look at this formula with ethanoic acid, it's a weak acid. Look at the formula for ethanoic acid, it's a reversible reaction. Meaning CH3 COOH, which is the ethanoic acid, it breaks apart to make H+ and CH3COO. That should have a minus there. But these two things, they combine to make more ethanoic acid, CH3COOH. At the same time, that CH3COOH, the ethanoic acid, is making more H plus and breaking apart to make more CH3COO minus. So that's a ethanoate ion. And then the H plus in this negative ion here, CH3COO minus, they're reacting with each other to make more ethanoic acid. So you never end up with all the H plus being released. It's only partially released. And then hence we call it a weak acid. So where the pH of these will be 0, 1 and 2, our weaker acids will have a pH of 3 sometimes, but more 4, 5 and 6. Okay, so what's a base? A base is a substance that reacts with an acid to make a salt and water only. So that means a base is a substance that reacts with an acid to make a salt and water only. So in other words, neutralization. So metal oxides, an example is copper oxide, hydroxide, sodium hydroxide, metal carbonates, calcium carbonate. So alkali is a type of base that dissolves in water and it releases hydroxide ions. It releases OH minus. So examples, sodium hydroxide, NaOH, which is found in drain cleaners, potassium hydroxide, KOH, is used in fertilizers. So reactions of acids, these are usually redox reactions. What that means is reduction and oxidation happens at the same time. So when an acid and a metal react together, it makes salt and hydrogen. So what we need to know, now from a redox perspective beyond this, we will go into a lot more detail that comes in topic 4. It comes in topic 3 as well with electrolysis, and then it's explaining a lot more detail in topic 4. So tomorrow we'll be spending a bit of time on redox. I'm hoping I can finish this. I'm nearly on slide 20. I'm about a third of the way through. Exactly a third of the way through, sorry. And we've got 40 minutes, so should hopefully be able to finish this. Although electrolysis does need a bit more explanation, so I will give time for electrolysis. Just to recap for tomorrow, the main priority tomorrow will be biology topic 5, chemistry topic 4, and physics topic 5 and 6. Hopefully we'll finish topic 5 off today for physics. So it'll be topic 6 that will be the priority. That does mean there's some elements of Edexcel triple that I've not been able to complete, but I will do my best over the next week by the end of the holidays to make that video. It means I've missed out a little bit of biology topic 5. We will have finished physics in completion because I'm doing astronomy today. And I will have missed out. So the main thing I'm missing out is chemistry topic 5. The rest is just a few small topics here and there. So I have still covered 95%, the majority of it. I've done my best. With the Edexcel, I've been doing 1 hour after each session. So it was supposed to be an 18-hour course, but for Edexcel, by the time I finish it, I'll have spent 22 hours. Around 22 hours. So it probably needs a couple more hours beyond that, but I'm not going to be able to add in those extra hours. So I'll do my best to fit it in. So acid and metal makes salt and hydrogen. Now when a metal oxide or hydroxide reacts with an acid, it makes salt and water. So you've got to remember this. Metal carbonates make carbon dioxide and they also make salt plus water, but it's a carbonate, so it also makes carbon dioxide. So acids and metal make salt plus hydrogen. So here's a few examples. Magnesium oxide and hydrochloric acid. Now, whenever you have hydrochloric acid, it always makes a chloride. So we've got magnesium chloride. So we've made, that's our salt. Now, what do oxides make? They make salt plus water. So it's going to be magnesium chloride plus H2O. Magnesium carbonate and nitric acid. Nitric acid always makes a nitrate. So when magnesium reacts with nitric acid, it's going to make magnesium nitrate plus carbon dioxide plus water because it's a carbonate. Sodium hydroxide and sulfuric acid, sodium sulfate. And it's a hydroxide, so that means we're also going to make water. Then we've got copper oxide and hydrochloric acid, so copper chloride, that's the name of our salt, and it's an oxide, so just like the oxide before, we're just making an additional H2O. Next one is lithium carbonate, so and it's reacting with hydrochloric acid, so it's going to make lithium chloride, and because it's a carbonate, it means we automatically make water and carbon dioxide. Doesn't matter which way you write that. I know I put carbon dioxide first on the previous one. Then we've got sodium hydroxide and nitric acid. Nitric acid makes a nitrate, so that's sodium nitrate. And it's a hydroxide, so that makes plus H2O plus water. Okay, now if hydrogen has been produced, so when a metal, so one thing to note here, when a metal oxide and hydroxide reacts with an acid, it makes a salt and water. But when it's a metal carbonate, it makes salt, water and carbon dioxide. If it's just acid and metal, it makes a salt and hydrogen. So you need to know the test for hydrogen. And the test for hydrogen is squeaky pop test. So you collect the gas in the tube. After a reaction that produces hydrogen, you hold a burning splint near the mouth of the tube, so around here. Observe the reaction. If the hydrogen is present, it will burn quickly with a squeaky pop sound. Now that's because the hydrogen has been provided with enough energy to react with oxygen in the air, and that makes H2O. And in making H2O, it releases that squeaky pop sound. So how can we test for carbon dioxide? Because when we have carbonates, it makes carbon dioxide. So what you do is you get some lime water and you bubble through, or you get carbon dioxide and you can either shake it through, or a better way is to bubble it through the lime water, and the lime water turns to a cloudy colour. So these are 2 tests out of 4 tests. You need to know all 4 chemical tests, which will come as we go through the curriculum. So what is a neutralization reaction? It happens when you react an acid with an alkali, and it always usually makes a salt, and it makes salt plus water. So here we've got hydrochloric acid and sodium hydroxide making sodium chloride and water. Now if we look at this, let's break it down. We've got HCl, we've got NaOH. Now what does it make? It makes NaCl, so NaCl, that's our salt, and the H and the OH, what do they make? H2O. H2O has a pH of 7 and salt has a pH of 7. So therefore we've neutralized them. We have now got a neutral pH. Now how can we prepare salts? I'll explain, I think better if I explain this in my own words. We use this apparatus here. So the first thing we do, this liquid here is an acid. So let's say we're trying to make copper sulfate, for example. If we're trying to make copper sulfate, so let's just write here CuSO4. what two things can we add to make copper sulfate? So we can use copper oxide, that's a metal oxide. To get the sulfate part, we can use H2SO4, the acid. So now this is full of acid, this beaker. This beaker is full of acid. So now what we're going to do is using, we're going to now start adding copper oxide. One spatula full at a time. And then mixing it with this stirrer. We're going to mix it to make sure it all reacts. When are we going to stop? When at the bottom of that test tube we can see solid. We can see this solid. Now we can see it now. What that solid represents is an excess of copper oxide. It means we put so much of the solid in, all the acid is completely reacted. There's no acid left. So what we've actually produced here, if you have copper oxide and H2SO4, inside here now what we've got is copper sulfate plus H2O. And this solid is copper oxide. And there's now nothing left to react with it because we've reacted out all the acid. But we need to get rid of that copper oxide because what we're trying to do is make a copper sulfate crystal. We're trying to make a pure dry crystal. So now we take this whole thing and we put it through the filter. That means the copper oxide will be left behind and the drops of water that go through are actually copper sulfate and water. Now we transfer that to our evaporating dish and we start heating it. Now it can be done in the boiling tube, so it can be done in a water bath, or it can be done using a Bunsen burner. But the main thing is when you're left with just a small amount of liquid at the end and it's saturated with the salt, that's when you stop. You leave it on a windowsill and you let the water naturally evaporate. The longer it takes, the bigger your crystals will be. The crystals start to form, and then when you remove the crystals, you dab them dry with a tissue and you have got pure dry salt crystals. So that final process was crystallization. And this is just essentially saying exactly what I said. The only thing here is the question says describe a method to make pure dry crystals of magnesium sulfate from a metal oxide and a dilute acid. Now everyone knows the process. What I just went through is the process. Where people lose marks is here. What do we react with each other? How can we make magnesium sulfate? So how do we make magnesium sulfate? How do we make MgSO4? Number one, we use magnesium oxide. And where do we get SO4 from? H2SO4, sulfuric acid. Okay, so this is just saying exactly the same thing. Okay, 3.16, preparing salts. How can we prepare a pure salt when the reactants are both soluble? So you can carry out a titration. So this is something different now. This is titrations, this is not preparing of salts. So, although it links to preparing of salts. So you carry out a titration to find the exact volume of acid and alkali needed for neutral solution. Mix the acid and alkali in the correct amounts with our indicator to form a salt and water. And then after that, you do exactly the same thing. It's just crystallization. You heat it up, but not so much that it becomes completely dry. You let the water slowly evaporate on a windowsill. How do you use this equipment to make pure dry salt? Mix it. You keep adding an excess amount of insoluble base like copper oxide. Keep mixing it, and you put it through a filter. And then you put it into an evaporating dish and heat it up, and then you leave it on a windowsill to slowly cool and evaporate. Titrations. What can titrations be used for? Now titration is a triple topic. So I will go through it because this is the only, let me just check. Yeah, so I'm going to go through it because it's part of it. So it's only going to be an inconvenience for combined students for a few minutes. So what can titration be used for? The concentration of acids and alkalis can be analysed using titrations. During the reaction, hydrogen ions from the acid react with hydroxide ions from the alkali to make water. So we know if you've got an acid in here and an alkali in here, as you keep putting them in, it will neutralize. So an indicator is used to determine the point of neutralization. So something like phenolphthalein that is colorless in an acid. So let's assume this bottom half is an acid. If you put a few drops of phenolphthalein, then initially it will be colorless. So if we go back to the color I had before, it would be colorless. Now, as you start to add the alkali in and mix it, eventually what will happen is the solution so what will start to happen is you add the alkali, one drop at a time. You keep adding the alkali, keep adding the alkali, keep adding the alkali, and as you're adding the alkali, it's neutralizing the acid until it reaches the point where it just turns to an alkali. At that point, the phenolphthalein turns pink. So you start to see some pink. Then you add a little bit more until the whole thing turns pink. Now it happens all of a sudden. It literally takes one drop between it being an acid to alkali. That shows you the point of neutralization, the point where all the acids just been neutralized. So titrations can be used for, as with all quantitative analysis techniques, titrations follow a standard procedure. This ensures the data is collected safely using appropriate precision. This is what I've already just explained. Use a pipette to measure a known volume of alkali into a conical flask. Add a few drops of indicator like phenolphthalein or methyl orange to the alkali. Except this is the other way around. Fill a burette with acid and record the starting volume. Slowly add the acid from the burette into the alkali, swirling the flask. You stop adding the acid when the indicator changes color. This is the neutralization point. Then we record the final burette reading and calculate the volume of acid used. You repeat the experiment till you get consistent or concordant results. Concordant means when the results are within 0.1 of each other. So if you've got 29.2, then after that you've got 29.5, they are not concordant. Concordant means, now if I got 29.1, that's concordant. 29.15 is concordant with these two. So now I've got my three results. That would not be concordant. So you have to be really accurate with this. How can we ensure the results are precise, accurate, and repeatable? So precision, we use a pipette to measure the alkali accurately, and we read the burette volume at eye level to avoid parallax error. Now with accuracy, we add acid drop by drop near the end point to avoid overshooting. So initially you can put 1 centimeter cubed at a time, but when you can see it's starting to change color, just a small amount, at that point you only put 1 drop at a time. So you reach the end point and then you do 1 drop at a time. Use a white tile under the conical flask so you can see the color change clearly, and you choose the correct indicator. So phenolphthalein is for strong acids and strong bases. Methyl orange is better for strong acids and weak bases. And then repeatability. Repeat the titration at least 3 times until you get concordant results. That means within 0.1 centimeters cubed. Now this should be a little a there. It's 0.1 centimeters cubed. You use the same person to take readings, reduce human error, and you calculate the average of the concordant titers for more reliable results. Okay, so we're back to combined. So what is solubility? Solubility is a measure of the maximum mass that will dissolve in a given volume of solvent at a particular temperature. Substances that are very soluble have high solubilities and will dissolve readily. Now, this is something that you need to have learnt, because if it comes in an exam question, you've got to know what's going to happen. So, when you react to soluble substances, usually what you end up with is one precipitate, which is a solid that's insoluble, and one new liquid, one new solution. Now, if you've got potassium, sodium, and ammonium salts, for example, potassium chloride, anything with potassium, sodium, and ammonium within it, then they are all soluble. Anything with potassium, sodium, and ammonium, soluble. Now, anything that's a nitrate, like lead nitrate, potassium nitrate, etc., they are soluble. Common chlorides, so something chloride like sodium chloride, potassium chloride, ammonium chloride, beryllium chloride, all these are soluble. They'll dissolve in water. Now, silver and lead chloride are insoluble. And common sulfates, anything with sulfate at the end, is soluble. But lead, barium, and calcium sulfate are the insoluble ones. So look here, common chlorides are soluble, but silver and lead chloride are insoluble. So you've got the ones that go against the rule. Common carbonates and hydroxides are insoluble, except sodium, potassium, ammonium carbonate, sodium, potassium, ammonium hydroxides are soluble. So let's look at an example. So this makes precipitates. So a precipitate is an insoluble solid that forms when two solutions are mixed in a chemical reaction. There's an example. The yellow part is the precipitate. The reaction that produces the precipitate is called a precipitation reaction. So we can use this table to help us predict what's going to happen. So let's have a look. We've got calcium nitrate and sodium carbonate. Let's go back. Calcium nitrate, so that's here. Calcium nitrate. So we know that's soluble. And what's it reacting with? Sodium carbonate. And common carbonate, sodium carbonate, is soluble as well. So we're going to react it with sodium carbonate. Now if we react those things together, let me just correct this. What we're going to end up with is sodium nitrate, because there's a displacement reaction when they react. So when they react, we have displacement reaction. So we end up with sodium sodium nitrate, so NaNO3, because this sodium here has reacted with the nitrate here. Let's cancel them out. And CaCO3, calcium carbonate. Now if we go through this table, sodium nitrate, nitrates are all soluble, so this will dissolve and make a solution. But calcium carbonate, it says here, common carbonates and hydroxides are insoluble. So this would be our precipitate. That would be the thing that's solid inside there when you react them. That's our insoluble salt. So look, calcium nitrate plus sodium carbonate make calcium carbonate, which is insoluble, and sodium nitrate. So calcium carbonate forms a solid precipitate because it's insoluble in water. So now that's why it's important to memorize this table. So when a question like that comes up, it's a nice, easy question. The easiest way to do it, however, is just memorize what's insoluble. If you know what's insoluble, then you know everything else is soluble. So just learn the insoluble stuff. That's only 3 lines. So if you learn the insoluble, everything else is soluble. So we don't need to worry about anything else. Okay, electrolysis. So what is electrolysis? When an ionic compound is melted or dissolved in water, the ions are free to move. So when an ionic compound is melted or dissolved in water, the ions are free to move. So think back to ionic compound. What did we learn about ionic compounds? NaCl, let's draw the compound differently. It's positive and negative ions. If you've got lots of positive and negative ions, what's going to happen? As a solid, can it conduct electricity? No, it cannot conduct electricity. But as a liquid, once it's melted as a liquid or dissolved in water, the ions become free to move. So then they can conduct electricity. So in electrolysis, an ionic compound is melted or dissolved in water, and that way the ions are free to move. And using electrolysis then, we can separate the positive ions from the negative ions. So the liquid or solution that's able to conduct electricity is known as an electrolyte. So when we say electrolyte, we mean water or liquid that has an ionic compound dissolved inside it. So there's our beaker full of water, and if you add your salt, then the positive and negative ions separate from each other. That's what we refer to as an electrolyte. That means it can conduct electricity. Okay, so what is actual electrolysis? It's when an ionic compound is melted or dissolved in water, and that means the ions become free to move. The liquid solution is known as an electrolyte, and it can conduct electricity. Electrolysis is the process by which ionic substances are broken down into simpler substances through the use of direct electric current. So it's taking your NaCl and separating the Na from the Cl. Now, let's label this diagram. It says label the diagram to describe what happens during electrolysis. So electric current flows through the electrolyte. So we've got electricity flowing through this pink thing here, the pink part, the electrolyte. In fact, let's zoom in. So the pink part, let's even zoom in just a bit more. Yeah, that'll do. So the pink part is the electrolyte, and that conducts electricity. It's a liquid containing the dissolved or molten ionic compound that can conduct electricity. Now the negative ions within that fluid, so I'll have to draw it here. Within the fluid, you are going to have positive ions, which we call cations because cats have paws, and cations are positive, and negative ions are negative because anion sounds a bit like onion, and onions make us cry. So we've got a negative and we've got positive ions. Now what happens is the negative ions, this is our positive electrode, and our positive electrode is called the anode. So all our negatively charged anions, all the negative ions will come and join and attach here. Now all the positive ions will come and attach on this side here. So if you have an electrode and it's positively charged, like an anode is, the negative anion will come and attach itself there. And so what we've done now is we have separated the negative ions from the positive ions. So if this was our positive anode, then a negatively charged anion will come and attach itself here. And what the anion will actually do is give us electrons away. Now on the negative electrode, which is the cathode, the cations, so this is negative, the cations will come and attach here. And they will gain electrons. So anode is the positive electrode where negative anions move and lose electrons, and the cathode is a negative electrode where positive ions, cations, move and gain electrons. Remember, what are both of these made of? They're both made of graphite. And why is it they're made of graphite? Because graphite can conduct electricity, but it's also unreactive, so it doesn't mess up the experiment, doesn't react with anything in the experiment. Yeah, actually, apologies. I did read 3.18 as combined, but I've obviously made a mistake. So 3.18, you're absolutely right. It is combined. Apologies. So if anyone missed that section and you're doing combined, please do go back and watch that section. There's a triple element to it, which is in topic 5. So apologies. So you're right, it is combined. My mistake. So electrolysis is the breaking down of something a bit complex into a simpler substance using electricity. An electrolyte is a liquid or molten substance that contains ions and can conduct electricity. Okay, products of electrolysis. The ions discharged when an aqueous solution is electrolyzed using inert electrodes depends on the relative reactivity of the elements involved. Okay, at the cathode, the least reactive positive ion gets discharged. Okay, so this is gonna make sense in a second. So what that means is, we've got a negative ion, we've got our negative cathode, and we've got I'll just put the anode over here, which is positive. Now, when you add, I'll just do it here. Now there's two things you can do. Let's say you've got salt, table salt, NaCl. You can heat that up to about 800-900 degrees and it'll turn to a liquid. Now if that turns into a liquid, what will happen is if we've got positive and negative electrodes, now and our electrolyte, the liquid part is just NaCl. So the only thing in here is Na plus and Cl minus. What's going to happen is all the Na plus will attach itself at this electrode, the cathode, and all the Cl minus will attach itself to the positive electrode. There's no other ions inside there. It's just Na plus and Cl minus because we've just taken NaCl and melted it. Now, if you get NaCl and you add it to water, and then you blast it with electricity, now the NaCl breaks up into Na plus and Cl minus, but also the H2O. So think back to what we just did in acids, breaks up into H plus and OH minus. So when we're answering this question here, we're talking about solution, aqueous solution. So not only have we discharged a positive ion and a negative ion from the salt, we've also got H plus and OH minus. So the rule is whatever gets formed at the cathode is the least reactive positive ion. So if it's H plus is the least reactive, then it makes hydrogen gas. But if it's more, so for example, hydrogen gas is produced unless there's a less reactive metal like copper or silver. But if this was, for example, sodium, let's use sodium chloride as an example. That means that's going to be Na plus. What's more reactive, Na plus or H plus? But Na plus is much more reactive. So H plus is the less reactive one. So what's going to attach here is H plus. So if you've got a choice of Na plus attaching and H plus attaching, the Na plus won't attach. It's much more reactive. The less reactive one attaches. Now at the positive anion, the positive anode, we've got, if we're again using the example of NaCl, we've got Cl minus and we've got OH minus. The rule is at the anode, if it's a halide ion, meaning if it's from group 7, so if it's from group 7, it is discharged, meaning it's Cl minus that's going to attach here. And it's going to give its electron away. Now, if Cl minus doesn't discharge, let's say this wasn't NaCl, instead it was Na sodium sulfate instead. Then we didn't have Cl minus here, instead we've got SO4, 2 minus, then it's the OH that attaches itself. So the rule is if a halide ion is present, it's discharged as a halogen gas, chlorine, bromine or iodine. If there's no halide ions, then oxygen gas is produced from the hydroxide ions, OH minus. So here's some examples. So we've got copper chloride. In fact, I know this topic is, so excuse me. I know this topic is poorly understood sometimes. I hope everyone's understood what I've just gone through. But what I'll do is I'm going to restart tomorrow from here. 5.25 products of electrolysis. So tomorrow we are going to start with products of electrolysis, 3.25, and then we're going to move on to topic 4. Now that will be starting at 2:30, and we're going to continue the chemistry session then until all of 3.25, all of topic 3 is finished, all of topic 4 is finished, and then finally then we'll move on to physics, topic 6, which is radioactivity. Okay, so we'll be back in 5 minutes. So we're going to take a 5-minute break. Thank you, everyone. I'll see you guys in 5 minutes. So at 3:33. So just to answer a question by Subhan, will there be enough time for separate chemistry tomorrow? I don't think there'll be enough time tomorrow, but what I'll do is I'll make a separate video for it. There are certain elements of triple that I've covered the majority of triple. I'll have covered all of combined, and I will have finished all of triple, except a small amount from topic 5 biology and topic 5 chemistry. So I will make a separate recording of that, and I'll post it onto YouTube as soon as I can. Okay, welcome back. We're going to continue with topic 5 chemistry. Now, there is going to be some sections within this that are triple award. I'm going to teach them at the same time. And then as soon as this finishes, we're going to move on to topic 7, physics, which is space, astronomy. So electromagnetic waves are transverse waves that transfer energy and travel at light speed through a vacuum. They can move through a vacuum and include various types such as radio waves, microwaves, infrared, visible light, ultraviolet, X-rays and gamma rays. Actually, so just to clarify, tomorrow we're covering biology topic 5, so that will include all the triple award content, hopefully. Tomorrow I'm just going to continue until I finish. Now, I think my first session might finish early, so I might do the recording for topic 5 at that point. For topic 5 chemistry, so if I do start early, we'll send a message out on the WhatsApp channel, so just please keep an eye on the WhatsApp channel, and we'll send it out on the YouTube channel as well. If we do start early for combined, it will just be for triple award only. It will just be a slightly quick start, maybe a slightly early start so we can fit it in. Otherwise our normal session will start at 1:30, but if I can start an hour earlier, we can try and fit in a topic 5 chemistry, because it would be nice to finish everything. At that point then there'll be the odd spec point that might be missed, but the majority has been then covered, and we've finished like 95, 96, 97% of the content. Okay, so electromagnetic waves. Transverse waves, so that's very important to keep in mind. That electromagnetic waves are transverse waves. They transfer energy and travel at light speed through a vacuum. So all electromagnetic waves are that. And examples are radio waves, microwaves, infrared, visible light, ultraviolet, X-rays and gamma rays. They are all electromagnetic waves. Now the electromagnetic waves spectrum is a spectrum, encompass all the EM waves. So we've seen the different examples, and as you go from left to right, on the radio wave side of things, there's the radio waves, you've got much larger wavelength, and when you get towards gamma rays, you've got much smaller wavelengths. And so you've got a big wavelength on this side, you've got a smaller wavelength on this side. On the left, on the radio wave side, you've got a smaller frequency, but gamma rays, when you get to that side, as you're going across from left to right, the frequency increases. Now, if you consider that each wave carries the same amount of energy, what that means is when you get gamma waves, where you've got lots and lots of waves, it's really highly energetic waves. So a lot of energy that gets transferred through. So how do electromagnetic waves transfer energy? These waves transfer energy from one place to another and are classified based on their frequency and wavelength in the electromagnetic spectrum. So energy is transferred by electromagnetic waves as radiation. So electromagnetic waves are a transverse wave. So they transfer energy and they transfer information, but they do not transfer mass and matter. So the higher frequency waves carry more energy. So this is what I've just explained to you guys. If it's got a bigger frequency, because each wave essentially carries roughly the same amount of energy, it means you have now got something carrying more energy. While lower frequency waves like radio waves carry less energy. Energy absorption. So electromagnetic waves can also, when they can also be absorbed and emitted. So when electromagnetic waves interact with matter, they can be absorbed, which can lead to effects like heating, electron excitation, or chemical reactions. So one thing that we do need to understand is when, for example, an atom absorbs energy, the electron in a lower energy state can absorb the energy and go to a higher energy state. And when that electron releases the energy and goes back to its normal state, it releases it. It can release it in multiple ways, but it can release it as also as an electromagnetic radiation. Okay, core practical. So this core practical is linked to refraction. So we went through refraction yesterday, and now this is a core practical link to it. So place a block on a piece of paper and draw around it. Then shine a ray of light through the block, and the ray of light should be as narrow as possible to give the most accurate results. Then you've got, now mark the ray of light close to the point it leaves the box and where it enters the box. So you're marking it there and there. Now at the same time, you mark the ray of light as it leaves the box. And then you can join your first crosses, your second set of crosses, and in between you can join as well. I know mine looks like a straight line now because I'm using quite a thick pen. And now you draw the normal. The normal's at 90 degrees. Finally, the last two steps, use a protractor to measure the angle of incidence and the angle of refraction. So incidence is the angle that it comes in at. Refraction is the angle, is the amount that it bends when it enters the glass. Now we shine the light from the ray box at different angles by increasing the angle of incidence at 10-degree intervals. So we keep increasing it by 10 degrees and we keep measuring the angle of incidence and the angle of refraction. Okay, groups in the electromagnetic spectrum. Sorry, just excuse me 1 second. Yeah, so the groups that we have in the electromagnetic spectrum are radio waves, which we use for, and these kind of questions do come up, so it's important to remember this. Radio waves are used for communication, broadcasting and radar. Microwave, which is in cooking and mobile phones, satellite communications. Infrared is emitted by warm objects. It's used in thermal imaging and remote controls. Visible light is what our eyes use. That's what enables us to see. UV radiation, ultraviolet, is what's emitted by the sun. Well, they're all emitted by the sun, but that one specifically comes to the earth and it's absorbed by the ozone layer. So where you've got gaps in the ozone layer or holes in the ozone layer, then there's more likely that it can cause skin damage or skin cancer. So emitted by the sun, it can cause skin damage, but can also be used for sterilization. So if we think back to the topic of potable water, to make water potable, you can blast it with some UV radiation and it kills off the germs. X-rays are used in medical imaging to view bones and detect fractures, so using X-rays. Gamma rays are used by radioactive substances. So produced by radioactive substances, they're used in cancer treatment and nuclear reactions. So they're using in nuclear power plants. Now it can cause cancer, but it's also used to treat cancer. So this is something we've already explained. When you go from radio to gamma, you're going from long wavelengths to short wavelengths. But you're also going from low frequency to high frequency. But our eyes can only detect visible light. So we can only detect a really short, narrow band of wavelength. Anything bigger, anything smaller, we can't detect. In fact, we only know of, the only reason we found out about these other electromagnetic waves is because of the temperature. So if you have, for example, a rainbow, So there was a scientist doing an experiment, and he was checking from here to here, and he was checking the temperature of each color. But he did, and it wasn't just through a thermometer. There was an actual experiment, that a reaction that was occurring. Now, when he put it into this area here, the area that we call infrared, there was still a heat energy given off. When you put it into the ultraviolet area, there was still a certain temperature given off. So they then understood that there's other waves that can't be seen, and they started doing lots of experiments around it. Now, all transverse waves, electromagnetic waves are all transverse. These are all points to memorize. They all travel at the same speed. They all travel through a vacuum. They can all be refracted, reflected, absorbed, and transmitted. Now, our eyes detect light from the electromagnetic spectrum, but our eyes have a limited frequency range. So our eyes can only see between 400 and 700 nanometers in wavelength. That's visible light. We have sensitivity to other specific wavelengths. So our eyes contain photoreceptor cells, cones and rods. They only respond to certain frequencies of light, allowing us to see colors within visible spectrum but not beyond it. That doesn't mean that those other electromagnetic radiation don't interact with our eyes, but the only one that we can detect is between 400 and 700 nanometers. And as you get closer to 400 nanometers, that's on your blue side of color, indigo violet. And as you go towards 700, the bigger wavelengths, that's towards your red side. So invisible electromagnetic waves, other electromagnetic waves such as infrared, ultraviolet, X-rays and radio waves exist, but they are undetectable to human eyes because their frequencies fall outside of our visible range. So how do different wavelengths influence absorption and transmission? So material properties, different materials absorb and transmit waves differently. So for example, glass lets visible light pass through it, but it absorbs ultraviolet rays. So different materials have different effects on transmission and absorption. Wavelength and energy. Waves with shorter wavelengths, like X-rays, have more energy and are easily absorbed, while longer wavelengths like radio waves can pass through materials. Now this is something that will come in use at this point when you learn about greenhouse gases. It's a topic from, I think it comes under topic 8. No, sorry, topic 7. No, yeah, topic 8. So when you start looking at the Earth's atmosphere, when we do the next AMALathon, in the Earth's atmosphere, you'll see when they enter long wavelengths, not as short wavelengths, and they leave as long. Sorry, I'm getting this mixed up. I'll just carry on. So waves with shorter wavelengths have more energy and are easily absorbed and reflected, while longer wavelengths like radio waves can pass through. So initially the waves that come into the Earth are longer, but once they've been absorbed and re-emitted, they become shorter, which means they're more likely to reflect and bounce off things. Now, colour and absorption. Objects appear coloured because they absorb some wavelengths and reflect others. This is actually something that's linked to triple. So a red object absorbs all colours except red, which it reflects. So if you're looking at a red object, the only reason you see red is because it reflects red, but it's absorbing every other colour from the spectrum. And this part here we're about to go through in a bit more detail, because it's triple award. And it's essentially that heat absorption, infrared waves are absorbed by many surfaces, converting the energy into heat. This is why dark coloured objects feel warmer in the sunlight than light coloured ones. So darker objects and colours and shinier ones, and duller colours absorb heat energy, whereas brighter colors, white for example, and shinier objects reflect better. So velocity of electromagnetic waves, so this is something which is constant in electromagnetic waves as well. They all have a constant speed in air, and in a vacuum they travel at around 3 times 10 to the power of 8. But when they go into denser materials, they slow down and then they refracted. So if you think back to what I covered yesterday, that was using light as an example, but actually it's all electromagnetic waves that refract. And then refraction occurs. The change in speed causes the wave to bend, leading to refraction, which affects how we see objects through materials like lenses or water. Okay, 5.15, 5.16, so 5.16, 17, 18, and 19 are all triple. So I'm going to go through this so that it's all in line. So we're not having to cut and paste everything afterwards. And so it should only take about 10 minutes to get through. So those doing combined, if you want to do if you want to have like a 5-10 minute break and then come back, that's absolutely fine. I'm just going to go through the next 5 spec points. So 5.15, 16, 17, 18, and 19. That's all triple. And then 5.20 up until the end is 5.20 to 24 is combined. Now for 5.23 and 5.24, I'm actually going to explain it to you through two past paper questions. The best way to explain 5.23 and 24 are through past paper practice. So, what does it mean when waves are transmitted or absorbed? Black bodies. So emission of radiation. All objects continuously emit electromagnetic radiation, regardless of the material or state. So everything is constantly absorbing energy and releasing energy, absorbing it and releasing it. It's just that some things are better at absorbing and some things are better at absorbing and emitting, and some things are better at reflecting. So both things do occur. Now, temperature dependence. The intensity and type of radiation emitted by a body are directly influenced by its temperature. It depends on how much energy it gets. So it's what we were saying before. The electron gains energy, goes to a higher energy state. When it releases that energy, depending on what type of material it is, it will release a different form of energy. This is how radio waves all the way up to gamma rays are produced in the first case, in the first instance. Now, wavelength distribution. So higher temperature bodies emit radiation with shorter wavelengths and greater intensity. So if it's got higher temperature bodies emit, so if you take something to a higher temperature, now it says temperature here, but it can just mean energy as well. So if you add lots of energy into something, it will emit radiation that has a shorter wavelength and greater intensity. So it's more towards gamma radiation side. While cooler objects emit longer wavelength radiation with lower intensity. So we're leaning more towards the radio wave side of things. So what does it mean when waves are transmitted or absorbed? Absorbed is when a wave is taken into a material. So it's absorbed. Transmitted is when the wave passes through the material. Now, how does a surface... Now, there are two other scenarios. One is the light can reflect off. And as we've just covered a second ago, the light could refract as well. Now, radiation. How does the surface of an object affect the radiation emitted from it? So when it's white and shiny, the materials are poor emitters and they're poor absorbers of infrared radiation. So white and shiny materials, as I've said before, are poor absorbers, poor emitters. Whereas black and matte materials are good emitters and they're good absorbers of infrared radiation. So let's look at a couple of scenarios. A body is at a constant temperature. What that means is the body emits infrared radiation at the same rate it's absorbing it. So if something stays at one temperature, it means it's taking heat energy at the same rate it's releasing heat energy. Now if the temperature of a body is increasing, that means that it's absorbing more infrared radiation than it emits, causing temperature to rise. If something's cooling down, then it's releasing more infrared radiation than it's emitting more infrared radiation than it's absorbing, causing the temperature to reduce. So if we look at temperature of the Earth now, it depends on the rate of absorption and emission of radiation and the reflection of radiation into space. So over the last 200 years, the temperatures on Earth have started to increase. This is because in the past, radiation emitted into space. So in the past, we didn't have an increase because the radiation emitted into space was equal to the radiation absorbed from space. Today, there's less emission going back into space, and so there's an overall net absorption of radiation. Because we've increased the carbon dioxide gas around. So let me just use a different pen. So we now have this additional carbon. We always had carbon dioxide, and carbon dioxide is a greenhouse gas. And what that carbon dioxide was doing was absorbing. So what carbon dioxide does, if I show you, it takes in radiation, so it absorbs the energy. So that could be thermal energy in the form of infrared, but it's energy. But what the carbon dioxide also then does is it re-emits that energy, but it re-emits it in every single direction. Some of it back towards the Earth. Now, the more carbon dioxide that we have in this layer, this layer of carbon dioxide, then the more energy that's being absorbed, that's being emitted back towards the Earth. And that's what's happening now. We're putting more and more carbon dioxide into the atmosphere, so more and more energy is being emitted back towards the Earth, hence causing all these greenhouse effect issues. Okay, how can we measure the infrared radiation emitted by different surfaces? So this is something called a Leslie cube, and with a Leslie cube you have you can put hot water into it, and you've got different surfaces on each side. So you've got, for example, black and shiny, black and matte, white and shiny, white and matte. Then you pour hot water into it and replace the lid. Then use an infrared detector to record the amount of radiation from each surface, and then that allows you to compare and see that the best emitter, the best absorber of heat energy and emitter of heat energy is black and matte. So for valid results to be taken, it needs to be repeated. And what do we do with repeated results? We remove anomalous data. This is so that you can identify anomalous or outlier results and calculate averages. There also needs to be control variables. For example, the infrared detector should be held at the same distance from each surface. Detector should be at the same distance from the surface each time. So white surfaces will emit less infrared. Black surfaces will emit more infrared because black surfaces are better at absorbing and emitting. So matte surfaces, they also emit more infrared. They are good emitters. And shiny surfaces will emit less because they are poor emitters. They're better at reflecting. And we are back to combined. as soon as this is finished. So because we've got 20, 21, 22, 23, we've got 5 spec points to go through now. And the last 2, we're going to go through 2 past paper questions. Now the 2 past paper questions are partly linked to radiation, but it's not the radiation part that I want to look at. We'll still cover it. It's how electromagnetic waves are made. That's the part that I want to be looking at. And I just want to show you what kind of questions come up and how the answers are exactly the same. Now, like I've explained to you guys before, the AMAL method is really beneficial if you're aiming for grade 9s. So right now, we're doing the activate phase. We are activating our brains. We're understanding everything. And once this finishes, it is then really important from Friday onwards. I'm hoping some of you have been doing it anyway after sessions, but especially Friday onwards, it's one exam paper per day. If you hit that 1 exam paper per day, you can get to your grade 9. By the time you get to your exam, you'll have gone through, for biology, chemistry and physics, you'll have gone through every single past paper. When it comes to your paper 2 exams, it's going to be a little bit tighter, but at that point you're going to be right in the middle of exam season, so you're going to naturally be able to do a lot more prep and get through a lot more past papers. And you'll obviously have the pre-exam workshop with myself as well. Okay, so what are the risks of some parts of the electromagnetic spectrum? Now, one thing we've just got to keep in mind here, they're more ionizing. The bigger the frequency, the more ionizing they are, because the higher the frequency, the greater the risk, because they have more energy. When we're at radio waves, and when we're talking about radio waves, we're not talking about anything dangerous because it's such a big wave, it doesn't carry much energy. Microwaves can, to some extent, we obviously use it to cook food. Infrared can be used to cook food, so there's an element of burning. Your ultraviolet, X-rays and gamma rays, these are 3 that actually cause cancer. So infrared radiation can cause burns. Microwave can be used to heat food, but this can also cause burns as well. Ultraviolet waves, X-rays and gamma rays. So you probably noticed they didn't say anything about radio waves. That's because radio waves are harmless. Ultra waves, X-rays and gamma rays can have hazardous effects on human body tissue. In other words, they can cause, in a sense, almost like similar to what we would call a denaturing of tissues. So the energy is absorbed, and it causes a breakdown it can cause a breakdown of the bonds between the cells, between the atoms within the cells. Ultraviolet waves can cause skin to age prematurely and increase the risk of skin cancer. X-rays and gamma rays are ionizing radiation. That means they give things. Imagine if you have an atom and the gamma ray goes through and it passes on so much energy that that electron breaks free from the energy shell. So you know how we said the electron can go from one shell to the next shell? If it's already in the outer shell and it absorbs all that energy, it can then separate from the rest of the shell. At that point, it's been ionized because it's lost an electron. But because they're so ionized, they can cause mutation of genes and cancer. So you can imagine if something causes that, can pass on that much energy to an electron and cause the electron to dissociate or delocalize from the rest of the atom, it can break bonds. Okay, uses of electromagnetic waves. These are all very common questions. Radio waves, microwaves and infrared radiation are all relatively safe. They all have wavelengths longer than visible light and have a lower frequency. They're all used for communication purposes. So radio waves can be used for transmission. Now this is a lot of extra information that you don't need to know. The main thing is transmission. Propagation through space and reception and different frequencies. It's all correct, it's all additional info. Again, so microwaves can carry signals between Earth and satellites, so it's using satellite communication. Microwaves are used in phones. Microwaves send data between devices, so this is not all microwaves. But the main thing is it's communications, and it's used in our microwaves to cook food as well. So like I just said, we can use it in cooking, we can use it in radar, we can use it in medical use, astronomy, so we can use microwave based telescopes, and we can use it to dry things like paper and wood in industry. Infrared, use it in remote controls, fiber optic communications, night vision, there's medical uses as well. You can use infrared for thermal imaging, remote controls. I'm not going to go through all these things. You don't need to know this much. This is just a lot of extra info. The main thing is thermal imaging, remote controls. It's used for cooking. Visible light. The main thing is we can use it to see, but we also use it for fiber optic communication. So that's really fast Wi-Fi, or it helps us connect our internet and phone. Okay, so radio waves, if we're going to summarize it, used for communications and for communication. Microwaves are mainly used for satellite and mobile communication and cooking. Infrared is used for remote controls and optical communications. Visible light to see things. Traffic signals, LED screens, to see. Optical fiber communication, also high speed internet and data transfer also uses light, visible light. Ultraviolet light can be used in lots of things as well. So we can use it to sterilize. It can be used to kill germs in hospitals and labs for some of the equipment that gets used. It can be used in skin treatment for things like psoriasis and jaundice. People use it on an everyday basis, although it's not recommended to use it too much. It can be used for tanning. It can be used for fluorescent lights because it helps makes bulbs glow and give off a visible light. It can be used for forgery checks. So you probably notice sometimes when you pay for things, they put like white mark on it. So if you check for forgery, it can be used in forensics to help find blood, fingerprints and other clues. Okay, so X-rays show bones and organs. So X-rays can be used for X-rays to show bones and breaks. It can be used in CT scans. Now CT scan, PET scan, all this is triple award. So if you're not doing triple award, don't worry about what a CT scan is. For triple award, it is something that we've already covered, but it helps make detailed 3D images. And it's used for cancer treatment. That's a really big one. High energy X-rays kill cancer cells as radiotherapy. It's used at airports for scanners to check luggage, custom checks. It can be used for material testing as well. Gamma rays, the main ones are cancer treatment and to sterilize equipment. So in radiotherapy, it's used to kill cancerous cells. So anyone that covered CT and PET scans, you can use gamma rays in those. You can use it for sterilizing equipment. So, for medical tools, just before you get your scalpel, the doctor uses a scalpel, it's blasted with gamma rays a certain while before, and it's left in a plastic bag, meaning nothing can now contaminate it. It's been irradiated. Once it's been irradiated, we're going to learn about this tomorrow. So I'll just explain this to you now, but we will go through this tomorrow as well. So you've got a plastic bag. Inside here you've got a knife, a scalpel that a doctor's going to use. You irradiate it, so you blast it with gamma rays, and the gamma rays have short half-lives, meaning they finish quite quickly, stop being radioactive quite quickly. So when it touches the surface, it kills all the germs. Now the doctor will only open that bag now just before he or she operates. Okay, so how are radio waves generated? Now I'm going to go through this and I'll do my best to explain it to you, but it makes a lot more sense when you see past paper practice questions linked to it. So radio waves are generated by the movement of electric charges, typically using an oscillating current in an antenna. So radio waves are generated when there's an oscillating current in an antenna. So it's the movement of electrical charges, meaning electrons. When the electrons, so remember what we said before, when an electron moves from one level to another energy level, when it drops back down, it releases energy, electromagnetic waves. And that's what's happening here. When the oscillating current, and what moves the current? The electrons. When they're oscillating, they release radio waves. So now a transmitter creates an alternating current at a certain frequency. The AC, the alternating current that is, makes changing electric and magnetic fields, meaning the electrons are oscillating. They're moving from, because it's alternating current, it means you've got electrons going in one direction, the other direction. That constant movement of electrons going back and forth generates a radio wave. And that creates a radio wave that travels through space. Now that alternating current flows into an antenna. So when an alternating current flows into an antenna, it makes electrons move back and forth, and that creates the radio waves. So the main part you need to learn here, I'm just going to get rid of all this. What I need you all to understand is when an electron goes from one state to another state and it drops back down, it releases energy. So what's happening here is when the alternating current, because the alternating current means it's going in both directions, when it flows into an antenna, it's making electrons move back and forth. So it goes to a higher energy state, it goes to a lower energy state, it goes to a high energy state, a lower energy state. So it's absorbing energy, it's emitting energy. It's absorbing energy, it's emitting energy. What it's emitting is radio waves. That creates radio waves that spread out into the air. Now, when those radio waves are absorbed by an antenna, so let's say those radio waves have now been made. Now I've got a little radio, I've got my antenna. When this antenna absorbs my radio waves, so radio waves hit the antenna, they make electrons in that metal move. Now, when you do electricity, what you're going to understand is what makes electricity, allows electricity to travel through something, or what allows a charge to carry through a wire is the electrons. So now we've got electricity flowing through this metal and into this device. So the moving electrons create an alternating current in the antenna that matches the wave's frequency. The radio receives boost and decodes the AC signal to get sound, pictures, or data, like in radios, TVs, or Wi-Fi. So what we're saying is an alternating current causes an electron to get excited, so it absorbs energy and releases it. In doing that, it makes a radio wave. When that radio wave, because radio wave is a transfer of energy. When that energy reaches the antenna, ultimately it's just a thin strip of metal that excites the electrons, and the electrons start now carrying charge based on the amount of energy that they've just absorbed through this metal wire. And then the movement in the electrons is then converted into sound or a picture or some sort of data. Now, atoms can undergo changes in their structure due to various processes. So an atom loses or gains electrons and becomes an ion. This can happen due to radiation or collisions. So something collides into this. Now that's more linked to the next topic. And this is all linked to, this is an explanation for the next topic. But the main thing that you need to understand from here is what I've already explained. If you have an electron here, if it absorbs energy, what's it going to do? It gets excited and goes to a higher state. When it drops back down, it releases that energy. So let's have a look at these questions. This first question says X-rays and radio waves are part of the electromagnetic spectrum and have different uses. These radiations are produced in different ways. X-rays are emitted when electrons within an atom go through their energy changes. So what we've just been explaining. Radio waves are produced by electrons in circuits. So we're talking about what we were just explaining here. I've explained it in my own words, but I want you to see what the ideal answer looks like now. And it is when AC causes electrons to oscillate. And the oscillation produces electrons, produces radio waves. So radio waves are produced by electrons in circuits. Compare X-rays with radio waves. Your answer should refer to the uses of both type of radiation. So that's your first half of the question. No, this is a 6-mark question. They've been nice. This would have been a nice paper. This is a paper where they would have wanted students to do better in physics, because the year before, it's possible the results weren't as good. So that's a nice 6-mark question, because this is a higher level question. And look what it's saying. Just to write down uses of both types of radiation. And then the different ways that electrons are involved in producing X-rays and radio waves. That's the bit I want you to memorize. So let's look at it. Let's first do a comparison. X-rays are high frequency, short wavelength, they're ionizing, and they're high energy. Whereas radio waves are the opposite. They're low frequency, they're long wavelength, they're not ionizing, and they have low energy. Now, X-rays, let's look at uses first. X-rays are used in medical diagnosis to check for broken bones. It's used in radiotherapy, treatment of cancer, airport security, revealing counterfeit art. So as long as you put, I'd say 2 or 3, that's sufficient. Radio waves are used for broadcasting television, broadcasting radio, communications, satellite transmissions, mobile phones, radar. Now there's one point I just want to make before we move on. You know, if someone's doing really good revision, and there's a few different ways to revise. One of those ways is making flashcards. And when someone makes a flashcard, the best way to use flashcards is write a question on one side and write an answer on the other side. Okay, now what's better than a flashcard? Just past paper practice. Sometimes people make lots and lots of notes, but making lots and lots of notes is pointless if it's just passive. The better use of your time is to look at past paper questions. Past paper questions automatically become like flashcards, because you got your question on one paper, you got your answer on another paper. So use flashcards in that manner. Look, we've just gone through all of that learning, but look how much of that learning just got covered in this one 6-mark question. We've covered the uses, and now we're covering how they're made. So past paper practice, past paper practice, past paper practice. You want to get a grade 9? Past paper practice. Now, X-rays are emitted when electrons change energy levels. So why do they change energy levels? So electrons in lower energy levels can absorb energy. The electrons become excited, they move to a higher energy level. And when the electrons return to the lower energy level, the electrons lose energy as radiation. The electrons need to lose a large amount of energy, so they emit X-ray radiation. So if an electron loses lots of energy, then it's an X-ray. If it doesn't lose too much energy, then it could be a different electromagnetic wave. Okay, so I hope that makes sense. Because what we're saying here is, you've got electrons in a lower energy level. And the electron gains energy, it absorbs energy, it goes to a higher energy level. When the energy returns to the lower energy level, it loses energy as radiation. Now if it loses lots and lots of energy, then it will become an X-ray or a gamma ray. Now let's look at radio waves. So electrons oscillate in electrical circuits. So in a sense, the similar thing is happening as this. The electrons are still moving up and down. Now the current, which is the flow of electrons, they continually change direction because it's AC. the current flows up and down the aerial. That's alternating current. This generates a radio wave in the air around the aerial. The frequency of the radio waves correspond to the oscillation frequency. Now next time a question comes up on radio waves or on X-rays or even a gamma ray, for example, if it says gamma ray, ultraviolet, another form of radiation, ultraviolet, X-ray or gamma ray, then you just write this down. If radio waves come up, you just write this down. So let's try another question now and see and compare. This question is now radio waves and gamma radiation. Now gamma radiation, is a little bit different. You know how we've just said X-rays emitted? It's true. This is how gamma radiation is made. That's correct. But gamma radiation is also, it's not just electromagnetic radiation. It's also radiation, as in alpha, beta, gamma radiation. So it comes into both categories. Now this question says compare how these two electromagnetic radiations are produced. So let's look at the mark scheme. So let's first look at radio waves. Radio waves are often produced intentionally by humans. Let's just go through the radio wave bits first. Radio waves are produced by free electrons. The radio waves are produced by oscillating electrons, alternating current, we've said that already. Radio waves are produced by electrical circuits or aerials, we've got that down already. Now this doesn't want as much detail as what was in the previous question. Now let's look at gamma rays. Gamma rays are often produced spontaneously and randomly. We haven't covered that yet, we're going to cover that tomorrow. Gamma rays may result from radioactive decay, we're going to cover that tomorrow. Gamma rays produced in the nucleus. Gamma rays produced by energy changes, that's something that we've got, so that's okay, we'll get a mark for that. At the moment we've covered this. Gamma rays produced to stabilize the nucleus, that's something we're going to cover tomorrow. Gamma rays may be produced as a result of fission and fusion, that's something that we're going to cover tomorrow. So there's a lot of points there from gamma that we need to cover tomorrow, but part of this answers gamma, but not all of it. That would answer for X-ray and ultraviolet. This will answer for every single time radio waves come up. And if you see what's written here, where I've put the dots, this is your answer for how gamma rays are produced. So now if a question comes up, you've got ultraviolet sorted, you've got X-ray sorted, you've got gamma ray sorted, and you've got radio wave sorted. Now, I've noticed this question's come up every 3 years. The last time it came up, I think it was 2022. So there's a possibility it could come up now. I think it was 2019 it came up, 2022. These two questions are 2019 and 2022. And both times radio waves came up. Half the marks were on radio waves. Okay, that is the end of topic 5. So topic 5 is now complete. So tomorrow we're going to move on to topic 6. Now right now we are going to move on to astronomy, which is topic 7. So those that are going to be doing astronomy, if you can give me 2 or 3 minutes, then we're going to start astronomy. So we're going to restart at 25 past. welcome back everyone. Let's continue now. So I'm assuming everyone that's on now is triple. Thank you for those that have been watching live. It always helps to have a few people on. I know there's a lot of people that watch out of sync from a time perspective, because usually when our sessions finish, there's been around 300 views, which means people are sometimes 5 minutes behind, 10 minutes behind. It would be wonderful to have all 300 or 200 watching at exactly the same time, but it doesn't make a difference to us. We're happy if it's one person or if it's 100 people, 1,000 people. It's exactly the same. So if we look at this, we have completed a lot of this actually. We've got biology completed, topic 1, 2, 3, 4. So we're finishing 5 off tomorrow now. So that's going to be there. We've almost finished topic 3 off. We finished physics. We didn't look at physics 1, no, chemistry 1. We finished. And we've now finished acids and alkali. We've just got this bit to finish. We've got this bit to finish. We've got C4 to finish. And we've got radioactivity to finish. So that is tomorrow's aim. So we've finished all of physics as well. Now from the triple stuff, we finished this. We did the EM triple, but not the light triple. And we have not covered B5 health triple only, and we're going to finish off astronomy now, hopefully. So, and we're going to have this left. So by the end of tomorrow, I've just made a mess of this. Hopefully by the end of tomorrow, we'll have completed this, all of this. So the only things that we'll have actually missed out, and I apologize for not getting this in, is P5 light, B5 triple, but I'm going to try and finish that alongside the biology and separate science chemistry. So I will do my best to do the separate science one as a separate video and then upload it. I'll probably just do a live session randomly. So if you see it pop up and you want to join, you're more than welcome to. Otherwise, I'm just going to do the recording and upload it onto YouTube over the next week or so. It'll definitely be before your exam, but it will be next week because I'm going to be away for the next few days after this has been finished. But again, thank you for those that have been here for the full AMALearning journey. Okay, so let's get started. Astronomy. So what is the equation that you would use to calculate weight? Weight equals mass times gravitational field strength. W equals mg. Weight is measured in newtons. Mass in kilograms. Gravitational field strength in newtons per kilogram. So we know this already. We know what weight is. Weight is a force that pulls down on any mass or matter. So what is the gravitational field strength on different space objects? So, or different parts of space, so different planets. So Mercury has a gravitational field strength of 3.7, Venus close to 8.9. Well, you can see there. So Jupiter has a really strong gravity. So that if we were on Jupiter, we would feel that force. On some of these other countries, other planets, we'd feel a lot lighter, but on Jupiter, we would feel like we were getting crushed. Now, if you get a question, it always goes back to W equals mg. And if it says it's on Jupiter, they will give you this gravitational field strength. So instead of timesing by 9.8 like we usually do, what makes this triple is that you'll instead do W equals mass times the new gravitational field strength that you've been told in the question. And for that reason, it's been made higher and triple content, or triple content only. Right, solar system. What are the main components of our solar system? So our solar system is made up of the sun, 8 planets, dwarf planets, and the moon. And now, just so you're aware, I'm going to go through the more straightforward stuff quite quickly, but please ask questions if I've gone too fast, because I do want to spend more time on things like redshift. Topics like redshift, I think, need a lot more time. So our solar system is made up of the sun, the 8 planets, the dwarf planets, and the moon, and also the moons that orbit the planets. It's part of the Milky Way galaxy. The sun formed from a cloud of gas and dust pulled together by gravity. So that's how all stars are formed. Now, one thing we've got to understand here is that everything that has mass or matter, particles, has a gravity. So the sun is obviously something massive. So for that reason, it has strong enough gravity to pull everything towards itself. But if you think of even a human being, although we can't see it, we attract dust towards ourselves. Tiny little microscopic particles that will be orbiting us. Because we have a mass, that means we have a gravity. So when a star explodes, it releases something called a nebula. Lots of dust and gas clouds everywhere. So if you imagine this whole slide now is dust and gas clouds, what will start to happen is, if I've got more dust and gas clouds here compared to everywhere else, any dust that comes close to it will get pulled in. So this particular bit of dust and gas cloud here starts getting heavier and heavier, increasing and increasing in gravity. And the stronger it increases in gravity, the more it pulls towards itself, until you have enough particles to start causing changes in temperature. But we'll go through that in a bit more detail when it comes up. So we know the order of the planets: Mercury, Venus, Earth, Mars, Jupiter, Saturn, Uranus, Neptune. So what are our early models of the solar system like? So in early models of the solar system, Earth was considered to be at the center, and there was some supporting evidence for that theory because people only observed things with the naked eye, as they didn't have telescopes, so they would not have known. The sun and the moon seemed to traverse the sky in the same direction every day, so it just seemed like the sun and the moon were going around the Earth. So the automatic conclusion was that the Earth is at the center. That's called a geocentric model, and the thought process makes sense. We know it's not correct, but it makes sense. And then it moved to the heliocentric model, from geo to helio. Helio meaning full of helium, like the sun. So the heliocentric model replaced the previous model showing that planets orbited the sun, and this was based on Galileo's observations of Jupiter's moons. And what Galileo noticed was that, because prior to Galileo saying this, everyone assumed the Earth's right in the middle and everything's going around the Earth. Everything is going around the Earth. But Galileo noticed that where Jupiter is, there's something going around Jupiter. Afterwards, we found out that was a moon. Galileo's observations disproved the geocentric model, but he lacked sufficient evidence to confirm the heliocentric theory. So he presented then that we must be going around the sun, but there was a lack of evidence. Now Copernicus, he made clever observations with a new discovery called a telescope. Although Galileo was using a telescope as well, but Copernicus would have been more advanced. And it placed the sun at the center of the solar system. So Copernicus provided the evidence. So what other ideas have changed now? Solar system. So we now know planets orbit the sun, and have moons. We know that the Earth is the bit that rotates. We know Pluto, well, Pluto is still a planet. It's not correct to say it's no longer a planet. It is still a planet. It's just classed as a dwarf planet. So if anything, it's classed as a dwarf. So but it's not one of the major planets. It could potentially just be a moon that was knocked out of sync by something, like by another object that hit it. We also now know that orbits aren't circular. They're elliptical, like a squash circle. There's more planets than they initially thought, and multiple solar systems exist. So around every star, we have another solar system. And from a geocentric model perspective, we now know the Earth isn't flat, and planets follow a fixed path around the sun. Orbits. So how do the orbits of planets, moons, satellites and comets differ? So there we've got planets orbit the sun in an elliptical path. A moon orbits a planet in a circular or elliptical path. Artificial satellites, so man-made objects that orbit planets or other celestial bodies. And we've got comets. They orbit the sun in an elliptical path, often with a tail. So if you have a sun, the comet goes quite far from it, but then it's pulled back, and as it gets closer and closer, it gets faster and faster because the effect of Earth's gravity, the sun's gravity, is getting stronger and stronger. Now it's made of, comets usually made of ice and dust and things, and it starts melting as it gets closer to the sun, so it ends up with a tail. And when it goes around the sun, it increases in more speed, and when it's released, it's almost like a slingshot. So what happens during a circular orbit? So this all links to centripetal force. But now all we're doing here is we're taking our understanding of centripetal force and we're applying it to planets. So nothing too different to what we've already learned. So for circular orbits, the force of gravity can lead to changing velocity but unchanged speed. Okay, now let's quickly just tackle this one point first. If something is going around in a circle and is going around a circle at 30 meters per second, then we would say its speed is unchanged, because its speed is a scalar quantity. Scalar meaning it just changes in size. It only has magnitude. So from a speed perspective, the magnitude just stays the same. It's 30 meters per second. But from a changing velocity perspective, velocity is magnitude and direction. Now what's happening? Every second, the direction is changing, which means the velocity is changing. So for example, when the moon is going on the Earth, it has an unchanged speed, but its velocity is constantly changing because the direction is constantly changing. So the force of gravity causes a satellite to accelerate. Now, when we say acceleration, what do we mean? We mean acceleration is the change in speed, velocity divided by time. Now, if the direction keeps changing, the velocity keeps changing. So that means our velocity is changing. That means we have acceleration. So the gravitational pull, the centripetal force, as in the pull of the Earth towards on the moon, that centripetal force is causing the satellite to accelerate. And the acceleration causes a change in direction. The velocity changes because, so the velocity, don't worry about this part too much, this part. Velocity changes because the direction changes. And that means we have acceleration. Now, why does it maintain a circular orbit? So let's have a look. there's the Earth, there's the Moon. This is what we went through before when we covered this. The Moon is naturally, through its momentum, is moving that way, but the Earth's gravity is pulling it inwards, so that's centripetal force pulling it towards the center. So now the Moon moves somewhere in between that way. So now it's over here. Now if it was just to continue moving, it would keep moving that way, but instead the Earth pulls it towards the center because of the gravity, centripetal force. So it goes somewhere in between, it goes over here. Now it would continue moving this way, but the Earth is pulling it towards its center, so it ends up moving somewhere central to the two. And in the end, what do we end up with? The Moon moving around in a circle. And what stops it from just flying off in a random direction? The centripetal force stops it. The gravity pulling it towards the center. So it's like having something attached to a piece of string and spinning it around in circles. So you could get a glass full of water, put it on a tray, attach it to some string, spin it around in circles, and the water won't fall out because the centripetal force stops the water from falling out. So for a stable circular orbit, an object must travel at the correct speed. So for a circular orbit, an object must travel at the correct speed. An object maintains its orbit when its velocity balances the gravitational force of the object it orbits. Meaning, when we have force equals mass times acceleration. So when something has a constantly changing velocity, we have acceleration, it already has a mass, so we now have a force. So when the Moon is trying to go that way, it's due to the force, it's the force due to acceleration. But now the Earth is pulling it also in this direction. Now, if the force is balanced out, then what's going to happen? It's just going to maintain, sorry, I've done it wrong there. Let me show it here. If the forces are equal, at the point that they're equal, it'll maintain a steady circle. It'll maintain one particular orbit. Now, what's going to happen if it speeds up? If it speeds up, this force going in that normal direction increases, because now if the velocity is faster, the speed is faster, and we've got a greater velocity, acceleration has increased. That means the force has increased. The gravitational pull towards the Earth hasn't changed, so that means the orbit goes further away from Earth. If it gets faster, the orbit goes further away from Earth. If it slows down, that means acceleration decreases. If acceleration decreases, force decreases. So now there's a bigger centripetal force pulling it inwards, so the orbit becomes smaller as well. Now I hope that makes sense. If someone doesn't mind, if it makes sense and you're watching live, if you can let me know if that makes sense, please. And if it doesn't, please say so I can explain it one more time. So if we increase the speed, increase the velocity, because force equals mass times acceleration, we increase the acceleration, which means we increase the force. So the force acting on the Moon is going to be bigger. And we've always got the centripetal force pulling the Moon towards the Earth. Now if we increase the acceleration, because we increase the velocity, we've automatically increased the force. Excellent, 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 thank you. So if you increase the force gets bigger. That means the moon starts using a bigger orbit. So this is what I've just explained on the previous slide. So I'm going to leave it there just for those that are revising, so you can read through it. And what you're going to find is that what I've just explained is explained within these notes now. So how does orbit size affect speed, and what is needed for a stable orbit? To keep a stable orbit, the radius must change with speed variations. So objects in small orbits travel faster than those in larger orbits. That's because they're closer and they have a greater pull from the Earth's gravity. But as they get faster and faster, the orbit also becomes bigger. So to change the orbital speed, an object must also adjust its orbit's radius for stability. Okay, now this is the bit that needs a lot more explanation. Steady state versus Big Bang. So two different theories that try to explain how the universe was created. The Big Bang theory suggests the universe originated from a small, hot, dense region, meaning the Big Bang theory is that initially all the Earth's particles, all the matter that makes up the universe right now, not the Earth, sorry, all the particles, all the matter that makes up the universe right now, and all the energy in the universe was all at one central point, one singularity. Now, if you can think back to what I was saying about gravity, the more mass you have, the more matter you have, the more gravity something has. So you can imagine the gravitational field strength. The gravitational strength of that initial point would have been so strong that it would have been able to condense all of the matter in the universe into something really, really small. So then at that point, the entire universe expanded outwards and is continuing to do so. Now, the steady state theory suggests that the universe has always existed. So, for example, it was that big, and what it says is the universe indicates this, and they believe that it always existed, but it's expanding while producing more matter. So it was that big, then it's become that big, then it's become that big, because it's all the time more and more particles are being made. Now, out of the two theories, the Big Bang theory is the one that has more evidence to kind of back it. So what is steady state and Big Bang theory? So there's additional support for the Big Bang theory comes from cosmic background radiation. So from the point of the Big Bang, it would have released different types of energy. And we have that as background radiation. Now, the remnants of radiation from shortly after the Big Bang are thought to have been distributed across the universe. And those remnants can be picked up. So that is evidence for the Big Bang theory. However, the steady state theory can't explain why we have cosmic background radiation. So galaxies exhibit a red shift. Now, redshift I'm about to explain. Distant galaxies display a larger redshift. Now, redshift, if something has a redshift, actually I'll explain this now because otherwise it's not going to make sense. So, if you think about, if someone's got a slinky, so let's just show it like that. Okay, you're stood on one side holding it, and another person stood on the other side holding it, except they're walking away from you. Now, initially, that's the wavelength. When they walk away from you, what happens to the wavelength? It gets stretched, and that's a red shift. Meaning if something's moving away from you, the wavelength gets stretched, it red shifts. If something comes closer to you, then the wavelength, so if something's moving away from you, the wavelength gets bigger because it's getting stretched. If something comes closer to you, we call that a blue shift. meaning the wavelength gets smaller. So currently the galaxy, whenever we're looking at things through telescopes and through the right type of telescope that can detect the waves, it shows that the wavelengths are redshifting, as in they're getting bigger than what they should be. That means they're moving away from us. The wave has been stretched. So that's redshift. That evidence is that the universe is expanding. So that's evidence for both. Because both steady state and Big Bang Theory both state that the universe is expanding. So that is evidence for both things. The movement of galaxies away from us indicates that the universe is expanding, so they're redshifting. The further away, that should say further, not farther, away a galaxy is, actually no, that's correct. The farther a galaxy is, the faster it appears to be moving away. So the further a galaxy is, the faster it appears to be moving away. This suggests that the universe originated from a single point. So that single point is where all the matter and all the mass was. The gravitational would have been so strong, like I said, it would have been like the size of a pinhead. Then after that, it started to expand and it's increasing in speed. And it's redshifting. You can see the redshifting in distant galaxies. So why is the Big Bang Theory the current accepted model for the origin of the universe? So distant galaxies have a greater redshift. Now, does that support the Big Bang Theory? Yes, it suggests the universe is expanding from a single point. Now, redshift directly doesn't fit with the steady state theory. It fits with the steady state theory in that if something's getting bigger, then it is expanding. But it doesn't fit in, because it says it doesn't fit in with the fact that it has to have come from a singularity, from a single point. So no, it doesn't fit with the theory of steady state, unchanging universe. Because the steady state theory says there's one universe and it's just getting bigger and bigger because there's more and more matter being made. Now, CMBR is cosmic microwave background radiation. And that backs up the Big Bang Theory, because that radiation is thought to be the after effects of the Big Bang. That's the radiation that's been given out. Whereas with the steady state theory, it contradicts the steady state theory model, which suggests there was no beginning of the universe. So now that's why Big Bang Theory is currently the accepted model. But with science, it's constantly developing. So what is considered to be correct 5 years ago, in 5 years' time, there's another theory that comes along and shows that that was incorrect, and this new scientific theory is correct. So at the moment, Big Bang Theory is the one that has the abundance of evidence backing the theory, meaning that it's quite widely accepted. But as scientists keep researching, there'll be many more theories to come. Okay, redshift. I did explain it, but let's explain it in a bit more detail now. What is redshift and how does it support the Big Bang Theory? An increase in light wavelength has been observed in most distant galaxies. So an increase in light wavelength has been observed in most distant galaxies. So the light's wavelength has increased compared to what it should be. So when you look at the light's wavelength, we've already studied that it's between 400 and 700 nanometers. Now, if it was observed to have been stretched, that indicates that the galaxy is further away. And that means they're moving faster. So the further the galaxy away, the faster it moves away from us, resulting in a greater increase in the observed wavelength. That means the wavelength's been stretched even more. So the phenomenon is referred to as redshift. Now, the detected redshift proves that the universe is expanding, which supports the Big Bang Theory. So galaxies tend to show redshift. And galaxies closer to our own show some redshift, but the ones that are further from our galaxies show greater redshift, meaning the ones that are further away from us are moving away from us at a faster speed. The ones that are closer to us are moving away from us because they're showing redshift, but not as fast a speed as they're not expanding and moving away from. Sorry, they're not moving away from us as quickly as the ones further away. So if you see a greater redshift, it means it's moving away from us at a faster speed. Now, galaxies exhibit a redshift. Distant galaxies display a larger redshift. The movement of galaxies away from us indicates that the universe is expanding. So, and the further a galaxy is, the faster it appears to be moving away. This suggests that the universe originated from a single point. Okay, because what the point is that if something's expanding in all directions, then it must have started expanding from one place, from one point. So, Big Bang and steady state. So both theories state that the universe is expanding. This means that redshift supports both theories, as we mentioned before. Okay, what is further evidence for the Big Bang? It's cosmic background radiation. And that's cosmic microwave background radiation. That's what CMB stands for. It indicates the Big Bang's existence. Now, it is believed that radiation remnants from the Big Bang are now spread across the universe. And no, that's fine. Let's move on. So, life of a star. How is a star formed? Dust and gas are drawn together by the force of gravitational attraction. So initially, you would have a supernova, meaning another massive giant of a star explodes. So now all this dust and this kind of star matter, all these elements are spread across quite a significant distance. Now what starts to happen is dust and gas are drawn together by the force of gravitational attraction. And once you've got one ball of dust and the gravity starts increasing, the gravity increasing starts pulling more dust and more elements towards itself. Now, in fact here, you'd assume that it gets bigger and bigger, but actually because of gravity, it gets smaller and smaller because now the gravity makes it a really small, denser and smaller, smaller and denser, smaller and denser, smaller and denser as it increases in particles, in the number of dust and elements that it attracts towards itself. So the more dust and elements that it attracts towards itself, the smaller and denser it becomes. Now, this then becomes a protostar. So you can imagine all these particles colliding into each other causes a massive amount of friction until we now start to get an increase in temperature. And at that point, it's not really releasing any radiation, but we start to see little specks of light. So as the mass increases, the heat increases, then the pressure also increases. And the heat and pressure eventually reach such extreme levels. Now, before it reaches extreme levels, the particles rubbing against each other start to cause flickers of light until the heat and pressure eventually reach such extreme levels that nuclear fusion begins to take place. Now, fusion is when, and we're going to cover this properly tomorrow, is when two hydrogen nuclei combine to make a helium nuclei. Now, there's more to the process, but that's the crux of it. And the amount of energy released from that is massive. That's what sustains the sun. Now, if we look at this star here, now as soon as nuclear fusion starts, that's when we reach main sequence star. That's what our sun is at the moment. So initially, we would have had lots of gravity pulling everything inwards, but now that fusion has started, we have an opposing force. We've got a force pushing outwards. So the star's now transitioned to a main sequence phase of its life, but gravity pulls it inwards. The energy released from fusion, the actual reaction, the explosiveness of it, pushes everything outwards. And the balance between those two forces, the gravitational field strength and the fusion, is what gives the star its size. So what are nebulas? Nebulas are vast clouds of gas and dust found in the expanse of outer space. So when you have a supernova, that's what's left, vast clouds of gas and dust. So when a star explodes at the end of its life cycle, that's what will be left behind. Some of that will become new stars, some of it will become planets, depending on how much dust and elements come together. So if a nebula is sufficiently large, it can collapse under the force of gravity, leading to the formation of a protostar. So this we've already covered. That's what we've already explained. Now, a protostar is the star created from the dust found within the nebula. So now everything's just going to explain what I've explained in a bit more detail. Nuclear fusion has not commenced in this star as of yet. Instead, we see bits of light flickering from it just because of the sheer amount of friction that's occurring. Now, what will happen to our sun once the stable period has ended? So eventually the supply of hydrogen fuel will have become exhausted, as it'll finish. So all the hydrogen will have fused to make helium. Like I said, there's more steps to that process. Anyone that does physics at A level will go through that in a lot more detail. The temperature will drop as its luminosity rises. So it becomes brighter, but the temperature starts to decrease. And it becomes a red giant. The temperature will rise while the luminosity will diminish. So our current now the red giant, the temperature is rising, but it start to become duller. Sorry, I've explained this a little bit wrong. No, I've not explained it wrong, but let me just re-explain it so it makes more sense. So the supply of hydrogen fuel has been exhausted, and that is being exhausted within our current star. So the temperature, as it gets used up more and more, the temperature drops, but it gets brighter and brighter. Eventually it becomes a red giant. Once it is a red giant, the temperature will start rising, but it will become less luminous until it becomes a white dwarf. And then eventually it cools down and transforms into a black dwarf. So how does a star remain stable? There are two forces that are balanced in equilibrium. Gravity, which pulls it inwards. So this is what I was showing you before. Gravity pulling inwards. And additionally, radiation pressure generates a force that causes the sun to expand. So we're talking about the force that's released due to fusion. That pushes outwards. And it's the balance between the two that gives us the overall size of the star. Now, how does a star remain stable? A star experiences a life cycle that is influenced by its size. The fusion process that occurs in stars generate all of the naturally occurring elements. So once hydrogen has reacted to become helium, helium will start reacting with some of the hydrogen as well, and to start making lithium. Helium will react with some more helium, that will make beryllium, and so on and so on. So as fusion occurs between different elements, we make new elements. And some elements are only made during supernovas. Elements that are heavier than iron, are formed during supernova explosions. And the explosion of colossal stars are known as supernova, and that disperses elements across the universe. So if you look at our star, you've got your stellar nebula. The nebula is because of the supernova. So our average star becomes a red giant. Now with the red giant, what starts to happen is
as it gets bigger and bigger, the central part becomes denser and denser, and the outside part becomes a gas. Then the gas is separated. It separates from the mass, and the mass that's left behind is what we call a white dwarf. Now, if you've got a massive star, that becomes a red supergiant. Now, it's the red supergiant that's supernovas, and that can either become a neutron star. It releases lots of gas to become a stellar nebula, or it can become a black hole. Now, when we say black hole, what that actually means is there's no longer any fusion happening here when it goes from supernova to black hole. Instead, all that matter that would have become a stellar nebula is now in a pocket of gravity, and gravity is pulling it inwards. So it's not as big, but gravity it means there's so many particles, so much mass there, that it pulls everything towards itself. So when we look at the Milky Way, and it's got this kind of wavy look to it, the reason it's got that wavy look to it is right in the center of our Milky Way galaxy is a big black hole. So even our individual galaxies would be orbiting that black hole until eventually everything will end up as part of that black hole. Or that's one theory anyway. So what will happen to stars larger than ours sun once its stable period is ended? So the hydrogen fuel will become depleted. It transforms into a red supergiant. It will subsequently erupt and supernova, dispersing elements throughout the universe. The fate of the star will be determined by its size. So it will either transform into a neutron star, which it spins really, really fast, emitting small amounts of light, or it'll collapse into a black hole. What is a black hole? A black hole is a dense entity formed when the core, so this is when the core of the massive star collapses under its own galaxy. So at this point, fusion has come to an end, and gravity is just pulling everything inwards. And the massive star collapses under its own gravity, and hence we end up with a black hole. It's no longer giving off light, it's just a well of gravity. The entire mass of the star is compressed into minuscule space. Because the gravity is so strong, it's pulled into a tiny, tiny space. And the gravitational force is so intense that not even light can break free from its pull. So now that dense entity, black hole, starts pulling everything towards itself. So including galaxies. But don't forget, just like the moon and centripetal force, it's just going around in circles. But it will get closer and closer until eventually it's taken up and becomes part of the black hole. Okay, now this is the last part of topic 7. Observing the universe. How have the methods of observing the universe changed over time? So the initial observations of our universe were conducted through the naked eye. Not sure we don't need to say it's naked. Just the eye, I'm sure, would be sufficient. So in your exam, you don't need to say that the eye is naked. Just writing eye is sufficient. Now, with the invention of the telescope, observations became much more detailed, allowing us to see objects that are significantly further away. So, with using the principles of magnification, we started to make telescopes that magnified images in front of us using light. So telescopes have evolved significantly, enabling us to observe objects from much greater distances now. So certain modern telescopes are now capable of detecting various electromagnetic spectrums, not just visible light. So like I said before, our eyes can only detect visible light, but we've now made devices that can detect other types of electromagnetic parts of the electromagnetic spectrum as well. So now we can identify radio waves, and just in Manchester there's a place called Jodrell Bank, which used to have one of the world's biggest radio wave telescopes. You can have infrared telescopes, X-ray emission telescopes that allow us to see radio waves, infrared, X-ray emissions from far off stars and galaxies. And if you Google this and you look at just to see what different stars look like, when you look at it from a radio wave perspective, an infrared and X-ray emission perspective, if you then compare it, if you get it to, as you look at it as superimposed, it looks amazing. It is just such a beautiful sight, and it just shows how amazing the universe actually is. Many electromagnetic waves in space are unable to pass through our atmosphere, which means if we were to use some of these telescopes from Earth, we wouldn't be able to see them because they don't pass through. So with orbiting telescopes, we can detect parts of the electromagnetic spectrum that the atmosphere blocks. So one of those is the Hubble telescope. It's capable of detecting visible light along with infrared and ultraviolet wavelength. Now, the reason the Hubble telescope was used is because when they were detecting the universe, and you can check this online, when they superimposed everything, they found there was an area where they couldn't see any light. So they made a Hubble telescope, put it into space, and looked into this area, and what they found was lots of bright lights inside. But each of those bright lights, because there were so many billions of light years away, were actually galaxies, meaning each one of those little dots of light that you can see using the Hubble telescope were billions of stars. So again, really, really amazing. So again, if you Google something like that, you can see that for yourself. Okay, thank you everyone. Really appreciate all your hard work today. We've been working now for nearly 4 hours, so thank you very much for joining. I hope you have a wonderful evening, and I'll see you all tomorrow at 1:30. If I'm able to start earlier, I will send a message out on the WhatsApp channel, and I'll try and send a message out through a message on the YouTube channel, and then I'll start topic 5 a little bit earlier. Okay, everyone, have a nice evening. Take care of yourselves. Bye.